A warm welcome to you too, sir. Dr. Ryan Lepicio, President and CEO, National Peanut Board, USA. A warm welcome to you too, sir. Dr. Charles Chen, Professor and Dean, Urban University, USA. A warm welcome to you too, sir. Dr. Haile Desmi, Principal Groundnut Breeder, Eastern and Southern Africa, Senegal CIMET. A warm welcome to you too, sir. Dr. Manish K. Pandey, Principal Scientist, Genomics, Prebreeding, Bioinformatics, and Chair AAGB 2025, ICRISAT. A warm welcome to you too, sir. We are also joined by Dr. Steve Brown, President, Prenut Research Foundation, USA, and co-chair of AAGB 2025, and Dr. Bosho Leo, Chief Scientist and Head, Crop Improvement, Chinese Academy of Agriculture Sciences, oil crops, and research Institute, Wuhan, China, virtually. A warm welcome to you too, Dr. Boschel.
Good morning to everyone, and nice to see you all here offline in person. And also, we are very happy to get connected to all those who have not come because of several reasons online. So, good morning, good afternoon, and good evening, where you are, to everyone. Ladies and gentlemen, esteemed guests and fellow delegates, it's my great pleasure to welcome you to the 13th International Conference on Advances in Arachis through Genomics and Biotechnology, that is AAGB 2025. I'm Manish Pandey, the conference chair, and I'm honored to host this gathering of experts and scholars around the world. We organized this conference way back in 2008. And when we started discussion for this conference in 2024, and many of the colleagues, they mentioned that we came to ICRISAT many times. So we'd like to have we would like to see the different parts of India. And that's how we choose this location for the conference. Um, Unfortunately, many could not join in person here because of, as I mentioned, multiple reasons. Nevertheless, we have arranged online participation, and that's the beauty of technology, which resonates basically that the science is borderless. So we can share our knowledge and experiences with each other and work together. First and foremost, I would like to express my gratitude to each and every one of you for taking the time to join us in person as well as those in joined online. Your presence here today is a testament to the importance of this conference and the significance of the topics we will be discussing. Over the 3 days, we will delve into the latest lineup of speakers, panel, importantly, flash talk from the younger researchers, all designed to facilitate knowledge sharing. This conference are invaluable, and we are grateful for your participation. To our sponsor, your diverse group of experts, and provide a platform for meaningful discussion. That's more important. As we begin this conference, I encourage you all to engage actively, ask questions, share your insights. Let us work together to advance our understanding of genetic diversity in Araiki section and its utilization for breeding future-ready crop groundnut varieties using modern technologies, including genomics and biotechnology, to benefit farmers. Once again, welcome to AAGB 2025. I look forward for a productive and enriching experience over the next 3 days. Thank you.
Thank you, Dr. Manish. Moving forward with the opening ceremony, it's my great honor to invite Dr. Himanshu Pathak, the Director General, ICRISAT, joined by the dignitaries on the dais to officially inaugurate the conference by traditional lamp lighting. Lighting of the lamp is a symbol of knowledge, wisdom, and enlightenment. At times of global challenges, let the light outshine the possibilities of research, innovation, and collaboration, bringing more peace and harmony in global food security. Ladies and gentlemen, please join together for the opening ceremony by lighting the lamp. Thank you. Thanking all for the ceremonial inauguration. Moving forward, Dr. Manish Pandey and Dr. Sanjeev Gupta have tagged along and compiled the wisdom of oilseed breeding genomics and the wealth of research, both in oilseeds and pulses, in the form of book. And we are happy to have the book release on this platform. I request Dr. Himanshu Pathak, along with the other dignitaries on the dais, to release the book here today on this platform. Ladies and gentlemen, please put your hands together for the wonderful compilation of the wealth from oilseeds and pulses. Thank you. Thanking the dignitaries for the wonderful book release. Now I request Dr. Charles Chen, Professor and Dean, Urban University, USA, to give the message for this AAGB 2025. Over to you, sir.
Okay. First of all, I will be very delighted to be here for the AAGB 2025. And also, I know in India, I've been here a few days. You have a long history of the agriculture, especially I think the ACSA, they did an excellent job about the peanut research here. So as a US professor, we would like here to exchanging the information just in the hope so the world can be a peace. And then we also like the fighting for the hunger. And then also I just see the book there is we need to have agriculture resilience. So therefore, I will be glad to be here. Thank you. I really appreciate the opportunity here.
Thank you, Professor Chen. Now I request Dr. Ryan Lepisier, President and CEO, National Peanut Board USA, for the message to AAGB 2025.
Good morning, and thank you so much for hosting us in this beautiful city of Goa. I had an opportunity with my colleagues yesterday to tour around the city and see some of the sights and breathtakingly beautiful place. This is my first time to attend an AAGB conference, and I'm so excited to make new friends and learn so much about what potential the peanut holds for the future. We face challenges, to be sure. For instance, in the United States of America, where I live, I represent America's peanut farmers, and the cost of production is a huge challenge. But I know that our brilliant scientific minds and those of us in the industry who work on the marketing side can come together to find answers that will help to unlock better opportunities for the future. So I attend this conference in hope and look forward to seeing all of the presentations, especially those from our young scientists. I know that they will think of things that we can't even today imagine are possibilities. I want to acknowledge my colleague, Demarquine Houston, who heads our research projects, and our immediate past chair and board member, Gregory Baltz, a producer from Arkansas. And also want to acknowledge Dr. Steve Brown, who leads the Peanut Research Foundation. He could not be here in person, but I know that he has been very much involved with this organization, and I look forward to working with all of you in the future. Thank you so much.
Welcome to the AAGB 2025, Dr. Lepisier. We are happy to have you, your first AAGB 2024. Let's tackle the challenges in peanut research globally, united in a united front to have a better solution and harmonious solution. Thank you once again. Now I request Dr. Ravi K. Mathur, Director, ICR, Indian Institute of Oilseed Research, Hyderabad, for the message to AAGB.
Very good morning, everybody. Honorable Director General, ICRISET, earlier our ICR Director General, Dr. Gupta sir, Director Oilseed Groundnut, and colleagues on the dais. Very happy to be here. Definitely, genomics and biotechnology needs to be integrated with the conventional breeding. People cannot wait for a long time to come out with the product. By the time product comes, it may be of little use. So these have to be integrated. I'm a conventional breeder, but at the same time, this technology, whatever I've developed on genomics and biotechnology, they will assist with conventional breeding in speeding up the process of development of cultivars and so on. So thank you very much, Chair Dr. Manish Pandey, for inviting me to this program. I wish that what recommendations come, this will not only for the areca particularly, but for other crops also, they will be useful. Thank you. Thank you very much.
Thank you, Dr. Mathur. Thank you for mentioning of breeding combined with modern biotechnological tools, driving the varietal release space globally. Thank you for this message. Now I request Dr. Haile Desme for the message to AAGB 2025.
Good morning. My name is Haile Desme. I'm actually from Western Central Africa, based in Senegal. When she introduced, she said I'm from East Africa. Well, AAGB is an important conference for groundnut community. I have attended a couple of them. Progresses in groundnut genetics, genomics are reviewed in detail, and there are a lot of learnings. It's nice to see familiar faces and new faces. Thanks, Manish, for organizing this year's conference. I was with him when he pitched for this conference. And it's nice to be back home. I've been in India for a couple of times. I did even my I did my master's there. So I wish us a successful deliveration of this conference forward.
Thank you, Dr. Haile, coming all the way from Senegal. Welcome home again. And thank you for sharing your insights on peanut research. We are looking forward for a few more. Now, Director Research ICRISAT, the message for AAGB 2025. Over to you, sir.
Thank you, Dr. Shruti, and thank you and welcome to all of the distinguished members to AABG, the 13th edition, to the eminent members of the dais, to all of our colleagues that are joining online. We're so grateful that you are here, as well as to esteemed friends and colleagues here within the room. As a reminder, you will know that ICRISAT speaks of an ethos of transforming dryland agriculture, a vision that talks about prosperity, food security, and resilience in the dryland tropics. And we're very proud of the work that we have done in partnership with so many groups. And I would certainly like to acknowledge the support, the engagement, the key links that we have made to groups across the world. You will also know that ICRISAT works across a number of the semi-arid dryland tropic crops, whether sorghum or millet, certainly groundnut, but chickpea and pigeon pea as well. And over the last 5+ decades have had a real impact in the lives of farmers and communities, releasing over 200 lines in the case of groundnuts to, I think, the most recent number, about 38 countries. But we also know that there are challenges associated with this crop. Weather disease, we've heard from the United States of America with respect to cost production. All of these issues, certainly we are well aware of, whether biotic or abiotic pests. The exciting message that I think we will hear from this conference is the fact that research and innovation can start to find some of the solution. We'll hear about how we're connecting genomics to agriculture, to food, to nutrition, to human health. All of these are the messages that are important for the groundnut industry worldwide. I think what I saw from FAO that over 32 million hectares of grass that are planted we are engaged very important to one public, and we'll have to work. And I know we'll hear at this meeting are those very innovative in the ways to actually make progress on the many challenges that face groundnut, and of course many of the opportunities as well. And I would just acknowledge I'm very excited as well that we will have several students joining us during this meeting. They also will give very short presentations on the work that they are doing. So just as we've heard earlier in the inauguration, the opportunity to hear the newest and most interesting information that will help to guide our industry, not only today, but in the next decades as well. So once again, welcome to AABG 13 in 2025. It's a great pleasure to have all of you with us. Thank you.
Thank you, Dr. Stan. Thanks for highlighting on ICRISAT's ethos of food security. Research and innovation driving solutions for challenges in crop improvement in dryland farming. A good message and a thought for thinking. Thank you once again, Stan. Now I request Dr. Sandeep K. Behera, Director, ICR, Indian Institute of Groundnut Research, India, to give the message for AAGB 2025. Over to you.
Thank you, Shruti. Hello, good morning to all of you. I welcome you all to this 13th edition of AAGB India. And this is the first time I am attending this AAGB. And thank you, Monish. Looking forward a very successful AAGB this time. And we'll interact more during our session. Thank you.
Thank you, Dr. Behra. Once again, we welcome you. It's your first AAGB 2025. Welcome you, and thanks for your message. Now, I request Dr. Steve Brown, President, Peanut Research Foundation, USA, and co-chair of AAGB 2025 for his message.
Good morning, everyone. I hope you can hear me.
Yes, Steve, we can hear you. Please go ahead.
Great, great. On behalf of the USA Peanut Research Foundation, welcome to AAGB 2025. Sorry I can't be there in person with you in beautiful Goa, India, but I hope you have a great conference. Some of the younger participants there may not remember or know that the AAGB originally started as an international initiative to describe the genome of the peanut plant. It was very extremely collaborative. It had contributions from all over the world, and of course it turned out really good. That amazing peanut genome that we now have as a resource is now being utilized to deliver important traits to peanuts worldwide. While every peanut growing region has its own unique problems, it is my hope that we can continue to collaborate on solving our common problems. We've already made some amazing progress, but many of the problems that remain that are common to all of us are complex and have many environmental interactions. I encourage you to continue to collaborate to solve these complex issues of aflatoxin, of drought tolerance, and quality traits like flavor and nutritional enhancements. So again, welcome to AAG 2025. I hope you'll learn a lot from the presentations that are going to be made there. But perhaps even more importantly, I hope that you'll meet new people and new collaborators that you can work with in the future. Thanks to Manish for organizing the conference and to the many, many people behind the scenes that helped make this possible. Hope you have a great AAGB 2025.
Thank you, Dr. Steve, for providing the overview of AAGB, the history and the progress it has made over the years. Collaboration, a best solution in the new era of challenges, tackling aflatoxin and the quality issues in peanut. Thank you once again, Steve. Now I request Dr. Bosher Liu, Chief Scientist and Head, Crop Improvement, Chinese Academy of Agriculture Sciences, Oilseed Crops Research Institute, Wuhan, China.
Thank you. Good morning, ladies and gentlemen. This is a warm greetings from Wuhan, China. Firstly, please accept my warm congratulations to the inauguration of the AAGB 2025 in Goa, India. As everyone knows here, the AAGB has been an international platform and a mechanism for advancing frontier and basic research which could support the sustainable development of peanut industry in the long term. It is very clear that the increase of global peanut production by about 17% during the past 10 years is highly related to the market progress in research, including the genetic enhancement worldwide. So here I would like to express our thanks to EcoResearch, to Dr. Manish Pandey and his team in organizing this important conference. I would also like to express our appreciations to other members of the organizing committee, including those from the United States and China, for contributing to the AAGB in different ways. For this time, even a lot of our Chinese peanut scientists had already planned to travel to Goa for a meeting. Our application, however, for the visa is still in Beijing. The Indian embassy in Beijing, it's easy for us to get a visa timely. However, we are very happy to enjoy and participate in the meeting by this virtual way, and very happy to see Dr. Pasak, the new Director General of EcoResearch. We're looking forward to meeting you somewhere, and some older friends, including Dr. Steve Brown, Dr. Bao Zhuguo, Dr. Charles Chen, and even my older friend, Dr. Shyam Nigam, the former President Program Operator of EcoResearch. Finally, I wish all of you have a nice day in Goa, and have a nice and great successful meeting. Thank you very much.
Thank you, Dr. Leo. Aptly said, the greatest brains of peanut research are here on this platform under one umbrella, here for more networking discussion and way more solution for tackling the challenges in peanut research. Thank you, Dr. Leo. Now I request Dr. Sanjeev Gupta, Assistant Director General, Oilseeds and Pulses, ICR India, for his message to AAGB 2025.
Esteemed dignitaries on the dais, Dr. Steve, Dr. Koshu online, distinguished participants of this important 13th Advances in RH Genomics and Biotechnology Conference being organized in Goa. Warm greetings from Indian Council of Agriculture Research. Really, it's a great occasion, and at the outset, I would like to express my gratitude to ECRISAT and the Peanut Research Foundation for organizing this important event here. Arachis is a rich genus, and two of its wonderful species could evolve an amazing species which we thrive as a groundnut or peanut, which is being cultivated globally for the oilseed production. This peanut is really an amazing crop, which provides more than 40% of the oil on an average and 20% protein. The truly oilseed crop, which provides the highest amount of the oil content. This is the crop which is very unique in its biology, very unique in its physiology, very unique in its adaptation, and very unique in a polyploid genome. Really wonderful. I'm really emotionally attached to this crop because my father has been a groundnut breeder throughout his life. He was engaged in developing good varieties of groundnut. So I have personal affection with this crop. I think a lot of work you have done, particularly in the genome sequencing. 2019, the genome sequencing of this wonderful species has been published. A lot of genes are there on the domain. So genes can help us to resolve some of the problems which are still unresolved, particularly the aflatoxin resistance, the drought tolerance, and some of the things which are very important, creating resistance against key diseases. This group has provided a wonderful opportunity to make our oil quality good in groundnut that could be possible through the use of genomics. Biotechnology, a lot of work is going on, and I'm very happy and sure that the good transgenic transformants will be developed, which could yield some of the key traits which are very important, need to be infused. I wish that this important meet will be very fruitful and purposeful. Thank you very much.
Thank you, Dr. Gupta, for always supporting the message, and glad to know that you yourself are the prodigy of groundnut breeder. Arachis is a rich species to explore globally as an oilseed as well as protein source. Thanks for walking us through the science of groundnut, the glory it brings into the dryland agriculture. Thank you once again. Now, I request Dr. Himanshu Pathak, Director General, ICRISAT, for his inaugural message for AAGB 2025. Over to you, sir.
Good morning, everyone. My colleagues on the dais, and who have participated online. I think there is a large number of people who have joined online. So first of all, let me welcome each and every one of you in this very historic and happening city of Goa. Goa is one of the most attractive cities, not only of this country, but I think of the whole globe. You know, the spelling is GOA. Go there and you will find it amazing. That's what Goa is. Again, let me welcome all of you for this very important international conference of AAGB. My colleagues already talked about it. Dr. Sanjeev Gupta, our ADG from ICAR, mentioned about his emotional connect. Let me tell you, I think many people are emotionally connected to this amazing crop of groundnut. It is not only a very rich source of oil, protein, tasty, healthy, a matter of livelihood for millions of farmers, particularly the dryland farmers, what my colleague Sten was mentioning about, grown all over the world, contributed immensely for food security of world and also for this particular country, and has got a potential to serve much better, contribute in a bigger way for food and nutrition security of the world. The ground on of our mandate crop, as it was mentioned, so far ICRISAT has contributed in a big way by releasing more than 100 varieties across many countries, again, more than 50 countries or so, and contributing very closely, working very closely with our NARS partners, National Agriculture Research Service partner, whether it is in India, whether it is in African continent, and whether it is many other countries. So ICRISAT has been working very closely with them. And today I'm really happy that I can see my ICAR colleagues, ADG, Dr. Gupta, two of our directors, Dr. Mathur and Dr. Bera, and of course, many of our colleagues from ICAR are already here. Many farmers, many scientists who are working on this particular crop for the last many years, students in particular, so who are also joining. So it's a really good combination of all of us joining here to discuss some of the issues, some of the challenges, and to find some solutions for addressing all those challenges of groundnut, not only through genomic or biotechnological tools, but also discussing some of the other. Now, friends, as I mentioned, that this crop is so important globally. And over the years, we could really achieve quite a good amount of success in terms of increasing the productivity of the crop, also increasing resilience to a certain extent, increasing or improving the quality of this crop, and also helping the farmer. But then, as we all know, that there are several challenges which this particular crop is facing, and also the farmers who grow these crops, they are also facing. If I would like to mention a few of these challenges, one is, of course, climate change. You know, not only this crop, but the whole lot of other crops, and of course, agriculture all over the globe is facing. And problem is becoming so severe. Almost every year we are facing new and new challenges. As you know, 2024 was the warmest year in the recorded history of climate on the earth. All kinds of aberrations, whether it is cyclone, heat wave, cold wave, prolonged droughts, floods, all kinds of aberrations in the climate we are observing, and this crop is going to face all these challenges because it's already grown in mostly marginal areas, the dryland areas. So whenever you are growing a crop in dryland areas, in the resource poor areas, again, climate change is going to pose some additional challenges to this particular. There are a few other challenges and we all really need to address those. Of course, in this conference, we'll be discussing in detail all of this, but few of the issues which we all need to address are followed. Number one is we have to develop varieties which are tolerant to abiotic stresses. And when I say abiotic, drought tolerance as well as heat tolerance, because the temperature is increasing almost every year. Drought is prolonging, and of course, the duration as well as the intensity of drought is somehow not known. So drought tolerance as well as heat tolerance is going to be one. This crop also suffers from several biotic stresses, whether it's insect pest or diseases, and some of these diseases are quite alarming to affect this crop. So probably this is going to be the second area of our work. Third one is, of course, how to improve its quality, particularly its shelf life, saving this from aflatoxin, and also other quality aspects. Because as all of you know, this crop is consumed in several forms. Most of it is consumed in many developing countries as stable crop. So we simply consume the grain. And if any kind of problems come, whether through disease or any kind of chemicals, then it is going to have very bad impact on the health. So this is one. And then, of course, when you go to oil content and all of these aspects. So how to improve the shelf life as well as the nutrition quality of this. And again, we have got some good options of biofortified varieties or some tools by which we will be able to improve the nutrient content, the mineral content, the vitamin content, the good protein content of this particular. This could be the third area of working or addressing the challenges which this particular crop is. Fortunately, we have got now tools, genomic tools, biotechnological tools, which can help us in addressing some of these challenges. Of course, genome editing is one, and probably the breeders as well as biotechnologies all over the world are really looking very, very positively of how to use genomic tools, genome editing in particular, to improve the variety in breaking the yield, which we call as yield barrier. Currently, if you see in this country, on an average, the yield is around 2 tons per hectare or 2.1 tons per hectare. But then there is potential to increase it at least more than 3 tons per hectare or 3.5 tons per hectare. And that we really need to achieve. And using the tools of genomics and biotechnology, certainly we can do that. So this is another area, and then tools are also. But besides biotechnology, genomics, and crop improvement, I think this crop also needs certain other dimensions to improve its yield, quality, and also sustainability. One is soil health improvement. How soil management, agronomic management, nutrient management can help in improving productivity, maintain soil health, and also having long-term sustainability. And again, for enhancing nutrient use efficiency, genomic tools can help. And I was told that there are certain good research is going on of how to increase nitrogen use efficiency or phosphorus use efficiency or micronutrient use efficiency in these particular crop, where there is a tremendous scope for its improvement. Next one is climate resilience to groundnut or peanut. This crop, as you all know, is grown in dryland areas, in the resource poor soil, less fertile soil, less water requirement. And in climate change scenarios, these are the conditions which we are going to see. Loss of soil fertility, high temperature, less of water availability. Now, this crop can really provide good resilience to all of these changes and can help the farmer. But if scientific tools and new innovations come in, then of course our farmers will be really, really helpful because of our intervention. Mechanization could be another area. Of course, not in genomics or biotechnology, but this is in a larger perspective. We also need to see how can we breed new varieties which will be machine friendly for sowing, for management, and also for harvesting and further processing. Again, breeding can help, biotechnology can help, genomic tools can help, because these are all interconnected. And finally, we also need to see how policy changes at every country and also globally, how we can influence the policymakers to come out with suitable policies to help realize all these points which I was mentioning. And of course, there are many other points which all of you will be discussing in this particular international conference. Because policy can play a real important role, starting from providing support or incentive to the farmers, providing many other inputs, procurement of the crop, and also value addition post-harvest technologies and funding, supporting research, which is again friends, as you know, this is one of the most important crops. The has been working on this for last several years, and some of the achievements which all of you know, and I also already mentioned. But ICRISAT now is going to strengthen our research on improvement of groundnut through genomics, biotechnological tools, and also we are now going to establish one center of excellence for this particular purpose. And I can assure all of you that ICRISAT will be really, really happy to work in very close collaboration with each and every one of you, wherever you are. We have got very strong partnership with all our NARS partners, and I'm really very happy to see that our NARS partners are here. And of course, I also led the largest NARS partner, ICAR, Indian Council of Agricultural Research. So I know how strong programs are, whether in our Groundnut Research Institute, whether in our Oilseed Research Institutes, and many other institutes and universities. They are really doing excellent work for the particular crop. So let's strengthen that partnership. Let's again come together to address all these challenges and issues which I mentioned. And before I conclude, let me also tell you just one or two more points. For making groundnut cultivation profitable, sustainable, climate resilient, 2 or 3 things we need to do. One is we need to bring in our traditional knowledge with new science. This crop has been grown by our farmers for centuries. So if there is good knowledge, good practice, and good innovations with the farmers, let us bring those and let us include our new science, new technologies, new tools like genomics, like genome editing, or any other approach which is available with us, so that we will be able to make a very sustainable development for this particular crop. And to do that, our collaboration, our partnership is going to play a very crucial role, not only with ICRISAT, but also with one CG, not only with one national partner, but all other national partners, wherever this particular crop is important. So let us work together. And I'm sure in this particular conference, you will be discussing all of these points, plus many others, in great detail. We'll be really looking forward for the recommendations which will be emerging out of this particular conference. And I'll be very, very happy to see that ICRISAT in particular, and our NARS partners and also one CG in general would be happy to implement the recommendations which will be coming out of your all deliberations. So my congratulations and best wishes to the organizers, particularly Dr. Manish Pandey and all others who have been playing a very key role in organizing this particular event, bringing all of us together. And I'm looking forward for a very fruitful discussion and excellent recommendations to address all the challenges which groundnut or peanut is facing. Thank you very much, and welcome each and every one of you once again in this very important international conference. Thank you very much. Thank you.
Thank you, Dr. Pathak. Thanks for emphasizing how the groundnut secures a livelihood of millions of farmers, alongside contributing for global food security, and ICRISAT's pivotal role in it. Always happy to know ICRISAT's role in groundbreaking solutions for global food security again and again. Yes, we take your message clearly. Collaboration for research, and we are looking forward for integrating genomics and biotechnological tools for increasing the yield barrier to 3 to 3.5 tons, alongside working on quality and bringing mechanization, influencing policy makers, supporting national as well as international policies. Thank you once again, sir. Ladies and gentlemen, here we finish our inaugural talks, but over next 3 days, we'll have an exciting lineup of the keynote presentations, panel discussions, poster sessions, and networking opportunities. This year's conference will highlight the latest breakthroughs in genomics, biotechnology, and breeding innovations that are shaping the future of peanut science. We encourage you all to participate actively, share your insights, build meaningful collaborations that will drive impactful research. Before I conclude, let me thank all the key people who have involved with us for the success of this AAGB 2025 GOA. We extend a heartfelt gratitude to ICRISAT leadership, whose unwavering support has been instrumental in organizing this event. ICRISAT has been at the forefront of peanut research, ensuring food security and sustainability for millions worldwide. A special acknowledgement to the Peanut Research Foundation for their continuous dedication to funding and advancing peanut research and innovation. Your contributions are truly valuable, sir. We also recognize the International Advisory Board, the organizing committee members, and our generous sponsors from Mars, South African Groundnut Association, Bombay Super Hybrid Seeds Limited, Green Crop Seeds, DCM Shriram Limited, Takara, Premas Life Sciences, and Spinco for significant contributions, without whose contribution we would have not possible to have the successful AAGB 2025. Your commitment for peanut research and scientific progress is deeply appreciated. Ladies and gentlemen, please give a round of applause for everybody, including yourself, for being a part of this auspicious and a good platform of the wealthiest, rich minds coming together for networking, discussion, networking, and driving impactful solution in peanut research. Ladies and gentlemen, here I close this inaugural session. Before we wrap from here, certain housekeeping announcements. Kindly keep your cell phones off during the sessions. The conference program and abstracts are available on website. Please do check on. And for any queries, please visit registration desk. We'll be shortly beginning our plenary session at 10 a.m. Now we will break for tea, followed by group photo. Volunteers, please take the audience and the dignitaries to the tea venue and the photo venue. Here we conclude. Welcome you all for AAGB 2025 once again. Thank you one and all. Hello, hello, mic check. Ladies and gentlemen, requesting all of you to move to the photo venue. Kindly please walk. Volume. Hello, hello, mic check. Ladies and gentlemen, please move towards the pool side for the group photo. Kindly requesting the volunteers to escort the audience to the pool side.
दुपारी
Ladies and gentlemen, please requesting all of you to take your seats, please. Shortly, we'll be beginning with our plenary session. Requesting all of you to get in and take your seats, please.
चलेगा ना क्योंकि बीच में थैंक यू
आपका फिर से डिस्कनेक्ट हो गया।
हाँ डिस्कनेक्ट हो गया था
मेरा
चल रहा है एक्चुअली बीच में चेक करना पड़ता है
Speakers of the
इसलिए
sessions are requested to
उनको
hand
तो
over your
बोल के
presentation. Ladies and gentlemen, requesting all of you to come and take your seats, please.
इंटरनेट कनेक्शन इज अनस्टेबल
हाँ आते रहते हैं
18 participants
Ladies and gentlemen, welcome once again. We are moving forward with plenary session, and I invite Dr. Sham and Nigam, the chair of the session, and Dr. Ravi K. Mathur, the co-chair of the session. He'll shortly join us. Before that, I'll hand over to the chair, Dr. Sham Nigam, for conducting the plenary session of AAGB 2025. Over to you, Chair. Kindly requesting all the participants to take your seats, please.
Well, friends, good morning, and I welcome you all to this plenary session. My co-chair will join me shortly, and we will, without waiting for him, we will start the proceedings. And first presentation is by Dr. Bujau. I believe it's online. So
yes, I'm
are you
online.
ready for that?
Yes, I'm ready.
Hey, my friend, how are you?
I'm great. How are you doing? So really
good.
miss you.
Yeah.
So
Are you ready now?
yeah, I'm ready.
Okay, the floor
Can
is
you see me?
yours.
Can you see me?
Yeah.
Okay, it's hello everybody in AAGB 2025. I'm so sorry to cannot make it to GOA. GOA really, it's my idea to have this AAGB 2025 in GOA, but I couldn't make it. And but thanks to the technology, I still can see you, online. And first, I'd like to thank Manish Pandey and his team make this GOA AAGB 2025 a reality. And it's absolutely my joy and my honor to present you AAGB history and significance. I want to move on. Okay, I'm going to talk about these three parts. The history. So we just talk about AAGB, any event, any conference has a history, where started, who started, who used the AAGB, the terminology, for example. Then the significance, the accomplishment we all experienced through those years, particularly evidenced by the publication recently years, the quality and the number. And also I'd like to present you the challenge, particularly to the young people, also to us, and not that young. AAGB is so important. It's my passion. So this slide just summarizes you the history of AAGB. So AAGB really started 2006 in Guangzhou, China, then Atlanta, USA, Equator, India, Bamako, Mali, Brasilia, Brazil, Zhengzhou, China, and back to United States, Savannah, Georgia, Brisbane, Australia, Cordoba, Argentina, Sali, Senegal, Jinan, China. So we had a few years COVID break. So we all experienced that. But 2023, we resumed AAGB 2023 in Huntsville, Alabama, United States. I congratulate you, Manish Pandey. We have this AAGB 2025 GOA, India. But the challenge is, what's next? Where we go from here? That's the one objective I'd like to present to this conference, AAGB 2025. This slide is showing the first international conference of groundnut in Guangzhou, China, 2006, hosted by Dr. Luo. He was the president of Guangzhou Academy of Agricultural Sciences. Just from this meeting, we have this take-home message. Number one, it's international. Number two, groundnut. Number three, focusing on aflatoxin and genomics. So next year, 2008, we had first time created this term, advances in Arachis through genomics and biotechnology. That was created in 2008, Atlanta, Georgia. And this conference, international conference, provided this foundation. That's the goal. That's the objective of this conference, international conference, to provide a foundation for the integration of different research strategies. Second, then create an environment. That environment can foster the collaborative relationship. So we need this relationship. Then lastly, this conference can promote interconnectivity. So this interconnectivity through this international collaboration, research, focusing on those different priorities, like high yield, disease, pest resistance, tolerance to different stresses, and increased nutrition. That were those priorities in 2008. 2008, the third AAGB, that's the first time we used AAGB 2008, created by ECRISAT. And Dr. Rajiv led his team created this AAGB. That's the first time we used AAGB, remember. And also that's the first time we used international conference of the peanut research community. So AAGB is for international peanut research community. So that's the first time we used this terminology. And also AAGB 2008 ECRISAT India, we expand the topics. You can see here, we expanded the topics, expanded the priority. Then from these three meetings, we laid the foundation, laid the tradition. So everything AAGB is a history. So this slide I want to see showing you some the significance of AAGB really is started because of the genome. So 2003, the group of legume research group, they come together, talk about legume genome. The next year, 2004, first time talk about the peanut genome initiative in Atlanta, Georgia. So then you can see here, it's 6 years later, 2010, the first International Peanut Genome Project, 2010 inaugural meeting hosted in Atlanta, Georgia. Then 2 years later, Peanut Genome Sequencing Project launched. Then 2 years later, 2014, to deploy the peanut genome sequencing released. That's the significance. That's the contribution, accomplishment through AAGB. Then this slide can see the AAGB conference continue through those years. The accomplishment through AAGB first, 2015, we published the first software, bioinformatics called SWEEP, to analyze the peanut genome sequence, because the peanut genome is so complicated. That's first 2015 published the first peanut software. Then 2016, next year, we published the first to deploy the genome on Nature Genetics. Then because of that resource, so we developed and produced first version peanut SNP array. Then improved, that's the first version 2 peanut SNP array. In 2019, we published tetraploid peanut genome in USA and China. And because of those accomplishments, everyone here experienced and used those resources and evidenced by the publication. So we think we did it. This is Mr. Valentine. He's the one behind this International Peanut Project, behind AAGB. But his name is not on the publication. So I called him his one unsung hero. So AAGB 2019, I was honored to present this, the path to success. So I think we should celebrate the achievement, and also we should recognize those unsung heroes. Without those, we don't have AAGB, we don't have International Peanut Genome Project. And also we remember, we not just celebrate and recognize, also we win. We are winning for our farmers. That's our duty as a scientist. So this is Dr. Wood. He hosted the first International Peanut Conference in 2006. He was recognized at the 2019 conference. And also Mr. Harvard Valentine was recognized over there. He's another one unsung hero, very important person. And Rajiv Varshini, he also recognized his contribution to International Peanut Genome Project in 2017 in Argentina. So I want to give you, particularly those young people, some number to recap the history. As you saw already presented, that Peanut Genome Project really started 2004. So that 2004, what do we have? So for that meeting in 2004, we talked about a common cultivar. So which peanut cultivar we're going to be sequenced? We're going to sequence. So why Tifrunner? So that year we choose Tifrunner. Really, that Tifrunner not released yet. Tifrunner was called C3424. And another thing is EST. So how we can increase peanut molecular marker quickly, cheaply. So that meeting, I had an assignment to develop EST. So that year, I presented some numbers. Why EST? Why express sequence tag? So why EST? So that's 2004. Just look at the number. At 2004, what do we had? 2004. That year, we had EST 1366. Remember that number? And the total nucleotide that year, 2004, we just had 1,700. So that year presented like this. So traditionally, you clone a gene just like you use a fishing pole. But I proposed this high throughput. That's the high throughput. You threw the fishing line. So instead of fishing pole. So fast forward 8 years later, 2017, I presented this at the Orphan Legume Genome Whose Time Has Come Symposium. So we think peanut genome era has coming. So that year, I can see here from 2004, 1,700 the total numbers. 2012, July, peanut had 283,000 nucleotides. So fast forward, AAGB 2023. At AAGB 2023, one graduate student, PhD graduate student, Ethan Thompson, presented this. Beyond one reference genome, a paradigm shift. Pan-genome-based study is coming. He made this bold statement at AAGB 2023. Pan-genome era is here. So you can see we came a long way. From 2004, only had 1,700 nucleotide sequence. In 2012, we had over 200,000 nucleotides. In 2023, we made this bold statement. Pan-genome era is here. And along with the pan-genome, we have to, as a scientist, we have to produce multi-parent population because a magical population increased recombination, increased the diversity. So last year, 2024, we published the first peanut magic population. And the peanut magic population has 3,187 real lines. So we used about 10% this peanut magic, 310 real lines we call a magical corn. They studied this total peanut magic and also constructed pangenome. This just give you example, the power, how powerful pangenome and the magic population. So this slide is showing you that because of we use the pangenome, we discovered a third FAT2 gene. We named it FAT2C. So this FAT2C really solved a mystery. We had this mystery for a long time in peanut community that why we had middle lake. Why it's not high lake? You have FAT2A and a FAT2B mutant, but it's not high lake. Just because we have this third fat gene, FAT2 gene called FAT2C. It's 1.3 million base upstream FAT2B. That's the powerful of pangenome. So I can give you another example. We used the pangenome, we identified TSWV resistance. That bothered us for a long time. Because of the pangenome, we identified this TSWV resistance caused by this insertion. This insertion is about 64 kilobase. But this insertion has 4 copies, 4 identical copies. Glutamate receptor, because of that, caused this resistance. So next, I want to take a few minutes to talk about the challenge, the tomorrow and the beyond, the AAGB. So I believe we are at the crossroad. So either we rest on the success and the party to celebrate, or we can stand on the shoulder of giants and fight for our farmers. That's the choice we have to make. So that's my dream. I kind of proposing that the next 20 years. So because the last 20 years, we made this orphan legume genome whose time has come a reality. I think that's a reality. We are really ahead of many crops. Before 20 years ago, we're really behind many crops. So second, the global issue to seek global solutions. The aflatoxin contamination, food safety is a global issue. We need to work together to solve this global, to seek a global solution. So AAGB is the best global platform provide this venue. Therefore, together, I believe we work together, we can look forward to the next 20 years to fight for the sustainability of peanut production and the peanut safety and aflatoxin contamination. So I like to use these slides to conclude my presentation and also challenge you, everyone here. Who with food has too many worries, and who without food has only one? So which group you belong to? I can tell you, everyone here, we belong to the first group. We who with food. So therefore, we have this challenge. We have this responsibility to fight for those who without food. They only have one concern, is their stomach. That's our challenge for next 20 years. 20 years for the sustainability for peanut and the food safety and the aflatoxin contamination. Thank you very much.
Thank you, Dr. Bojao. I'm sure you would like to have few questions if the audience has them for their own clarification. Meanwhile, I welcome my co-chair, Dr. R.K. Mathur.
Any questions?
Would you any question or clarification from the floor, or everything is so clear?
If no questions, I do have a question for everyone.
Well, I'll just make one comment.
Okay.
First, thank you very much for taking us through this long journey of AAGB. It has been a long journey. A lot of ground has been covered, yet a lot still remains to be covered. And that is where the future need is there. I'm glad you suggested aflatoxin as a next major issue which can be tackled through international collaboration. We spent a lot of years, almost 30 years, conventional breeding, trying to improve aflatoxin resistance. We could not improve over the sources of resistance. And in one of the publications, I admitted the defeat of conventional breeding as far as aflatoxin is concerned. Now the ball is in your court. You people have to provide the solution through genomic tools. And I hope with the teamwork that you are carrying out here, you would be able to do it. Thank you again.
Thank you.
Next speaker is Dr. Sanjeev Gupta, a stalwart in the area of pulses and oilseeds, currently holding charge of Assistant Director General, Oilseed and Pulses, Indian Council of Agricultural Research. So Dr. Gupta, floor is yours. Tell us where we stand as far as self-reliance of oilseed in India is concerned.
Once again, warm welcome. I think the topic which has been given to me, ensuring self-sufficiency in oilseed production in India, is a challenging task for me. And doing self-sufficiency in terms of oilseed production in India is again a big challenge. Dr. Basu told that it is the time to stand on the shoulders of the giants and fight. The next 30 minutes, I will try to fight to deal with this important challenge of ensuring self-sufficiency in oilseed production in this country. India is committed to seek self-reliance in most of the agricultural commodities by 2047, when on this auspicious occasion of 100 years of India's independence, India would like to see that should be the self-reliance on all agricultural commodities. If we take the history of last 10 years, in most of the agricultural commodities, we are in the comfortable position, whether it is egg, meat, fish, vegetables, and the growth, agricultural growth in those sectors has been so tremendous that we will easily achieve the self-sufficiency in those sectors. Even the cereals in some of the commodities will have the exportable surplus. Now we are working on the strategy how to achieve the self-reliance in most of the agricultural commodities. The pulses and oilseeds, these are the really the sectors where we need to work more. These are some of the estimates that we are working out. It is ICAR, then the Niti Aayog, the National Institute of Nutrition, Indian Council of Medical Research, and we are putting several layers because we have to envision that in those 2047, what will be the scenario of our society, our growth, economy, and other sectors. We are further refining these estimates to take care of. In horticulture sector, we are really becoming comfortable. I think we are growing very fast. Cotton, we are also doing good. And some of the sectors where we can perform better. But in edible oil, achieving self-sufficiency by 2047 really is the bigger challenge. if you see the edible oil sector in this country, India is the fourth largest producer of oilseed after USA, China, Brazil. And in the last so many years, the last 30 years, if you can imagine, the production of edible oil has increased 3 times and more than 3.5 times in last 3 decades. But the consumption, per capita consumption, if you see, that is also increased more than 3.5 times in that period. We could see the yellow revolution, the self-sufficiency in oilseed once, when the technology mission on oilseed was launched by the government of India. But soon after, that was becoming ephemeral, and we could still struggling for achieving self-sufficiency in oilseed. When we are saying that we have increased 3.5 times in the production in 3 decades, still we are relying on 55 to 65% of the dependency on imports for this particular sector. And we are now investing more than $19.5 billion per year for importing of the edible oil in this country. Next, after the crude oil and gold, this is the third largest item of import in this country. But at the same time, I would like to mention that we are exporting the oil meal, oil cake, and some other oil products to the tune of 70,000. So if you take the balance of that economy, I think we are in the comfortable position. And in the future also, when we seek reliance, I think I would put forward that you should have the balance of the particular sector, not only the actual figure that we are importing so much in a particular agricultural commodity. So the first thing I would like to have, if we seek the self-reliance, we should have the mass campaign for reducing per capita oil consumption. It is 3 times we have increased since 3 decades. So if we take pledge to reduce 5 grams per day per capita oil consumption, I think our situation will be much more comfortable. So for that, there is need to have a mass awareness, information, communication, knowledge, and mass use of mass media for a big campaign to reduce the per capita consumption of the oil in the country. Again, we made a survey for the consumption, the pattern of the consumption. About 23% of the edible oil is also going for non-edible uses like foam industry, like paint, varnish, and many other uses. So how to reduce it, how to restrict it? Should we find some other alternative, non-edible oil for that purpose? The second thing can be mainstream some of the secondary sources of the oil seeds, because in oilseed production this year we are really comfortable. We are going to produce 41 million ton of the oilseed, which will be the record production, ever highest production in the history of the oilseed production of this country. And groundnut is also, we are going to produce the record production of 11.3 million ton in this country, which will be the record highest production ever. And this is the latest estimate provided by the second advance estimate of the government of India, that oilseed production in this country will be the ever highest this year, and the groundnut in particular will be really the ever highest production. But we need to also mainstream some of the secondary sources like the cotton seed oil, the rice bran oil, coconut, then palm oil, tree and forest origin. I think altogether the contribution of these important sources of the edible oil is getting down. We need to have some of the efforts to mainstream these important sources, particularly the technology upgradation, refining, bleaching, and deodorization, and the quality improvement of the secondary oils. If we knock out the FAD2 gene in cotton, I think the cotton seed oil can be very wonderful oil for the consumption. That effort is also important. And most of these secondary oils, they are less stable. So can we improve the quality of these secondary oils? That will be more useful for us. And there is always potential from 11 lakh tons to 16 lakh tons from the rice bran oil. That is also important. But again, the quality of the rice bran oil need to be improved. Advancing productivity is the only answer for most of the situations, whether it is groundnut or other oilseed crops. If you see the situation, the India is now in the groundnut, the average productivity is 2.1 ton. And in one of the states, in Tamil Nadu, we could reach up to the 2.6 ton. In the farmers, some of the farmers, we could see that they are harvesting more than 3.5 ton. And in one of the farmer in our Uttar Pradesh, he has harvested more than 4 ton. So that is the potential of groundnut varieties which we are having, and will exploit fully through various programs to advance the productivity, closing the yield gaps, and realizing the genetic potential of the important crops. But at the same time, the harnessing new science, new breeding techniques need to be used, like fast-track breeding, genomic selections, gene editing, cis transgenesis, gene silencing, gene integration. I think those are the new sciences need to be infused for improvement of the groundnut, not only the yield, but the quality and other parameters we need to resort. And I'm happy this particular conference, where the lot of genomic informations have been developed, will be dealt and to be exploited for some of the important objectives of the groundnut breeding, and those will be really helpful for other crops also. So those need to be taken care of. We are constantly requesting our government create a oilseed development fund for research, a specific fund for research, by levying the 0.5% cess on the import. This amount can be easily made for this fund. And for this fund, we are also requesting for establishing a center of excellence for genomes and genome editing, creating breeding hubs, speed breeding platforms, and import of the new germplasm, which is available elsewhere in the country, and the hybrid development program, some of the crops where the hybrids are important, like mustard, sunflower, castor, and Niger. Biotechnology and genomics, they are playing greater role in improving crops. If you see, this is the old data up to 2020, but now 186 million hectare area under oilseed is under GM crops. It is nearly half of the total oilseed production is in different countries. 78% of soybean is GM in the world. 29% of the canola is GM in this world. And all canola from Canada is GM. So this is one of the technology which need to be resorted. But again, we need to see that research on this front should not be restricted. It should continue. And we need to convince our society that how these technologies are important, because I believe in last 20 years, there was not a single case of health hazards because of taking GM oilseeds. It is all depend upon how we convince our society and take up and accept this important technology, which can be a boon for interbreeding. Government of India, in the recent initiative, established the genome edited platform, and for different crops, we have prioritized the traits, we have identified potential candidates, and we have identified the institutions, organizations, collaborations to resort this important technique of genome editing. It is an alternative technique for the transformation. Transformation is there, but we can use STN1 and STN2. These are the two events which are excluded from the restrictions that can be used very carefully, and we can achieve some of the difficult objectives, particularly if you see the groundnut. We have done something good through marker-assisted breeding, improving the high oleic acid content, but low phytate, drought tolerance, aflatoxins, and some of the difficult objectives which are not possible. I think our genomic information will be helpful to identify gene, and if we can identify gene, whether it is orthologous or heterologous, can be traced back and can be edited to improve our crops further. Likewise, in other crops also we have identified genes, prioritized traits, and the techniques we have also focused, and I think some of the things are coming up. In mustard, 2 of the genome edited products, they are already in the trials, and with the fast-track breeding methods, fast-track testing methods, we are hopeful to bring these 2 genome edited projects in next 2 years. Recently, last year, I think our Honorable Prime Minister dedicated 109 high-yielding, climate-resilient, and biofortified varieties to the nation. And some of the objectives which we are taking care of in our breeding program or through the use of genomics and marker-assisted breeding. But Prime Minister, in his interaction, he was focusing about 2 important things. The first, he was stressing, can you increase the oil content in groundnut? Our answer was, on an average, we are having more than 40% oil content, but we are having the varieties which are on the farmer's field, which are giving 53% of the oil content. His request was, can you make it 59? A very, very difficult objective to pursue, particularly if you are taking a 1% increase in the oil content, you have to resort several cycles of the recurrent selections, and then it will be possible. It's a really difficult task. Then again he asked, no, if it is possible, can you do it at least 2% more, 55% oil content in the groundnut? And second stress was that can you improve the quality of the oil groundnut? So we are having the increased shelf life because of the high oleic acid content. But again, some fatty acid composition you need to alter to make this oil more good, more important, more safe, and more stable. So these are the 2 things in groundnut he stressed that you should do it. And aflatoxin is also was in his mind because we are exporting some of the quantity of the groundnut produced in the country to European Union, Middle East, and other parts of the country where the aflatoxin is a real issue. We need to see and we need to organize our research in such a way. I think in our system and the globe, I think we need to make a good collaboration to find some way to have the resistance against aflatoxin. So these are some of the things he stressed, but most of the things in our objective, it is there. And in the recent budget announcement, the government of India has allocated a sizable fund for high-yielding seeds development, and that is the National Mission on High-Yielding Seeds. I think, and in that, the groundnut will be a most prioritized crop for that mission, and we need to use some of the funds, some of the facilities that is being created under this, and that will be really helpful to see how groundnut is moving in this country. These are some of the if you see there is huge yield gaps in this in our oilseed crops, and particularly if you see the groundnut, the 40% yield gap is there because our crops are growing in a very suboptimal management conditions. There is need to focus for the secondary major nutrients and micronutrient deficiency in the soil. We need to provide better nutrition to the crop to produce more. We need to see that how the key diseases and pests are affecting our crops. I think if all we can do it, our target is we can reduce this yield gap with the good agronomic practices. We can close this yield gap up to 25% in the groundnut. Likewise, in other crops. So we need to address the secondary major nutrients and micronutrients. We need to address the life-saving irrigation. We need to use the customized fertilizers. We need to use the ICT for improving our crop yield in the farmer's field. The efforts have been made in other crops, in groundnut also, for improved access to the quality seeds. But again, 72% of the seed is coming from the private sectors. We need to see the quality of the seed that is being given to the farmers. If we provide the good quality seed to the farmers, I think we will be able to achieve some of the yield targets which we are envisioned in our program. In this country, we need 46 lakh quintal of the seed of the groundnut. It is a high volume crop. So the organized sector and the formal sectors, they are not able to provide the good quality seeds. And if we are resorting to the private sectors, still the quality of the seed is another problem. So we have to find out some the alternative seed system for the groundnut, because it is a high volume crop. You need to give 120 to 130 kg per hectare seed to the farmers. I think it's a very difficult task. So we have had a provision of the seed hubs. We have organized the seed hubs. So up to now, 35 seed hubs were there. But now the government has again sanctioned 30 more seed hubs that will be helpful for the groundnut seed production program in this country. We need to explore the newer niches of production, because whatever the area is there, where the crop is being grown, and it is also competing with other crops, because most of these crops are grown in the rain-fed ecology, and in the rain-fed ecology, pulses are also grown. If you are increasing area under one commodity, there will be a decrease in area in other commodity, and country is facing both the things, the deficit in pulses production and the oilseed production. So we need to manage somehow and explore near areas of the production. So not only the rice fallow, some of our oilseed crops can be fit for the rice fallow condition, but there is large area of potato fallow that can be also resorted for the production of the our oilseed crop. Particularly, we have identified some of the areas in the groundnut that can be tapped for the newer area. Those are the newer area after potato fallows, and that will be really good areas where the productivity of our crop will be very high, because the potato cannot use all the DAP, all the nutrients which we are giving. The residual nutrients can be very helpful to our crop. Our 11 green revolution states, they are facing a lot of the crisis of the sustainability. And I think if we are including pulses and oilseed crops, that will be the part of our diversification campaign, and we can increase more area under some of the crops. The higher import duty regime we need to adopt, because this year, when we imported large scale soybean, the domestic prices of the soybean reduced so much that farmers were really in the stress. Same thing is happening in the groundnut, because a good crop was in the Brazil, Argentina for the soybean, and similarly for the groundnut. So there was a competition and intake of the lot of the produce which are cheaper in the rate from the outside be imported. If we increase the import duty, and that import duty, as I suggested, if we levy the 0.5% cess on those imports, we can create a fund of edible oil development fund for research of worth of 2,000 crores that can be established in this country, that will be helpful in having good science in our oilseed improvement and production program. We are also exporting some of the crops like groundnut, the same to the other parts of the country, and we are earning more than 10,000 crore rupees every year. So we need to improve the exportable quality of the crops. And our groundnut, as I told, the aflatoxin is very important, then high sucrose content, low free fatty acid content, low peroxidase values. I think if these are some of the things we can take care in our breeding program, we can make much more exportable surplus of the groundnut. And again, the bold and uniform size is small and round and tinged seed coat, more small with bright red seed coat. These are some of the demand of the export community, and they are asking us to breed some of the varieties for with these traits, and we can do it. Really, it will be helpful for us. This taking campaign of oilseed production in this country is not easy task. We need to invite cooperatives and corporates, because the lesson from our first technology mission, we invited NDDB, a cooperative model, and when they came from production to packaging, they had the model, and that model, Hara brand, they have adopted. That could be really a trigger point for achieving the self-sufficiency in those areas, those days. And now I think this is time to invite more cooperatives and corporates for taking edible oil sector in this country. Now, the Government of India this year has launched the National Mission on Edible Oil, and it is to the tune of $1.3 billion. And in this important mission, we are targeting to increase the production to the tune of its double. And we are having the model of developing a value chain clusters, where in those clusters we'll have all demonstrations of high yielding varieties, demonstrations of high end technologies, good POP, and we will develop extraction units also. We'll have a total cluster from production to marketing in those clusters, and that will be 600 clusters in this country in 387 districts. So this mission has been launched, and in this mission, finally, Government of India has agreed to provide funds for research also, and their stress is importing good germplasm from anywhere it is available in the world. So we need to import, and they will support through funds to importing good germplasm, and that will be taken care of in this program. Again, we have the National Mission on Edible Oils, oil palm particularly, and it was launched in 2021, and now the results are on the field. So we are hopeful to have a good amount of production in this country in oil palm, because 40% import, and this year we imported 49% of the total import is oil palm. So we need to have some program for the oil palm, because oil palm is a crop where you can have a high oil production, and that can be achieved also. Earlier we were thinking that in this country oil palm will not be successful. Dr. Mathur is there. He was the director of oil palm research, and now he has convinced us that what kind of the plantation we had in this national mission on oil palm, really it will bear the fruit. This country was always serious in oilseed production program. And if you see in the first program on technology mission, it was launched in oilseeds in 1986. Then we had the program in 1990, then 1995, 2005, 2012, and now 2021, 24. Government of India is always with the newer programs. And I think some of the programs have been very successful. And we are hopeful that two missions, National Mission on Edible Oils and National Mission on Oil Palm, will be successful, and we will achieve not self-sufficiency, but will reach to the near self-sufficiency in coming years. Our target is 2031. So we will reach to the near sufficiency, self-sufficiency in oilseed sectors. Thank you very much. This is all about we wanted to share. Thank you very much.
Thank you, Dr. Gupta, for taking us through the journey of oilseeds in India and also giving an overview of how the things have evolved. Making country self-sufficient in edible oil remains a dream. The only period when we achieved near self-sufficiency was when there was TMOP, Technology Mission on Oilseed and Pulses. We nearly achieved self-sufficiency, and the program was led by late Dr. M.B. Rao, who was Additional Director General ICAR. But then things started slipping back again, and things went to square one. So the new initiative is coming. More or less components are the same. Maybe if you hope that we will succeed this time. We have failed once, we should not repeat the mistake. You made a brief comment about reducing or having a pledge of reducing oil consumption for 5 grams per individual. The problem is oil is the one which gives taste to food. Now, but we want to eat the food fried in the air, those who are is going to be difficult to be made. But anyway, what pursuing that line of approach, one thing that I have always found in oilseed, on legumes, including oilseed and pulses, importance of agronomy has not been properly recognized. And I'm a firm believer, if you improve agronomy, you can double the production with the existing varieties. When I was posted in Africa, and when after doing 1 year there, then I wrote back to Ikrit, you have appointed the wrong person here. Instead of a breeder, you should have appointed an agronomist to improve the agronomy. So they said, you are firing yourself out. I said, I'm willing to do that. So agronomy is the one where we need to give more emphasis if we really want to improve the situation. This is what my few observations. And
Thank you very much, Chair sir. Thank you, ADG sir, Gupta sir, for a wonderful presentation. Only 4 or 5 points I want to make, sir. The thing is, it is high time to go for the multiple cropping or intercropping system approach, or rather farming system approach. Crop as a monoculture, monocropping will have always limitations. So again, while working on groundnut during 93 to 2000, we understood that while genotype interaction we are talking in intercropping, just like that, giving a blanket recommendation that groundnut can be intercropped in this and that crop, especially castor, will not work. Whatever varieties we release, definitely their response in intercropping may be different. Second thing is, sir, while talking at the national level, always policy decisions are very important. Policy decision from year to year changes that affects the program adversely. At present, we are, sir, looking after the sunflower revival program. Now also, non-lifting of seed is a problem under such a beautiful situation of oil seeds. So hand-holding with our farmers or stakeholders for 4 to 5 years minimum is required so that it becomes a sustainable approach. Otherwise, today be happy with that scenario and tomorrow again there are problems. Third point is some good area needs to be released for oilseed sectors also. Otherwise, these, sir, marginal and sub-marginal land under a poor resource, poor conditions, they are grown. And there definitely instability is a problem. Fourth is, sir, says, rightly said, ADG, sir, while we are working on oil palm also, sir, we have suggested same thing. 0.5% is not a big percentage. if that fund is available for R&D, it can support like anything, which is a link really required. And quality seed are very, very important, sir. 28 to 30% of the productivity is generally governed by the quality seed. And that needs to be really made available. If you talk of areca or groundnut or any crop, water stress is important. And again, water stress is much more difficult task. It may be early water stress, mid-season stress, late season stress. And now if it is late season stress, again, temperature stress is confounded with that. So definitely these genomic tools and biotechnology is the answer where favorable genes can be brought in one population or one cross or one variety, maybe through developing magic populations, and then using the speed breeding facilities, using the marker assisted breeding, and so on and so forth. You know better that one. That is really required. And we cannot wait for 10 to 12 years for developing a variety. No, within 3-4 years, products should be out. So that it is a big challenge. Still, leaf spot is a problem. Rust is a problem. And now just now you're talking with the young chap, stem rot, they are getting breakthrough. So these things can be tackled through genomic, definitely selections and genomic technology, which can assist conventional breeders so that the cycle of breeding cycle can be reduced and the product can be out in no time. Definitely consumption pattern is important, but non-edible use, sir, Gupta sir, non-edible use is also important. So let's think of the crops which can support the oil which is used for the industrial. They also require, otherwise this edible oil will keep on going towards a non-edible usage also, and always will feel a problem of shortage of the edible oil. And as far as yield gaps you have seen, yield gaps are very wide. And probably I feel that if our crop cultivations are 75% under control, maybe 25% nature is important, then things can change. Otherwise, leaving the crop to the rainfed system or dryland ecosystem, it is not going to make a dent. And lastly, sir, what Chair told is really correct, that production technologies are the most important. And probably in the process, now good varieties are there thanks to Dr. Bera and team Indian Institute of Groundnut Research here, who have come out with very high olic varieties of Ginnar 4 and 5, and further they are in pipelines. But then this agri-techniques also are very, very important, which are location specific again. And India being a big NARS system, where so many ACRP centers are there, this issue can be taken care easily. So thank you very much, Chair, for giving me the opportunity to interact. Thank you.
Any comments or observations from the floor? Sir.
Thank you, Dr. Gupta, for such a comprehensive overview. You've answered all of my questions, but maybe I just would like your observations about the future. I know many of the groundnut breeding groups are working on things like high oleic materials and others. Do you see, especially in India, a future where producers will also be rewarded for higher oil content or different fatty acid profile, so that really the producers will also benefit from these increases in both quality and quantity of oil? Thank you very much.
We are making efforts and convincing government to give premium prices for premium quality seeds, premium quality varieties of the crops. In our country, the high oleic acid really that has been accepted in a big way. We are told that your Ginnar 4, Ginnar 5, that has been developed through the collaboration of ICRISAT and our Directorate of Groundnut Research, really finding place in the farming community, and they are fetching premium price for that. the Ministry of Consumer Affairs, they have especially asked to make a publicity and a media agenda to launch these specialty crops, specialty varieties in a big way that will be helpful in fetching good prices in the market also. And those campaign in some of the commodities really beneficial. Say for example, the campaign like Sunday ho ya Monday, Rooj Khao Ande. That is the campaign from the Ministry of Consumer Affairs and AAGB Corporation of India. Similarly, we should have the campaign for that. And I also urge the groundnut community, the peanut community, can you make like AAGB Corporation of India, Peanut Corporation of India? If you can do it with the involvement of the researchers, corporates, cooperatives, I think that will be a wonderful thing, moving our groundnut in a big way in this country. So I urge to establish a peanut corporation in India. Thank you very much.
Yes, sir.
Oh, yeah. I'm really enjoying Dr. Gupta presentation. Is very informative. My question here is, as we know, the agriculture challenges mainly coming from the population size growing. So I wonder if you have any projection of the population in India in 2047. Also, we know right now that India is the largest population in the world, is past China. We know right now that India has a very high economic growth. Do you think India will follow the the same pattern as China? China population now is the turning point, goes down. What is your thought about that? Thank you very much.
You want to respond to that?
Yes, sir.
Yes, sir.
What Dr. Gupta sir has conveyed to the question of whether Indian farmers are ready to accept this high olic concept. Sir, it is, I have seen all biofortified and many other crops it's coming. But here in groundnut, the Girnar 4 and 5 has become the yield front also to the farmers. So I am since last 30 years in the groundnut from as a technical graduate, postgraduate to the industry, to the processor. My stall is there, number one. I have groundnut oil also right now. So here the oil is also there and farm productivity is also very good. So the acceptance increasing like anything among the farmers. So we are very bright future, and what the sir has told, the peanut corporation is the need of an hour. Rather, sometimes I used to say ICRS now two alternate, either BSNL or be the global, right? So from the global perspective, is required to have cater this demand and expand our operation in that. Thank you very much. More, I think Dr. Pandey sir has told that we will discuss in the panel discussion, but here are some few points. Thank you, sir.
These ICG, no, Girdar 4 and Girdar 5, sir. These are the joint releases between ICRISAT and IIGR now. And they are really becoming very popular all over India. Yes, this book.
Thank you, Dr. Gupta, for proposing the idea of putting the hashtag for groundnut. I would like to propose the hashtag or tagline of like Dil Se Dil Ke Liye, highlighting the requirement of heartily healthy, heart-friendly oil of the groundnut, especially high oleic. So again, input here for the Ayushad mission could be heartily for the heart. Dil se dil ke liye. Thank you.
Thank you for this interesting suggestion. I hope this will catch up in due course of time. You know, keep on promoting it.
Hello, sir. Sir,
Yes, I see. Yes, sir. Two more questions, then we will move on to the next one. Where is the mic? Please go ahead. You go ahead.
Sir, I have a small question. Sir, when we are developing a variety with a special feature to it, for example, aflatoxin or let's say dormancy or increase in oil content or oleic acid. Sir, my question is when we give it to the farmers, like if the farmers are growing it, how do we tackle a problem of mixture when it comes to the market? So because there are specific things that we are making, but when it is coming to the market, we are unaware if it is the one which is the requirement. So that is my question, sir.
I think a good breeder can tag the important morphological markers in their varieties. I remember when my father developed a variety Chitra, it is a variegated seed coat color. And I was a student of graduation. I think I was doing graduate. I asked why you have made this kind of variety. It will not be acceptable to the market. But that variety was having some quality features. So variegated seed coat means Chitkabra, Chitra. It can be easily identified in a genetic stock that it is really Chitra. There is no other variety. So breeders, the intelligent breeders, they add a specific morphological marker in their variety that can be distinguished. So my request to all young breeders, kindly take this important message. You add some specific morphological marker in your breeding program. Thank you.
Yes, sir.
Yes, sir.
Last question.
Yes, sir. So groundnut is a very nutritious food for the community, for a human community. So in this regard, in 2023, we had an International Year of Millets. So in the similar line, to promote the nutritional value of groundnut, can we have an International Year for the groundnut?
The same thing I requested to Honorable Prime Minister because I was dealing the section of oilseed, pulses and millets with the gracious acceptance of our Honorable the then DGICR. He put me for that front. And he was really interested in groundnut. So he asked one particular question, what need to be done in oil seeds? Can you suggest one single thing to us? Then I told, simply your blessings. Then he laughed at, what blessing will do? Then I said, you have gave blessings to the millets. At international level, it was recognized millet as International Year of Millets. And now the millet is very popular, not only in India, but many countries of the world. The similar blessings need to be given to groundnut. And if you can do that, sir, the groundnut will move faster and faster. So these are some of the things. As I in the morning told, it's amazing crop, no parallel, no crop I could see like groundnut. It is in the international discussion that we need to promote groundnut in a big way, and that will be the day someday we will observe the International Year of the Groundnut.
Okay. Okay. Thank you, Dr. Gupta, and also the audience for this interactive. Sir, sorry.
Not a question. I shall certainly, with respect to the chair, just only one suggestion or request, and I am in the perfect position to request at least. So I was heading ICAR and now, of course, CRISAT. So, Dr. Saab, my request to you, to CRISAT staff or Manish Pandeyji, and of course, Dr. Bera, that you please prepare, and also our industry partners, please prepare a roadmap for at least next 5 years of what we should be doing for groundnut. What is going to be shared, it is said, our industry partners, and also policy aspects. You please prepare that document, and that could be one of the output of this particular conference, including your suggestion of peanut corporation or groundnut corporation, whatever it is, and also good tagline, so that we will be able to take it to the competent authorities in Government of India and also elsewhere to implement it. So this is my suggestion to you, and you are in a perfect position being a DG of ICAR. You can take the lead, bring everybody together, and then prepare that roadmap. That will be a fantastic document to work on. Thank you very much.
I think Dr. Gupta can take lead on that, and and DDG Research from ICRISAT side to organize this group. Right, so thank you again, everybody. And we can move on to the next presentation. I don't see Dr. Shan here.
No, my apologies. I'm not there at the moment.
Okay,
But I'm
okay.
online.
Good, good, good. Sorry. Please, go ahead.
Let me share my screen. Can you see that? Is that visible?
Yes, for online participants, it is.
It's clear.
Okay, my apologies for not being there today. What I was going to do was really look at where ICRISAT is and where we're involved in obviously groundnut, but more broadly. And at the moment, we're going through a little bit of a restructuring as well in terms of how we do research. And we have just recently published our strategy for the next 6 years. I just want to pick up some themes within that, which I think are relevant. So I probably don't need to give this background, but very briefly, obviously, ICRISAT is a non-profit organization established in 1972, and we focus on the improvement of sorghum, pearl millet, pigeon pea, chickpea, groundnut, and finger millet, as well as minor millets. And our vision really is in terms of a prosperous, food secure, and resilient dryland tropics. And we take a very market-inclusive approach. And really, the aim is to reduce poverty, hunger, malnutrition, and environmental degradations, focused very much in the drylands. So that's both within India, but also we have a number of centers and research centers in Africa. The way we try to work is to take a look at what the problem is and try to understand some of the barriers. So we have, organizationally, we have 3 programs. We have accelerated crop improvement, which is very much focused on the area that I look after, where we're looking at how do we produce new products, how do we produce new varieties, technologies included in the seed. We also have enabling systems transformation, which is focused about understanding what the market wants, understanding what farmers want, what the value chains are, but also understanding some of the nutritional components of that and how that brings in society in terms of women, youth empowerment. And then the other one is resilient farm and food systems. So that's very much around how do we make the best of the environments available in terms of agriculture. And that includes a lot of work in terms of looking at water management, but also things like digital agriculture and geospatial technologies. Those 3 programs work together. And our focus is very much on the sort of landscape level. So it's how can we identify and how can we capture the best resources available? How can we correct problems like soil degradation and nutrient loss? And then how do we fit the crops into that environment? And obviously, for many of the commodities that we're talking about, the value chains are there, although the logistics need work in some cases. I'm going to talk largely about accelerated crop improvement, but I think it's important to bear in mind that ICRISAT is focused on the entire value chain, because I think without that, it's very difficult. And the demand-led approach, which is the only approach that's likely to work in the long term, really has to take into a whole range of different accounts to be successful. We've restructured a little bit within ACI. So, I mean, originally we were structured into clusters, which were very disciplinary. We've moved away from that a little bit so that we're now focused in terms of the different components of developing material. So we're just about to appoint a head of breeding, and we're looking for a head of trait discovery and delivery. Manzul Dar is our head of seed systems. And the idea really is to look at how we work across crops. So many of the situations that we face in terms of the crops we work on are similar. So heat stress, drought stress, obviously diseases as well. Where can we work across the crops as a way of getting better money return on our investment? And obviously also understanding how the value chains work, but essentially what is needed, and that's needed at a whole range of different levels to make this successful, so that we don't simply breed, but we breed to specific target profiles which are informed by the markets, but also the farmers and also the end users. So what I want to do is just move on and say a little bit about the way we're enhancing what we're trying to do. So obviously the basis of all breeding is really in terms of having genetic variation. And now there are a number of ways in which we can work with genetic variation. So from crop-wild relatives, we're looking at secondary and tertiary, within species variation, where we're looking at the gene banks, or we're looking at advanced breeding lines from around the world, or existing cultivars and hybrids, mutation populations, and then obviously gene editing and GMO. So really secondary and tertiary are where we're struggling to really find the traits that we're looking for. Where we do have those traits of interest and we have a selection phenotyping mechanism, then obviously it makes sense to look at advanced lines, and particularly within the gene bank where we have extremely wide accessions. Mutation populations also allow forward and reverse screening, so we can look for phenotypes, but they also have the advantage that we can validate some of the potential candidate genes before going on to do things like potentially gene editing or GMOs. And it's potentially another way to do the same thing. And for environments or countries where gene editing is really not an option, then I think we may still have to work with some of the mutational side of things. And then obviously gene editing is really offers an enormous amount of potential, and particularly in India, where SDN1 and SDN2 are accepted, where we can understand how something works and we can directly modify those genes within the cultivars or hybrids that we actually want to work with. And these are all sort of underpin our philosophy in terms of trying to identify variation. The other thing I think that has been that we're tightening up on really is trying to understand a little bit more about how those traits work and where they work and where they won't work. So one of the things that we're investing a bit more in now is really trying to look at the individual components of those traits and understand which backgrounds they work in. And obviously we work in Africa as well as India. So which genetic cultivars and backgrounds will give that trait, how that trait's affected by environments, and really on the basis of that to decide whether the trait is suitable for deployment. And I think one of the things that we will be developing over the next 2-3 years is a system that allows access to those introgress lines. So it's not simply that it's a question of us providing material, but also the material being available for request. One of the things we're doing actually next week at Founders Day is really to launch a Centre for Pre-Breeding Research. And really the objectives of that are to develop those long-term resources that we will need, particularly in terms of introgression from secondary and tertiary gene pools. So that will involve obviously embryo rescue techniques, the pangenomic analysis, which has already been mentioned as incredibly powerful in terms of understanding what the equivalent genes are, but also how they may act. Cytogenetics, segregation and ploidy analysis. So again, we understand more about those relatives and we understand how to bring material in. And then using long-range sequencing and modern approaches to really understand what structural variation between the different genomes is, because that will affect how we bring in material. The other thing that I think we're going to get more and more involved in is actually working within crop-wild relatives to identify the genes which are providing the trait, and then looking at a sequence level how we transfer that trait directly through gene editing or other approaches. So hopefully that will give us a series of lines that actually really start to tackle some of those traits that are difficult to tackle at the moment. So things like pod borer complex, resistant, some of the disease resistances, which are quite limited in the formal germplasm, but also more and more as we go forward in terms of compositional and functional traits, so the nutritional values, processing values. And those long-term resources will be available for seed greening. And we would see ICRISAT, one of our key roles in many ways is to generate those long-term resources, which are difficult to generate within a short-term funding cycle. And part of what we're trying to do is institutionalize that. So it's not simply a case of we have money for 3 years, we'll do this bit and then everything stops. But how do we keep that going, even if it's at a relatively low level, so that those resources are produced? The pangenomes have already been mentioned. I mean, a great deal of work by Rajiv Varshney's group in terms of, and many collaborators in terms of trying to understand the relationships and the traits within different genomes. And I think the more sequencing goes on, the more genomes are sequenced, the easier it's going to become to identify genes conferring traits and really identify the equivalent ones. So we can look at mechanisms to either engineer those, or we can bring in variation from within that gene pool. And there's quite significant work ongoing at the moment in terms of developing larger scale pangenomes. And really just one single example of how this sort of approach is being used is really using wild species, secondary and tertiary species, as a way of introducing strong resistances in cultivated groundnut. So in this case, looking at stem rot disease, where the introgression is starting to give us material that looks good in terms of resistance, but obviously it's wild accessions, and it will need work in terms of either transferring the trait directly or actually breaking down the drag that happens when you make wide crosses. But that work is underway. In terms of the availability of material, obviously ICRISAT has a lot of genetic material available, and particularly that genetic material underpins almost all of what we do in terms of crop development. The key thing there is obviously to have a screening technique that works at reasonably high scale. But we are developing many approaches, and I'll talk about some of those very briefly, that will allow us to actually screen large sections of the gene bank and allow us to identify those maybe relatively rare cases where the phenotype of interest is present. We're also involved in gene editing. So we have gene editing systems working in a number of crops where there is that opportunity to really, either through homology with other species or through discovery in some of the wild species, identify the changes that need to be present to be able to transfer some of these traits. And obviously, a large part of what we do with a lot of collaborators within CGIR is to develop tools and resources, particularly in terms of genetic mapping. So we have a number of platforms developed which potentially allow quite rapid assessment of genetic crosses and structures. But also in terms of quality control, not just in terms of hybrids, but also how do we trace the quality within breeding programs? And then the next step, how do we trace that into the seed production side of things? Because obviously the quality of seed and the identity of seed makes a big difference in terms of the outcomes. And particularly modeling is quite a useful approach in terms of understanding where the environments, the best environments to get the best yield out of some of these crops comes, and how we use that as a way to start getting a little bit more predictive in terms of how do we know where things are likely to be good? How do we identify where the gaps in that are, including things like management? And then a whole series of basically phenotyping approaches, which allow us to look at things like composition, so protein, fat, oil, mineral content, actually size, shape, structure, and then some very simple approaches in many ways to speed up the whole process and the accuracy of phenotyping. I'll give one example. So this is X-ray CT from Janila's lab, where actually doing shelling percentage in groundnut has been modeled through using X-ray CT, and we have an algorithm which allows a number of parameters to be identified at high level, at high accuracy. And this year was the first year where actually the entire breeding program used this as a method, saving both labor but also time. So these sorts of developments, while they may initially look a little bit science fiction, can be implemented into practical breeding programs. And then clearly one of the other key things is developing rapid assays. So this is an assay that's been developed, a relatively simple approach to really look at the potential for stem rot disease resistance. And in many ways, it doesn't have to be perfect, but if it allows a large-scale screening to identify the most promising material, then it's a good way forward. Rapid generation has been mentioned as one way of actually decreasing the maturity cycle and increasing the number of generations. We do have a method within groundnut, which is not speed breeding as such, but it's modifying the conditions and modifying the way in which the material is harvested. There's still potential to develop a full-scale speed breeding approach. But at the moment, this is quite useful, but limited in scale, and it's something we're still working on. And then the idea is really to bring all of this together at the different stages within the breeding schema. And all of these can be applied as a way of really increasing the accuracy of selection, allowing a much greater level of selection in terms of sampling, but also moving things forward with more confidence as much as anything else. And the one at the moment that ICRISAT is implementing across all crops is genomic selection, to try and understand how understanding the value of a genotype potentially allows us to predict which genotypes we're looking for at the end of a selection cycle. And the big advantage of genomic selection is it potentially allows you to do a number of generations of selection quite rapidly without phenotyping, and also to recycle parents very much faster. Briefly, as you will probably know, we've signed up to the 1CGR in October 23, and we've recently released our strategic plan. And I just want to spend a few, maybe 5-10 minutes, just talking about some of the future areas that we think are likely to be important, and particularly may have an effect in terms of how implementation of changes within the system are made. And I think really just before that, just to say that this, obviously we work with CGIAR, everything we're doing is aligned to their current structure in terms of advanced breeding, which is the new program is called Breeding for Tomorrow. And we will work in collaboration with them, both in terms of sourcing technology and sourcing understanding, but also providing our understanding. And I think that is one area where there's significant potential for collaboration, both South-South and with other institutes. Within accelerated crop improvement, we've basically identified 7 areas, and I'm going to talk a little bit about a few of them. I think one of the key things that we see as being important in the future is to start, particularly for stressed environments, to start moving away a little bit of looking at single crops. I think we need to be looking at systems, whether that's relay cropping or whether that's intercropping. I think there's a lot of potential in terms of increasing resilience, but also potentially increasing yield by understanding some of these factors. So just very briefly, promoting millets is obviously One area, particularly minor millets, because they come from lots of different environments, but we're also starting to use ICRISAT's expertise and experience to really help support breeding in other crops. So particularly Bambara groundnut at the moment, which is an African crop, although it has just been introduced for testing into India, and sesame. But I'm sure there'll be other examples as well where we can use the experience we have to really help support the breeding programs and help some of those innovations to come through. One area that we think is going to be important in the future is really increasing resource use efficiency, partly because it will decrease the costs for farmers, and potentially maintain nutritional value without loss. It should increase disposable income and potentially empower women farmers, enhance water use efficiency, but breeding varieties and hybrids that are responsive to conservation and regenerative agriculture through their soil interactions, and reduce carbon footprints by reducing losses and also reducing inputs. And hopefully that will also reduce environmental pollution, which obviously is a significant problem in many areas. Clearly at the moment, most variety registrations do require an increasing yield, or at least a similar yield. But I think in the future, we may need more flexibility. If a particular crop variety or hybrid has a specific purpose and a specific benefit, then I think it may be we need to reconsider a little bit whether yield must be the per se requirement. And I think one of the things that we're very focused on, particularly in Africa and as well as India, is a focus on crop nutrition and economic value, not so much yield per se, although obviously at the moment the structures are focused around that. And some simple examples of the sorts of things. So we have a project about to start, which is looking at root architecture in pigeon pea, but we'll also include groundnut in some of that, to really get an understanding for the underlying genetics in terms of how do they deal with nutrient use, how do they deal with water use, and are there genetic systems in them which could be used to actually improve those qualities. We are involved in a number of the visions for adapted crops and soils, pigeon pea, finger millet, sesame, and groundnut. And we do have a project in sorghums looking at compounds which reduce the rate at which urea is converted into nitrates. And that will be an important area in the future because it will improve efficiency, but it will also improve it will reduce the impacts. So we're thinking very much in terms of systems. So maybe a sorghum system, which is intercropped with groundnut or pigeon pea or relay cropped, has the potential to actually produce better results in terms of the overall outcomes. And I think one of the other things, particularly, is in terms of functionality. So how much do we really understand about some of these, and how does that, at the moment, maybe limit the uses for them? So really understanding what the functional side, as well as the nutritional side, and particularly bioavailability, could open up new options in terms of ways we breed, but also improve nutrition. Finally, and this one might be slightly, well, controversial perhaps, I think one of the other things that makes sense, particularly in stressed environments, is maybe to look at how we do agriculture. And perhaps what we need is a more complex agriculture, which may include mixes of annuals as well as perennials, particularly things like corridor cropping in areas where one of the crops is contributing from a resources point of view, and the other crop may be taking advantage of some of that. So deep rooting maybe in some crops helping water access, but also nutritional access. And again, this sort of reflects in most of the areas we work. There's still a prevalence of undernourishment. And I think we really need to look at how we improve the nutritional value, as well as improving, obviously, the farmer's income and the overall balance of imports and exports. So I'm just going to give this one as an example where we're working within the pre-breeding center to look at hybrids of sorghum where we're introducing, effectively, we're introducing perenniality. And the aim behind this is really to look at systems which would benefit from not being tilled, but also in terms of things like access to feed, access to fodder, access to feed, eventually access to seed. And it may well be some of the systems we're working with would benefit. And obviously something like pigeon pea is already a perennial. And really it's a case of trying to understand what the additional chromosomes, what the additional genome is doing, what traits it's bringing, how do those traits have potential benefit, either directly within that hybrid or through using gene editing as a way to actually engineer some traits into existing cultivars. And really just to finish, I mean, I think I'll say very briefly about one project we have, which is the Bihar Center of Excellence for millet value chains, although we're now including legumes in that as well, to try and illustrate how we're trying to work. So that consists of 6 work packages, and they range across the whole, if you like, value chain. So from collecting and conserving existing germplasm within Bihar state in this case, to also screening the material that we have for suitability in Bihar state, strengthening the breeding pipelines, particularly for minor millets where crossing is not simple. And then obviously the seed chain. So if we're introducing new crops, if we're introducing diversity, we need seed chains that will allow access. And I think this is one of the key issues which has already been raised, is those seed chain sides of things. But also, as has been mentioned, there are large yield gaps in many cases. So understanding best cultivation, introducing mechanization, and adjusting agronomy and management to try and get the maximum crop that can be got. And then obviously, what do you do with some of those things? And particularly in the case of things like small millets, what are the products? How do you develop the value chains and processing? So we're putting together processing units in Bihar, both primary and secondary, as a way of how do the farmers make money? How does this feed into the overall picture? And we also have a quite strong focus on how does that affect gender and youth in particular. So we're not, by introducing innovation, we're not making life harder for female farmers, for example. And I think all of these integrated approaches are where we need to be going. Really looking again, as I said, I think while we can do the breeding, while we continue to do the breeding, I think we need to think more broadly about how agriculture works and how we get the best out of the land that's available. And it may not be monoculture is the most suited approach in some areas. And with that, I'd like to just really thank lots of collaborators and funders. We have a very vibrant interaction, both within India, but also within Africa, working with a wide range of funders and stakeholders and partners in ICAR and elsewhere to really bring some of these innovations through. And I think that the struggle we face, particularly in terms of climate change, means that these challenges are way beyond any single institute or institution. We really have to be working collaboratively. And that's not always easy, but it's absolutely essential in terms of how we go forward. Thank you.
Brought it in the front, world deliver, co-development, that is one thing very important, that creates confidence amongst the partners. big brother or younger brother or small brother attitude, the word co will disappear, will help to disappear that. My one suggestion is on your pre-breeding. Pre-breeding has been in practice at ICRISAT for a long time, and particularly legumes, oil, groundnut, and pulses. Similarly, in groundnut at other institutions, people have worked for 30 years, 40 years, and without programs have been closed down. There is a need to review the past progress, why the programs could not make the progress, what was lacking, and what is that we can do now, which was not being done at that time. Of course, now we have new tools, that is true. So one has to check out the program very carefully so that, we don't fail again. We have failed 2 times or 3 times, not only at ICRISAT, but at other places also. So these are just my observations for your consideration. And again, thank you very much for your very nice and comprehensive presentation. I'll request if my co-chair has any comment to make. Thank you. Anybody from the floor? You are off the hook very easy. Nobody is asking any question. You have done a great job. Thank you very much, sir.
Thank you.
Bye. So now the turn is of my dear friend Dr. Bera. And he will take us through the groundnut situation. Of course, you heard about oil seeds in general from Dr. Gupta, but Dr. Bera would be focusing mainly on groundnut. So, Dr. Bera, floor is yours.
Yeah, respected chair, co-chair, and distinguished guests. So this is the Indian Institute of Groundnut Research, situated in Junagadh, Gujarat. And Gujarat is the whole of groundnut in India, particularly Saurashtra region. In India, as Dr. Gupta said, groundnut is a major oilseed crop in India, and it has very, very significant 3 roles. First is a oilseed crop, now moving to oilseed to food crop, then it is really important as a fodder crop. And there is an example in Saurashtra region, if farmers feed their cattle groundnut fodder, the cattle of Saurashtra region gives 50 to 75 liters of milk per day. That is the buffalo and gir cow. That is the record milking capacity of Saurashtra region cattle. Sir, India is now both area and production in second position. Earlier, India was the second in production, but first in area. But last 5 or 6 years, we have lost a huge area under groundnut cultivation. Now we are second position both in area as well as production. The contribution of groundnut to the national oilseed, these are the 9 oilseed, major oilseed, and groundnut is the first one in terms of production, productivity, and 2 other competitor is the soybean as well as rapeseed mustard. So these are the major 3 oilseeds in India competing each other, both in production, area, and productivity. So groundnut is the topper in the productivity, but soybean and mustard, they are competing for area as well as total production. And total last year, 2023 and 24, this is the contribution of groundnut to total oilseed in India area. We are having 4.7 million hectare out of 30 million hectare, and production it is 10.18 million ton from around 40 million ton. And groundnut is contributing about 25% to the total oilseed production from 15% area. So as I told, these are the 3 competitors fighting neck to neck for production, productivity, and area. So here we can see this is the 3 decades comparison, where area, that is the soybean, continuously increasing. 117, then 129, and 132 lakh hectare. Whereas 2 other, that is rapeseed mustard and groundnut, here production is fluctuating. Coming to production, the soybean is again constantly fighting, and rapeseed mustard, that yellow, that 78, 102, and 131. But here see the groundnut. Groundnut is fighting for its productivity over the decades. So we are number 1 in the productivity, all the 3 major oilseeds. So as I said, 1 major oilseed is occupying the productivity, highest productivity, another one for production, another one is for area. So this is the groundnut atlas in India. This is the Kharif season. Kharif means that is the major rainy season. All over India, groundnut is being grown. And these are the green, deep green area is the highest, most efficient cropping zone, where productivity as well as trade of the crop is maximum. Then efficient cropping zone, then non-efficient cropping zone. Almost entire part of the country, groundnut is being grown. And groundnut in India is being grown for 3 seasons, rainy and 2 post-rainy seasons. And for post-rainy season, again, we have a 2 season, Rabi as well as summer. So here the lower part of the country is mostly concentrating for post-rainy groundnut cultivation. Here again, we have divided the 4 groups. Initial, we have a projection, and this is the achievement over the last 5 years. 2023-24, we prepared in 2018-19 roadmap for oilseed production in the country. Based on that, 5 years, this is the 2023-24, we targeted around 5.25 million area, 11.02 million ton production, and 2100. There we can see the achievement. We have achieved that 2100, 2163, but because of the loss of area, the 11.02, we stagnated 10.18. But I will go through the next 2-3 slides. However, we have achieved. So this is the country, major oil groundnut producing states. Of course, this Gujarat, we are here, our office is here. This is the productivity area. Similarly, this is the West Bengal, more than 3 ton, and some state, Tamil Nadu, again, 1,600. But other 2 or 3 states is giving a very poor yield, so they have a sufficient area. So due to this reduction in area, so we have our production restricted 10.18 instead of 11 what we targeted. And 80% of the groundnut is grown under rain-fed condition during rainy season. Sorry. This is the private estimate from APIDA, Ministry of Commerce, and so they are telling, as per their estimation, every year they have a estimation. They have a separate mechanism other than Government of India public survey. So they are telling this year it will be 11.312 2024. So probably this year we will achieve that 11 million ton. And area also, as per they are saying, it will be around 5.3 million hectare. This is the comparison from the private group. India is having 6 lakh hectare and going to produce 13 million ton. It is little bit higher side to my opinion, but will be somewhere between 11.5 to 13 this year. But this is the picture from last year and this year only single season. That is the first estimate based on Kharif. Another 2 season crop will come. So if we see the last year area production and productivity, this year we have increased the area and production already achieved. That is the based on single season only. Another 2 seasons are coming. And Kharif season, as I said, 80% area under rainfall condition and productivity will be little bit less. But post rainy season, 2 season crop are high productivity crop. So naturally there will be a huge production from the area, that total area. Area also we have increased in the post rainy crops. And again, it is going to be a bumper production in India. So these are the last Kharif season, rainy season crop. And this is the 3 major states, Gujarat, Rajasthan, and Madhya Pradesh. There are increase in the total area as well as production based on their normal area as well as area. And this is the production from these 3 major states, Gujarat, Rajasthan, and Madhya Pradesh. So there is a huge production in this country from the last Kharif season. And another major outstanding point is we have sustained the groundnut production in the country continuously over 5 years, and this is the last one point, that is 10.3. That is based on single season only. We are waiting from the for the production from another 2 season crop, that is post rainy season crop, and productivity is very high. So definitely that private estimate and our estimate is going to be around 11.5 or may reach up to 12. So there will be a bumper production of this crop is 24 and 25. So that is because of the some of the recently released high-yielding varieties, joint collaboration of ICRISAT and our national partners. And here we can see the productivity. The potential of all those varieties are above 3 ton per hectare. But our national average is hovering around 2 ton or 2.1 ton. So there is a mismatch or gap between the research data, potential of the variety, and achievement or harvest in the farmer's field. So we have to bridge that gap between the potential of this variety as well as the farmer's harvesting. So if we can bridge that gap, so definitely we can achieve further more production and productivity in this country. So this is the Ginnar 4, Ginnar 5, and later on another GG39 and GG40. This is the product of a joint project from ICRISAT and NARS partner. And Dr. Nigam was the leader in that project, funded by Government of India, around 7 crore. And we have released the Ginnar 4 and Ginnar 5 under his leadership. Dr. Bharshnay, Dr. Manish, myself, Dr. Janila, and Dr. Pandey. So these are the 5 project player. We have a 5 years project. From there, first ever high oleic groundnut we have released in India, that is Ginnar 4 and Ginnar 5 with a very high yield and 80 and more than 80% oleic acid. And we know the benefit of oleic acid because of less saturated fatty acid, and again increasing the unsaturated fatty acid. Again, they are MUFA and MUFA, polyunsaturated and monounsaturated. We have increased the monounsaturated fatty acid, that is the good for health as well as shelf life of the product. These are the major constraint. The 80% area under rain-fed condition where mostly biotic and abiotic stresses are there, then input starvation, and we are depending on the rainfall only. Then production fluctuates based on distribution and quality of the quantity of the rainfall, as well as very high seed rate. Indian farmers used to have a 150 kg pod, or even somewhere it is 250 kg pod. In Rajasthan, it is very high seed rate people used to have because of the soil-borne disease. And seed production chain, very poor seed production chain. It is not well established. We are not able to produce the quality seeds in sufficient quantity as per the demand of the farmers or government. So that is another drawback, lack of climate resilient varieties. We do not have that kind of varieties which can withstand all those biotic and abiotic stresses and give a sustainable yield under difficult condition also. So high cost of cultivation, another is a major problem for the farmers because of the labor intensive, high seed cost and poor mechanization. In oilseed in India, mechanization is very less. So in Gujarat, it is around 90%, but most of the other parts of the country, it is hardly 10%. So that is another challenges for us. Well, this last year study, we have seen there is a yield gap between 11% to 45% between the farmer's field as well as our potential of the newly released varieties. So on an average, there is a 21% yield gap between farmer's field as well as potential of the variety. So if we can target to reach that gap, 21% or at least 15%, we will have a huge bumper production in this country, and so it will be a profitable venture for the groundnut farmers or farming community. So, Government of India first priority is the production of quality seed, that is the breeder seed, and making available good quality seed to the groundnut farmers so that farmers can harvest or bridge that 20% yield gap and make the crop most profitable. So based on this rate, 150 kg per hectare and around 6 million hectare, we require around 24,000 quintal of breeder seed, and it will be multiplied to foundation, then certified, and ultimately we can replace the old seed of 33%, 1/3 of the total area, and that will give a huge bumper production in this country. And this is the last production of the last 5 years breeder seed. So we are not producing enough or sufficient breeder seed, quality seed, which can cover the or deliver the seed to 33% area of the groundnut. So we have to produce breeder seed to match that quantity. So that is the first and foremost mission of the government. So we are targeting, and government is giving a lot of emphasis to produce the breeder seed sufficient quantity. And one of that aspect is the establishment of breeder seed hub in groundnut. And groundnut, we have 22 breeder seed hubs, all these 22 breeders seed hub we have distributed across the India, different states of India, and they will have a target based on their list of varieties, which seed hub will produce how much quantity of breeder seed and which variety, everything is chalked out. And as per that plan, we are going to produce the required breeder seed, and that will boost the total crop scenario of groundnut in this country. And second target is to popularize the latest varieties through our outreach and awareness program. And India government have a online demonstration as well as training. So we are conducting, last year we have huge training and FLDs we have conducted and distributed huge quantity of groundnut seeds and implements for improving the mechanizations, side-by-side biofertilizer to reduce the cost of cultivations to different farmers, around 6,500 farmers we have given this benefit. And that is why we are seeing that boost in area as well as production of groundnut crop in last Kharif season. And this is the Girnar 4, Girnar 5, the Saurashtra region. This is the achievement. This is again the estimate from that private company. They are involved in procuring distribution of this high oleic groundnut. So average yield it is giving 3090 in farmers field. So here we have achieved or bridged that gap, and highest yield is 5.5 ton. So this is another one example in Saurashtra region. So if we have a good variety and good quality of seed, we can easily achieve 3 ton average yield per hectare. And somebody raised the premium prices for these biofortified varieties. There we can see this is the current market scenario, this year scenario. The biofortified groundnut variety that is high oleic, Girnar 4, Girnar 5 is getting a premium price per kg of pod than other varieties. And also Gujarat is a good area for producing a quality groundnut, exportable groundnut. And that's why the traders are giving the 10 rupees premium price to the high oleic groundnut farmers. And Gujarat, particularly from that district, and Rajasthan and Maharashtra, lot of groundnut is exported, and India is covering around 20% of the total export of groundnut to the world, and Argentina is #1. So India is also equally competing with the Argentina to export groundnut to the world market. And India mostly exporting all those groundnut to Asian countries, that is Indonesia, Vietnam, Philippines, and Malaysia. They are not able to catch the European market because of the aflatoxin problem. So that is another issue we have to solve immediately. Otherwise, this crop will suffer, and groundnut farmers in this country will again going to suffer. So this is all about. Thank you. This is the update of groundnut in this country. Thank you.
Thank you, Dr. Vera, giving us a flavor of groundnut in India. Is there any query or suggestion? Dr. Kuldeep Singh.
Well, thank you, Dr. Bera, for a very comprehensive presentation of the groundnut specifically. Well, we're looking at some of your slides, especially when you're classifying them as efficient vis-a-vis inefficient regions. I see the efficient versus inefficient regions are more or less of the same magnitude. How can we take care of those regions which are inefficient to make them relatively better than what they are now?
And somebody pass on the mic there, please.
Yeah, we have seen it is just traversing side by side that efficient and non-efficient zone. The thing is that it is district-wise or block-wise data. So that's why maybe soil is different, or farmers are not using proper agronomic practices or inputs. So this is based on the productivity as well as area of a particular district and also block level data. So there is a mismatch between the block or within a district. So farmers are not using the proper agronomic practices. Some farmers are using, some farmers are not using, or maybe their field is poor in nutrition, or some good patches are there, they are getting the good productivity. So we have to, this is recently we have developed this atlas, and based on this, again, we have to go and check it, why it is different within the same district or nearby blocks, between the blocks. Then if we can identify one or two points there and make efforts to clear that blocks. So probably most of the inefficient zone or efficient zone, we can convert to highly efficient zone. So really we are looking for that. And from this rainy season onwards, we are going to target at least Gujarat, wherever efficient and non-efficient or highly efficient is there. So we have to work on that. And if we can convert 50% or 100% area of efficient or non-efficient to efficient zone, so again, there will be a boost for production and productivity. Thank you.
Yes, there.
Yeah, thank you, Dr. Vera, for nice presentation. So I have my question is actually following Dr. Kuldeep Singh question. So now rhizospheric engineering is coming up as a big solution, right? So, and peanut being a legume crop. So like when you have this kind of block level data, so are you thinking about doing the micronutrient as well as metagenome analysis on those plots to understand actually at the microbiome level what is going on? And the next question is, what is the crop rotation? Because now we know that which crop was pre-grown, they have a lot of effect on the like post-grown crop. So do you have like, even Gujarat or nationwide data that what are the pre-crop that being grown, which is whether it is having any effect on the groundnut production? Yep.
Yeah. That your first question, that is microbiome data, is very, very crucial nowadays, because most of the farmer, except some states like Gujarat, Rajasthan, where the land holding is large and farmers are well-to-do, they are using the inputs or good agronomic. But for other part of this country, farmers are not using a good agronomic practices, and they are raising their crop under rain-fed condition without putting any good agronomic or resources.
Hello?
In such cases, this will play a major role, but not recently or immediately we are going to address this because we have to first generate the data, maybe district-wise or state-wise. Once we know these differences between the microbiome or rhizospheric climate, the differences is where the differences and which is playing the major role, then only we can address this. But certainly, as you said, this rhizospheric environment and microbiome is going to play a major role, particularly for resource poor farmer where land holding is less or farmers are poor, not able to use the good agronomy or input rich crop. So it is a very, very thrust area, but slowly we will take up. Second is the, can you repeat your second question? Crop rotation, it is well documented state-wise. The major, suppose in case of Gujarat, it is groundnut followed by wheat or some spices, that is the major. Similarly, each and every state, we have a clear-cut documented data for these crop rotations based on the market, soil type, and climate. Suppose if we go for a Tamil Nadu, the bottom part, there is a rice, groundnut, or sugarcane. Maharashtra again, sugarcane, arhat, like that. So it is different picture in different states. That is totally depending on the climate, soil, cropping window, which crop will fit with that gap. So that decides the intercrop as well as cropping sequences. And that also helps to develop or composition of that rhizosphere climate as well as microbiome also. And that is helping to have a good groundnut crop somewhere, or some cropping system it is not performing as expected or as available in Gujarat or Tamil Nadu or West Bengal.
Our friend from Malawi.
Hello.
Thank you, Nigam. Just two quick ones to Dr. Mbera. The first one is on the looking at the production mark 2 tons. Is it from both the Virginias and the Spanish, or it's for a specific one?
No, this is the national average, and it is a average of both Spanish and Virginia. In India, Spanish, that is called short duration groundnuts, and duration is 100 to 110 days, or maybe 95 to 110 days. Whereas Virginia, it is 115 days or 120 days to 140 days even in some states. So wherever crop duration is more, in case of Virginia, 120 to 140 days, definitely yield is around 3 to 4 ton per hectare. In case of Spanish bunch, where duration is 95 to 110 days or maximum 115 days, their productivity is less, maybe starting from 2 ton to maximum 3 ton or maybe 2.5 ton. And this is the average of entire country. So that long duration crop, that is Virginia crop, is being grown only in rainy season, wherever rainfall is very high or distribution is very normal. So that farmers prefers that Virginia crop. But wherever cropping window is less, there is a doubt in the rainfall as well as distribution in the rainfall. So farmers primarily go for this Spanish type, that is short duration. So either it can escape the drought or rainfall or whatever available window is there or rainfall is there, so that farmers can have a complete crop. Though it is a less yielder, but it fits their cropping pattern or cropping system. So in a summary, that long duration Virginia crop, the average yield is around 2.5 to 4 ton, and Spanish, it is 2 ton to 2.5, or sometimes it is 3 ton per hectare. And this 2 ton is average of entire India, average of Spanish and Virginia. Thank you.
Thank you. A second final one, still out of curiosity, marry your presentation with the one of Dr. Sajiv about groundnut being important for oil production, oil for groundnut being healthy friendly. What is your take in terms of the competitiveness of groundnut oil competing with, for example, soya in the coming future? Is there, for example, a willingness of the consumer to pay the higher price compared to the price of the oil derived from soybean? Just try to look into that so that I can try to compare with what's happening in Africa, whereby the investment in groundnut for oil production is not that much compared to investment in groundnut for food production. Thank you.
Yeah, as we all know, groundnut oil is a very good quality oil, edible oil. And traditionally, 4 or 5 states completely use this groundnut oil for their consumption or day-to-day food life. But side by side, we also know groundnut is also equally going for a food purpose. In India, around 50% groundnut production is going for a direct food purpose, and that has been increased after COVID period. So lot of groundnut is going for direct consumption as a food or different snacks, because it is a good source of protein. So entire export from the country, groundnut export is for kernel, not oil. We are not exporting oil at all. So total export, whatever 0.6 or 0.7 million ton we are exporting, that is exclusively kernel base, and it is going for food purpose to the outside country or making butter, peanut butter. So though it is a premium quality oil, but due to import of cheap soybean oil and availability of other oils and mixing of different oils, so probably that is another reason. So groundnut oil consumption is not increasing or maybe stagnant, but we are trying to highlight that this groundnut is a very, very premium quality of oil for food purpose. We must use, but as we know, the deep frying in soybean oil that sustain the high temperature or repeated frying also. So that's why most of the snacks industries, they are using the soybean oil or mixed oil. But nowadays, again, because of this COVID and post-COVID era, people are becoming health conscious day by day, and they are coming back to this such type of premium oil like sunflower, mustard, and groundnut. And after this release of high oleic groundnut, the quality of oil also improved, and people are favoring this high oleic groundnut. And in Indian market also it is available, especially high oleic groundnut, and it is available per liter about 300 to 350 rupees per liter. So now people are again coming back to good quality oil. So probably there will be a good market in near future because of this availability as well as health, looking into health issues. Thank you.
I think
Can yeah.
very brief, very brief.
Brief in the
And
sense.
very brief reply also.
Yeah, brief in the way I am saying, we are discussing right now on the research aspect. Some of the as I am from the industry and again a technical person, some of the points, because Gupta Sap is also very much at here. And so that point, which because the extension in our agriculture overall, we have observed that is not that powerful is not reaching to the farmer's field. So here from the farmer perspective, there are few things in the mind which is required to be break for a high production. Number one, in some typical areas, I say in the Khandes, SB11 is only the name which farmer is using, right? So the SB11 is 1969 variety. How to get rid of that and how to have a new variety, high productivity? Because the farmers are growing 1 lakh, 2 lakh hectare area is there in Khandes. Similarly in Vidarbha, Maharashtra, Tech 24 is only the mindset. They don't want to change. Even UP TG37 has entered in the mind, and it is whatever the high production, sir, as you rightly conveyed that 4 metric ton or so is achieved in, but that is not getting out from the mind, and farmers are demanding those varieties only. So how to penetrate in that market or in the farmer's mind, because that's very essential, because he is ultimately playing the role for the production. So that aspect is required from the farmer's viewpoint. And second, what I was saying, the global, because the groundnut, even this price this year is comparatively less, traded lesser than the MSP, even though farmer is having a profit. But where the export market of kernels has reduced, why? I observed in the Saurashtra floor, there was Chinese buyers 3 years back. At any price they were purchasing. Berasab is also might be knowing very well why they have disappeared. Because now we have to think the globally also that our crop is wide in many other countries. So that aspect is required to be studied in detail, because that export potential is only going to give the good price to the farmer. For the industry viewpoints, sir, 1,50,000 crore, 1,50,000 crore oil seed import is there. We want to reduce. Now, as an industry, groundnut seed production I used to start by May or June. My product is there by November in my warehouse. I have to start the at least commercial rate payment to the farmers. My liquidation is in next June only. For the 7th month, I have to hold the stock. I create an infrastructure with the various schemes and 5,000 metric ton of cold storage and everything. But the working capital need for the continuous purchase of groundnut is very essential, because seed is required to be purchased at the price, and the loan bank is giving at the what they are giving to the other industry, with the construction industry or anyone. While we are import substitute people, right? We are reducing the import. So the loan subsidy, interest subsidy, if applicable, then industry can be promoted. Yesterday I was there in the cotton conference also at Guntur or ICRIP. There were almost 35 companies that participated. Why hire, I think, year 1 or 2 only, the seed companies, because there is no, the finance is the huge requirement. Cotton, they do the production in March, and farmers payment to seed production, farmer payments in the June or July. That is, during that the sales period is started. So there is no huge block of investment at that time. While in the 1 minute, sir, while in the groundnut, it is the major problem, because huge investment is required and there is no loan subsidy also. And you know, the banking is having also limitations, unless and until some directive has been made from the policy level. Those who on the seed production of the oil seeds, this help should come. So these are some few submissions. Right.
Dr. Gupta, can you just very briefly, because she is standing there asking me to close the session.
So the first question, that infusing new varieties in the system. So this mission, which we launched, this national mission on oil seed, there is provision of promoting only 5 years old varieties. But I know groundnut is a special crop. There are certain special varieties which were released long back, but are still lifeline varieties of groundnut production. So for that, we are making provision to give the same concession which we are giving in the 5-year-old varieties. So that is one aspect. Second thing you have asked about that soft loan for the seed industry, particularly the high volume crops. I think government of India is aware about, because lot of demand is from the seed industry that for such kind of input, government should give soft loan. Prime Minister has launched a Dhan Dhanya Yojana, Prime Minister Dhan Dhanya Yojana, and there 100 districts have been identified. And I am hopeful that among 100 districts, there may be 30 districts where the groundnut will be the main crop, need to be focused. In that Dhan Dhan Yojana, the NABARD has been entrusted to provide the soft loans for the input management for particular crops. I think when the whole scheme will be, very soon it will be published in the newspaper and the media, I think those efforts are really useful for the groundnut production program. As suggested in the morning, the peanut corporation, if we establish, lot of problems can be resolved. This is important crop for the important country. Thank you very much.
Thank you, Dr. Gupta. Before I close, I'd like to thank all the speakers and also all the participants who have participated in the discussion. And the last comment, an opportunity.
Sir, thank you very much. I think you want me to wind up fast. But as an industry, and Dr. Bera also, and seed industries, my request is one technology which has been brought out by IUR, that is biopolymer. And recently, sir, we tested at the farmers field in farmers field house. And we will not say that it is giving 30% this and that, but yield advantage is much more. So this is rather valuation of the seed which you are going to give to farmers. It is proven technology. Kindly look into that. We are licensed to 2 firms also. And I feel it is a wonder technology which can add valuation to the seed. And there are prospects for the input delivery system, which is really very good in this technology. I will discuss with you separately how it can be taken advantage. Maybe next season or not, it should be in place. Second thing is, which we are advocating other crops also, what are productive, high productive areas. Our efforts should be there for the quality area expansion. Just like throughout India, we want to spread it. It may not work much, but quality area expansion is very, very important. Already identified area with high productivity, they can be the priority area for the promotion of this crop or that crop. So thank you very much, organizers, for giving me this opportunity to co-chair this session. Thank you, Dr. Manish.
Thank you so much, Chair and Co-Chair. Requesting you to be seated. From morphological markers to genomics, biotechnology, and high-throughput phenotypic tools to envisioning International Year of Groundnut. From groundnut or badam, Dil Se Dil Tak tagline to proposition to Government of India for promoting groundnut internationally as a savior in dryland agriculture. That was a good sum up. Ladies and gentlemen, please give a round of applause for all the speakers for walking us through ground oilseed scenarios, specifically groundnut, from both public and private industry lens. Special thanks to Dr. Sean Mays and Dr. Baojugo for virtually joining us. Now I request the chair to give away the token of appreciation for the first speaker, Dr. Sanjeev Gupta. Round of applause, please, ladies and gentlemen. Thank you, sir. I request Dr. Ravi Mathur to give away the token of appreciation to Dr. Sandeep Bairaji. Thank you, Chair and Co-Chair. Now I request Dr. Kuldeep Singh, Head Gene Bank ICRISAT, to give away the token of appreciation for Chair and Co-Chair. Dr. Shyam Nigam has been a forever pillar in global peanut research, and Dr. Ravi Mathur for carrying the legacy of IOR in key contribution for global peanut research community. Thank you, sir. Please have your moment here. Thank you, one and all. We close the plenary session here. Now, let's move on to the poster session. Poster session will be until 1:15. After 1:15, we'll proceed for the lunch. And after the lunch, we'll gather here around 2:20. Quickly, we will start our panel discussion at 2:30. So requesting one and all to stick on to the timelines and please proceed for the poster session. The presenters be present on your slot and the judges are requested to move around the posters. Thank you, one and all. We close the session here. Ladies and gentlemen, a quick announcement. The lunch is open. The people who have finished their poster session can move forward for the lunch. Rest of them who are not a part of this poster session can go check in, have your lunch, and can come back. Make sure that you are here by 2:20. Sharply by 2:30, we are starting our sessions. Ladies and gentlemen, welcome back. A quick announcement. Let's take poster evaluation committee, a quick announcement. Let's break for lunch and the poster evaluation shall continue after lunch. Requesting everybody to gather here by 2:20 so that we can start the panel discussion. Meanwhile, have the poster evaluation as well. All the participants are requested to go and have your lunch, please. Ladies and gentlemen, shortly we will be beginning our post-lunch session, requesting all to gather here at this inaugural hall. Ladies and gentlemen, welcome back for the post-lunch session. I hope everybody had a good lunch. Please, a request, don't doze off. It's a humble request. There are good panel discussion lined up for you, and hope everybody will stay tuned and participate actively in the panel discussion. Before we begin the post-lunch session, I now invite the Chair AAGB 2025, Dr. Manish Pandey, to give the token of appreciation to Dr. Himanshu Pathak, Director ICRISAT, and Dr. Stanford Blade, Deputy Director General Research, ICRISAT. I request Dr. Himanshu Pathak and Dr. Stanford Blade to join the dais. Dr. Patak and Dr. Blade, we are honored to have you in person here, and we appreciate your great support and execution of AAGB 2025 here at Goa. Thank you, Dr. Manish Pandey, for these honors. Moving forward, I now invite Dr. Sanjeev Gupta, Dr. Stanford Blade, Dr. Sham Nigam, Dr. Sandeep Kebera, Dr. Ryan Lipicio, Dr. Ravi K. Mathur, Dr. Hailey Desme, and Dr. Himang Bakshi for the panel discussion on the topic Roadmap for Increasing Global Groundnut Productivity, a Pressing Issue. Let's hear the experts' insights on this roadmap. I request Dr. Kuldeep Singh, Head GeneBank ICRISAT, to moderate the panel discussion. Over to you, moderator. Requesting Dr. Himang Bakshi to join the panel discussion, please. Dr. Himang Bakshi, please requesting you to join.
Good afternoon. It seems this side is heavier than the other side. Maybe I will ask organizers to look for some more people. I had to admit a couple of things that I was given this pleasant responsibility only this morning. So I haven't really come prepared. If I was knowing it, then I would really have come this better. But organizers did help me in providing some of the relevant things which probably we need to discuss. If there are more students, I would tell them that what are we going to tell? I mean, ask here, discuss here. The panel is already at the peak of their careers, and they are the ones who already know about the policy. They are the ones who are already involved in policy making as well. But their words of wisdom would be really what will matter on the other side, that they can focus their research for next 10 years. So if the audience was bigger, then definitely that would be important. Nevertheless, I think we have been given 1 hour, and we can begin it any way you want it. One is that voluntarily, all of you can give some hints, some advices to the conference and to the younger generation that how will we focus next 10 years? Or maybe we'll be asking we'll be raising a few issues. I will not be asking questions, we'll be raising a few issues. And the first issue which I will raise I will expect probably everyone to have their opinion on the same issue. And if you don't want to give it, you can pass on to the next one. And this is what my personal feeling is as well, that if we look at the crops with which ICRISAT is working, these are the dryland crops, which are traditionally grown in the areas which are harsh climatically, where the input capacity of the farmers has been lower. And as a result, if we look, their productivity levels is lower than a couple of other crops as well. Not always, but in general. So what is happening, if a farmer gets a better opportunity in his or her land, a region gets a better opportunity in their land, means if there's a government scheme bringing more irrigation there, if your government scheme supporting some fertilizer subsidies, other things for that region, the region is getting converted into other crops. So are we in that context prepared? Are our breeding programs really tuned in that way that tomorrow, if we want groundnut to be grown in high input conditions and farmers are willing, farmers have a capacity, government is willing to do it, will these be competitive crops to compete crops like rice? I mean, we are talking about groundnut or soybean, whose productivity is still less than that one. What should be the breeding strategy for next 10 years? That we really see if anything comes forward, you have high input conditions coming in, you have better input conditions coming in, our crops still remain there, they are not replaced by other crops. I'll start from here, and Helia is the plant breeder, the groundnut breeder with a long experience, and maybe we'll start from there and we'll continue this.
Kuldeep, actually, I was trying to sit far from you so that I don't get. This is really a very tough question. Are we breeding for tomorrow? I think I would say yes. We are breeding for resiliency, climate resiliency, as well as exploiting, opportunities for high environment. And the likelihood of, our crop, particular groundnut, being replaced by other crops is low, generally from the trend where it's being grown, where climate change is high risk. But as breeding community, we breed for, quick, combine the genetics, the crop management by groundnut growth this morning, we could increase yield such that profitability of groundnut would flourish and farmers as well as other actors benefit and will not abandon groundnut actually. That's what.
In my opinion, next 10 years, what situation will be? I have seen the present situation where groundnut is getting replaced with the castor crop, especially in Anandpur district. The concerns of the farmers are definitely productivity and profitability per se. That is one issue. And second thing is the animal component with damages, that's also important. And third is mechanization also becoming important, labor shortage, all those sort of things are there. So our breeding strategy should be to go for the maybe difficult but not it is achievable only, that these genome editing tools needs to be utilized in developing multiple resistance varieties. Through conventional breeding programs, things become very difficult. Combining one from 2, 5, 6 germ plots and then bringing in one, maybe a magic population development, these things, it's a not easy task, and then looking at that also difficult. But these gene editing tools should be used in going for the developing for the multiple resistance, multiple abiotic resistance, multiple biotic resistance also become very important. Second thing is valuation of the crop. That is really very important. And just I was seeing posters, and there are certain things where Indian issue of groundnut research has come out with the oleic acid, high oleic varieties. So like that, certain things have to come on that line, so that groundnut in present form may not be much beneficial or profitable, but other form of the high oleic and this market is also open. So that traits also needs to be looked into. And third thing is we have to breed for the resource use efficiency. That is need of the hour. Everywhere resources, especially water resources, they are being constrained, and under those situations also, advantage with the groundnut, other oil seeds also, pulses also. By nature, they are climate resilient also. So that trait needs, that characteristic needs to be taken care, and then see that it is having high resource use efficiency, especially water, and then coupled with nutrients, definitely productivity can be high. So productivity per se will become also important. And last point is, which I always feel I was advocating also, that multiple cropping system needs to be followed. And their need of the hour is that what system we are developing, within that, whether that crop is compatible or not, or even the variety also. I don't know how to do that. But in my practical experience, while I was working on groundnut, while we have worked on the genotype into genotype interactions, and Spanish varieties we were studying in castor and PGNP based cropping system. And again, PGNP and castor also medium maturity, high maturity, long maturity period also there. There, just like that, any variety cannot fit. And we have to really look into that and then see that which variety is compatible and which system we want to promote in another area. So as per that, we may have to. So that is my opinion on it. Thank you.
Thank you. So both the panelists kept more reliance on the resilience. But I would be very happy if we can divide our panel discussion on 4 important themes as far as particularly groundnut is concerned. Productivity 1, profitability 2, then resilience 3, then improved nutrition through groundnut. I think if we deal all these 4 together, I think most of the breeding objectives are very clear in this crop. In the morning, I told this crop is very complex, but very much scope is there for improvement in each segment. If we take the productivity, increasing yield per se, the highest yield we observe in US, 4.4 ton. India is giving 2.1 ton. But in India itself, we could see the farms where they harvested 3.5 ton, many farmers. And in some occasion, we could see that farmers are harvesting more than 4 ton also. Means we are not able to realize the full genetic potential of our varieties in the system which we are having. The reason being that not we are providing good input what the crop is needed. When crop is making oil, when crop is making protein, I think it needs heavy nutrition also. Most of the soil, whether it is a soil of Gujarat or Rajasthan, Gujarat in our India, producing 50% of the groundnut of entire country. At the same time, Rajasthan is the second state which is yielding 25% of the total groundnut of the country. And Rajasthan is the state where the crop is moving very fast. I hope and I foresee that in next 5 years, this crop will take the share of 30%. because other states like Andhra, like Tamil Nadu, like Telangana, the area is decreasing. Means there are certain regions where the specific diseases, specific pests, they're creating havoc. Say, for example, I was summoned by the Minister of Agriculture from Telangana, and there was the havoc of the peanut bud necrosis virus. Can be one of the objective of insulating our varieties against this peanut bud necrosis virus. If it is to be done, then we have to make infrastructure for a strong and robust viral diagnostic techniques and the deployment of virus-resistant gene in our system. Say, for example, Andhra, the crop is suffering because of the recurrent droughts. So there is need to insulate our varieties against the drought. Can we do it? Can we find the loci in our genome which are really helpful in manipulating a crop against drought? There is a situation that crop is suffering more because of the weed menace, and we are not having good post-emergence herbicides. I think weeds are number one, which are responsible for reducing the yield in a farming system. What needs to be done? Whether you are looking for the post-emergence herbicide tolerant genotype, or you are putting good post-emergence herbicide, I don't know. We need to have the solution for that. Fresh seed dormancy. It is a really, really big issue we need to address. Through constant breeding efforts in our system by the breeders, we could not resolve this problem. I'm very happy that your genomic resources and breeding programs could do well for imparting resistance for leaf, late leaf spot and early leaf spot. And you could do well in increasing the oleic acid content in the groundnut. But at the same time, there are certain diseases where we are not having any answer. Say, for example, if the crop has to be moved in Rajasthan in a bigger way, the collar rot is emerging a serious problem, and collar rot is really, really a notorious fungus and very difficult to manage through management practices. Can we insulate our varieties against collar rot? I think if it is possible, need to be done. This crop, if you need to make productive, it is leguminous crop. You need to use microbes which are friendly to this crop. Whether those are the phosphorus bacteria, solubilizing bacteria, or rhizobium or mesorhizobium. I think you need to address those things because those interaction will certainly help in boosting the productivity. Now in the genomics, another dimension is the metagenomics. Lot of good microbes are there. We can identify through techniques of the metagenomics, and we can manipulate the microbiome in the productivity zone, and we can have the increased productivity in the crop. I think for this moment, this is more than enough. In the second round, I will talk more about the quality of the groundnut.
Well, thank you very much to the panel members preceding me, and I'm sure to those that are also to my right that will be able to respond to this very interesting question. Since this is an international conference, and I may need the assistance of Dr. Desai for this, I lived in northern Nigeria for many years. And some of you may know that Nigeria now is the 4th largest producer of groundnut. But if you look into the history of groundnut in northern Nigeria and across that savanna, there was far more groundnut produced in previous decades. Now that had some challenges, of course, around an export culture when food security was maybe not prioritized by the powers that were in place. But the fact is that groundnut had a very significant role to play in the economy of that savanna belt across West Africa, which has declined over the past number of decades. So the intervention that I would like to make on this panel is to think in terms of how we within ICRISAT run all of our breeding programs, where we talk about a target product profile. So instead of breeders just thinking very widely, there's a very specific and a very stated set of goals so that there's no confusion about the work that needs to get done. So in the case of groundnut, what might be possible? We heard from Dr. Mathur this morning that maybe we should be targeting intercropping systems and mixed cropping systems for groundnut. That would need to be thought of very specifically as part of a target product profile. In addition, we heard from Dr. Gupta the idea of having groundnut fit maybe in either new locations or expanding the area here in India. That would also require selection around a particular area as part of a product profile. Others have been more eloquent, so I won't emphasize the areas, but clearly we do think about productivity. This goes without saying, but also thinking about the stability of yield. Should we be selecting for absolutely high performance in perfect locations, or should we be ensuring that the new genotypes that are coming to farmers have a more stable yield across an array of growing conditions, climatic and others that we might be aware of? So all of this becomes part of the product profile. We had the good fortune just coming from a farmer's field. We were seeing the diseases around late leaf spot. We were seeing rust. We know the issues of rosette, and we've heard others. That also becomes part of the product profile. And of course, in ICRISAT and so many of the national programs, and ICAR has done such a job of also thinking about aflatoxin management. All of these things have to fit into that product profile. Now, if you're doing the multifactorial multiplication of all of this, this leads to a great deal of complexity. And I would just add one more. In the case of ICRISAT, we're also very interested in dual purpose groundnut, both fodder for animals as well as grain for consumption as well. So what I would like you to think about is that the challenges that the breeding programs are going to face over the next 10 years that Kuldeep was mentioning in part of his question. How do we start to target in on a product profile in groundnut that actually will have the biggest benefit and the biggest outcome for what we want to do? Because all of the permutations that I've spoken about are almost impossible to handle in a way where we can make true genetic progress. So for the students in the room, to the question, think about that specific target product profile that we want to get for groundnuts here in India, also in other producing areas of the world, especially in sub-Saharan Africa. Thank you very much.
Well, several points have been raised by earlier panelists, so I won't be repeating them, but I would talk more about research culture in today's context. There are two words of groundnut. One word is have, another word is have nots. And these two are very different scenarios. The problems are very different. And even 40 years ago, hello, please, can you keep quiet? 40 years ago, when I was asked to make what are your breeding objectives, I started breeding for high yield, breeding for drought tolerance because it's an in-fed crop, breeding for foliar disease resistance, insect pest resistance, and blah, 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 long list. The same list still continues today. Nothing has changed. We have made some progress here and there. isolated cases, and we can be very proud of it, but still those issues still remain the same. Now, we can still take excuse, well, whatever tools were available at that time, we used them, and that is what we could do at the best. But now you people are fortunate, you have lot of other tools, more precision tools, you can do a much more precise job. But the problem is the research culture. What I see today is not the right research culture. Many of you would not know how to grow a groundnut crop. What kind of fertilizer groundnut required when it is to be sown. So unless you understand your crop fully, how can you do research on it? So to me, first thing for you would be learn how to grow groundnut. Then try to understand what is groundnut. Once you have mastered these two things, then you can start interacting with groundnut plant and you can do wonders. But without learning about the crop and its condition, you can't really do much. Groundnut is a cash crop, we always say. So its research agenda has to be market driven. Particularly in US, I know the crops, of course, these people are here, they will tell you. Slight variation in the quality parameters here and there, the variety is not utilized by anybody and it goes waste. So depending upon what market we are catering to, what purpose we are catering to, that will decide our research agenda. Now for the information is of house, maximum yield in groundnut was recorded 13 ton per hectare in 2-hectare plot in China long ago in Shandong province. In the past, there used to be a 10-ton club in Zimbabwe. Any farmer who could produce 10 ton per hectare groundnut, he was the member of that respectable club, 10-ton club. In India, Maharashtra, 1 or 2 farmers have achieved up to 8 or 9 ton per hectare. So what I'm trying to say, productivity is not the limit. We always say breeding for high yield. The potential is already there. You should know how to realize that potential. And it boils down to agronomy. It boils down to agronomy. Unless you have good agronomy, your genes are not going to function overnight. They would need food, then only they will give you results. So please understand the crop, understand how it is grown, what the crop is, what it likes, what it doesn't like, who are friends, who are enemies, then start working. Thank you very much.
All the previous panelists has said most of the points, but I will highlight some points which are not very research oriented, but strategic. Groundnut, when I started my career in groundnut, we used to call a orphan legume. And morning, Basho told, now time has come for this orphan legume. And we all realized it has potential up to 7 ton or 6 tons in normal condition. One difference between 13 ton, 15 ton giving cereals, rice, wheat or maize, all are carbohydrate manufacturing plant. And it is a plant for lipid biosynthesis, which require much more or double energy per synthesis of per unit of molecule. Similarly for protein. So obviously it requires much more energy calorie inputs to synthesize a single molecule of protein or lipid. So in such cases, if we want to shift from orphan ecosystem to high input ecosystem, our general tendency is nobody will take the risk with this orphan legume. Everybody will shift to rice, wheat, or maize, where productivity is very assured. Because this crop associated with risk, because good crop on the ground, but what is inside the ground, nobody knows until we harvest the crop. So no farmers will take risk under high input condition for growing the groundnut. Always they will, whenever there is a good inputs available or irrigation available, farmers will shift to secured crop for a 15 ton, 15 quintal, 15 ton or 10 ton or 11 ton crop. So in such cases, we have to have a very stable and sustainable productivity. Then only we can shift this crop to high input management condition. Otherwise, we will remain to stay there as a orphan crop. To have all those conditions, I think basic genetics or genetical information is missing. The rice, wheat, or maize, if we see the basic genetical information, basic genetical work, genetics is very, very clear. Hexaploid wheat, rice, maize. In case of groundnut, it is also tetraploid, but genetics and basic genetical work is missing, like rice we have, or wheat we have, or maize we have. So that manipulation of genetical manipulation or permutation combination of genetics in case of tetraploid with a huge large genome, it is not always very easy task. Without having a very clear background of genetics, each and every traits and each and every character. And for that, we need a very, very trained manpower. As Dr. Nigam said, the research, the trained manpower we had in the rice, wheat, maize, several legendary workers, they worked in rice, wheat, and maize. But in case of this groundnut or any orphan legume or pulse, oil seeds, we are still missing them. So we need to have a very, very trained manpower to handle such a difficult situation. And of course, it requires a investment, because most of the cases, major investment is going to rice, wheat, maize, or maximum pulse. Very little investment or resources are available for this oilseed crop or orphan crops. And now we have a good technological support like genome editing, mass, transgenic. Definitely these cutting edge tools will also support. It will be a supplementary to the basic genetical work or conventional breeding work. And I also worked for 30 years with the wild species and pre-breeding. My understanding is that if we want to resolve such type of aflatoxin issues or any critical issues, maybe yield or yield barrier, we have to work on pre-breeding. Very targeted, time-bound, targeted pre-breeding work to resolve all these critical issues. So these are some points I want to highlight. And long-term approach. We have to have a different approach, some short-term approach and some long-term approach, which will benefit immediately to the farming community and some in the longer run. I will give one example for our clearer understanding. This high oleic trait. When we got the high oleic mutant from Georgia through ICRISAT, and we started working on this project during 2011, and we were just referring all the previous work from US. It took Georgia University got that mutant 20 years back through mutation, Florida. And it took 20 years to finalize how to take it for breeding program and precise molecular breeding program. So that identification of locus, then gene, then expression, and ultimately marker to exercise in the conventional breeding program. So such type of work is missing for other traits also. And later on, in our study last 10 years, from 11 to 2020 or 21, I have seen there are two alleles responsible for high oleic, two mutant alleles in A genome 1 and B genome 1. A genome mutant is frequently available across the continent. Any germplasm you say, mutant A allele is available. But mutant B allele, that is the magic developed by Florida. Only single mutant allele available and developed by US. And when these two mutant allele comes together, that gives the expression of high oleic trait. So that is the magic, the mutant B allele here. So such type of work, research, investment is required if we want to solve this aflatoxin related problem or any such very tricky issues. Thank you.
Good afternoon, everyone. Again, my name is Ryan LaPCA. I'm president and CEO of the National Peanut Board in Atlanta, Georgia, United States of America. I have to point out that I'm not a doctor. The tag is incorrect. My training is in marketing, and I was asked to lead the National Peanut Board last year after working for the board for about 15 years. So I have an extensive understanding of peanut science, but I'm not a scientist by training. So I think that's fair to point out because my perspective is a little bit different. I agree with what all of these gentlemen have said, and they have all made excellent points. And I think for the next decade, we really need to lean in and focus on collaboration in a number of ways. Think back to when we accomplished the goal of working across origins to map the genome of the peanut and how exciting that was. And we have this genetic map of the peanut. And now we have folks all over the world who are doing work, but maybe there's some information that he could use from him, but there's no formal channel of communication built into the system that allows these researchers to share information. I'd also like to see us as a world peanut community lean into this revolution that we're in technology, artificial intelligence, machine learning. Imagine the power of technology to take the limited data that we have as a relatively small crop compared to some of our competitors, and to use this information to help us with things like phenotyping and really being able to come forward with our breakthroughs for our stakeholders. And that's the third point, stakeholders. We don't exist in one silo as a scientific community. We have shellers, we have manufacturers, and we have consumers. Consumers are at the top. And what consumers want dictates what brands want, or manufacturers, and that dictates what shellers want to buy. And at the bottom is the grower. I work for growers, and I'm saying this. The bottom of the funnel is growers. Growers will grow whatever someone will pay them to grow. So we have to work collaboratively around this circle of the supply chain to achieve outcomes that meet everybody's needs so that we can move forward as an industry. There's a lot of talk among my colleagues across the peanut supply chain in the United States about, when are we going to start utilizing technology for gene editing like CRISPR? Well, we're afraid what's the consumer going to think about that? We're going to scare the consumer off, but do we know that for sure? Do we know that the consumer won't accept this technology? What if we could reduce the cost of production by $100 per acre in a much shorter period of time than relying on traditional plant breeding methods? And I'll just close with this final point on collaboration. I think that this organization is a starting place where we can have those conversations about how we can work together. How can we come together as a world peanut community to face our common challenges? In the United States, most of our breeding happens at the state level. There's 12 major producing states. So what his state of Texas needs is way different than what my state of Georgia needs. So we probably need to make our own research decisions at the state level. But as a community level, there are common ways that we can come together to achieve results that will meet a broader number of stakeholders. Thank you.
Thank you, Krishat and organizers. As such, all the research was already covered many points. I am right now at the dual angle, being a PG in agriculture, but absolutely in the private sector, and a son of a peanut farmer only. So my point for the research direction will call all the three aspects. Sir, we have to continue the research for both rain-fed and irrigated. High input means agree is not like cotton or vegetables that so much of pesticide and fertilizers are required. So of course, rain-fed is the Berasab is rightly contended. But when we are targeting a mission to convert paddy land, potato land into a groundnut, then the irrigation responsive varieties are lacking at this moment. What I am observing, it is across the globe, applicable everywhere. So because we are coined the word, sir, that replacing 40 lakh hectares. First start with the crop improvement. What strategy we have? Then it goes to the crop production and protection and everything. So something is direction is required to be given to the government. That's really because this is the season is approaching. In this kharif, this much of paddy area we want to give the groundnut. Then what variety we can give, right? This should perform because if the first itself we give the wrong variety, then the perception is going to be wrong, right? So we have to be very particular and we have to recommend that. Second, the Indian aspect, if you can see, the fodder is also playing a crucial role. Lepoxy 1812, when it is released, it is popular like a wildfire. Through Dr. Nayak, I have contacted and I have multiplied here. But the fodder that I think in ICRIP Jaipur we discussed, nobody was knowing, but I was knowing, being a farmer son, that the fodder of lepoxy was creating a diarrhea in the animal. So here the fodder and foliage and that biochemical parameters are very important to study. Because this crop is Berasab also shown to be studied, and every farmer is keeping 3-4 cattle minimum in India and in the groundnut, so the rain-fed areas. So that aspect is required to cover. In our many areas, particularly, sir, we know in the Source ball, salinity tolerance. In salinity area, we have no replacement right now. Even that 11 number is still 11 they are asking, and 20 they are replacing. But still, the salinity tolerance is one of the important aspect which is required in the Saurashtra ball of the Gujarat and many other areas also. Similarly, some of the groundnut varieties, farmers are not preferring that seed production, particularly the Vidarbha area, which is Rabi to summer cultivation. Tech 24 seed production is a very difficult task. Tech 24 is in the mind of the farmers. One thing I was saying to Gupta Sahab, that is very difficult to change. Either we give 24 plus, but 24 farmer won't, 11 farmer won't. So that seed production aspect is also required to be evaluated, because those are the varieties which is performing very well in the Vidarbha and Khandes and many other part of the countries also. And of course, mechanization. Germplasm suitable for the mechanization is very essential, because labor is starving at everywhere. So the research strategy should go in that direction also. So the farmer aspect, I tell you that apart from I don't know comparison with paddy yet, but still I have compared it in the Bundelkhand area. Apart from paddy, soybean, and some of the other kharif crop, groundnut is performing very well. This year, even with the less price per acre, 50,000 rupees farmer are getting in Saurashtra. It is the price is lost this year, but still getting, because the production is very high. Our figure is going to certainly improve with Girnar 4 and 5. The production is very high. Even Gujarat condition also, sir, 4,000 metric ton, 4,000 kilo per hectare is achieved. So that variety needs to be spread at other area, because our India's geographical likings are different. The Rajasthan, Shekhawati, that whole area, there the irrigation, even US also, they have 140 days, 150 days preference. Gujarat is want for the second crop. Maharashtra is also rain-fed, so 100 and 120 days. So that all line we are required to cover in the research aspect. Last is not related to the topic I have so told, but the sir is here. The soft lawn for the seed industry is very much required. We are import substitute one, and we are getting loan as a construction industry rate. That and how a seed industry can progress, sir. It is a very important aspect. In cotton, you got 30 companies are there. Yesterday I was there in Ikrip Cotton, 30 companies are there, and sponsor list are full. Very difficult to find out name. Here the sir is calling, and we are uniting everywhere. Just so, because there is not a soft loan and infrastructure is just for one time, but this soft loan continuous purchase of groundnut is very much required. Otherwise, no extension. Our R&D is not going to reach to the farmer level, because state corporation, everything, they don't give on the debit. Farmer in every place, majority places want everything on debit. State corporation, NSC, they are not giving anything on cash only. So private players ventured it, but they cannot make the inventory, because there is no soft loan for that. Our loan not treated as a construction cost. Loan construction builder is asking the loan, same rate we are getting. We are import substitute rather doing that. So these are few submissions. Just thank you very much.
Thank you. I'm sure, although I tried to make notes of most of the things, but may not be sufficient. I'm sure the organizers may be making some notes, because these are the things which have come by heart out of at least 25 to 30 years of experience. Realizing that the time is short, I will try to modify this discussion a little bit differently. So what I say, I have formulated 4 issues, and I will throw these 4 issues to 2 people each. I will just read these questions. Probably you can make a note of it and then respond to it. Well, in fact, what Dr. Vera told is the time required for anything, what you said for this mutant, and the hap-inducer gene, which has become a commercial haploid production system in maize. It was discovered in 1963 and became commercial sometimes in 2010 or so. So all these things, definitely it requires time, all those things. And we don't have a hap-inducer system here in groundnut. I mean, that could be something to this part of the audience that if they can think of. Okay, how I would like to really move this debate is the first thing I will like Bera and Hele to respond is, if I look at this morning's historic lecture about the Peanut Genomics Initiative started in 2004, while we in wheat started talking in 2005, and it came into existence in 2010. Yes, the whole genome sequence of wheat came earlier, but peanut also came. But if I look at the practical application of the markers in wheat, rice, maize, vis-a-vis groundnut, we are lacking behind onto this one. Can you please find out the reasons and propose 1 or 2 suggestions? Because we're talking on AAGB, that how can we really make markers more useful for breeding groundnut, peanuts, traits and other things will come. This is the first group. The second group, probably I may ask Emma and Sanjeev. Genome editing, it has become a reality. The technology has come to age. The government of India has really deregulated SGN1 and SGN2. The two things remain. For the research community, what could be the most important 3 or 4 or 5 traits we should focus for genome editing? And second, even if you have the gene edited products now, there's no regulation. Really thinking of having a joint, a single license for the country, or it is to go the individual. So this is both of your question will be asking maybe our issue, not a Dr. Nigam and Rayan to respond to that. See, aflatoxin is not a new one. It's an old issue. And what Dr. Nigam told this morning, that they really tried and gave up at some continue. And what are the lessons learned? Maybe we can have, how is the stringent conditions in the Europe and the Western part of the world? And what the scientific basis, the genetic resources, be it from the cultivated or from wild species, how can we use these for really? And then, of course, my last issue will be to Stan and to Mathur, that how can we really enhance the global collaboration for taking the genomics, breeding, production, productivity, and all issues of peanuts at a global level. You, maybe you can go the same way as I raised the issues, one by one. So maybe.
Thank you, Kuldeep. I think the genomics question should have been suited to genomics people, but yeah. So I think, tremendous progress has been made in ground genomics the last couple of years. Manish Pandey, his lab, Rajiv, collaboration with universities in the US. We have a lot of genomic resources. The foundation is there. I mean, as you said, the question is how we should, apply genomics and ground improvement. So far, high oleic and LLS markers have been in use, but there are no markers for other important traits like rosette, early leaf spot, which are the main disease, particularly in Africa. And there is a need also to develop, genomic selection tools for very important traits like grain yield and drought tolerance. And as I said, I think the platforms are there. We have, sequencing, genotyping platforms for low density, mid density, high density. And it's up to us really to collaborate, to come together, work in a partnership that could benefit, the groundnut sector, could develop populations, mapping populations, to discover markers, to validate markers, and also apply, marker selection. And I think it's high time, we collaborate as, stressed from the high table here, and partnership. Even the DG mentioned it the morning during opening session. I think we should come and work such a way that we deliver all the technologies that the breeders can use them immediately. And genotyping is just easy now. It's getting even cheaper. Even we don't need to develop the facilities here and there. There are also facilities at commercial level where we can outsource all these genotyping and sequencing work. Yeah, thank you.
To my opinion, probably we are this genomic group and conventional breeding group, or maybe breeding group, molecular breeding group. working in isolation. Because of the huge development of the subject, it is very difficult for a single researcher or two researchers to venture equally on that genomic resource tool and handling all those informations, and again extracting all those information in usable form under field condition. It is not very easy task. So once we develop this genomic information or genomic tools, somebody must be there, or we have to join hand to have all those information in such a simpler form that one breeder can use very easily in his lab or in field, so that traits are introgressed with the high agronomic background and job is done, delivered. So probably that is missing, because I am a basically cytogeneticist. I worked on cytogenetics, then worked on breeding, then to some extent on molecular. But when we receive all these molecular tools or information in handier form to use, because being a conventional breeder or simple breeder, it is very difficult to understand all those high things of genomics, genomic information, how to use in the field or day-to-day breeding program, unless it is very handy. After 20 years, in case of high oleic, that handy marker came, and it is a simple job, just crossing and check the segregating population with that easy marker. And that help us to develop this, all those high oleic lines. And that is similarly for foliar diseases. But there are a lot of marker genomic information in publications, but not on use at all in the breeding program. So somebody or collaboration has to develop so that all those informations or resources are friendly to the breeder, and it is really giving the fruit to the variety in terms of variety or reaching to the farming community. So that is missing here. But in case of rice, wheat, maize, particularly wheat system, that time this much genomics was not there, basic cytogenetics was there, that aneuploids, that ploidy, the exploitation of that ploidy and anther culture or haploid technology, apomixis, we exploited in all those cereal crops. Probably that helped a lot. But now we have a much more better technology, genomic resources, all the cutting edge technology, but probably we are missing that bridge between high science and field science. Thank you.
We are asking for industries, industry environment, things they are very, very important. Then of course the disaster, you see
Basically, I have some reservation. Of course, the SDN 1 and 2 is okay with our work. Yet our crop is not in the purview of the NGOs and ethical issues people. That a lot of GM or biotechnology work we do, and ultimately the crop is going to be killed. So use it that from the quality parameters aspect, not that transgenic or things which can going to harm it. Not not to think on that line, because we are right fire aware, nobody's discussing, but only the aspect, the markers of sensor-based markers were in the field, because now everyone want to sit in the home and they want to control their farm. That is a reality in India now. So the drone-based sensors can be created there where the nutrient like is there in the field for the groundnut, and because the other crops there is some in advanced stage, but in groundnut yet there is not much breakthrough. When I have rather not found much the drone applications found in the groundnut field. So that sensor-based some identification in the field if it is there, because the best it was the white grub in those stage, but now it has drastically gone out after the this particular 32 and Girnar 4 in the pipeline. Earlier in 20 GG20 and other it was very much there. So that aspect is not that here. So anyhow, these are some points. That's it. Thank you.
I think very pertinent question when we resorting to the genome editing technology for the crop. You have raised that licensing. This is a major issue. Department of Biotechnology, ICAR, they are in the dialogue to have a group license for the organization. And that is important. And I think those issues will be resolved shortly. But before that, our research should continue. We have to do a lot of work. In genome editing, you need to increase the transformation frequency, the first step, and each crop group should focus on increasing the transformation frequency. The second is vector assembly. We should have the state-of-art technology in our laboratory to assemble the vector, and then expression. I think these three, there's a huge work our researchers are continuing in the research laboratory. ICR is focusing on few crops, and we are ready with the product in rice. 2 products are already in the trial, and 2 products in mustard for low glucosinolated content. Those are in the trial. I think in the next 2 years, they will be ready for the public domain. But before that, the licensing issue will be resolved, I hope.
Well, I'll try to summarize the present status as far as conventional breeding efforts and management efforts. Aflatoxin, always we think of resistance, we ignore the management aspect. If we do the right management, we can eliminate the problem of aflatoxin from the field. In conventional breeding, our success was that we could transfer the resistance in other genetic background, but we could not improve on the level of resistance. Probably there were very limited number of lines which showed some resistance, and the genes, they probably all carried the same genes. So we could not improve the label, but we could transfer it around. But that was not the purpose. We wanted something much more that now that we need zero tolerance to aflatoxin, particularly in Europe, then the requirement becomes very difficult. Then what we thought, the 3 places where the aflatoxin pathogen will face or encounter the resistance. One is the pod wall in case of groundnut pod wall, and second is seed coat, and the third is cotyledons. Now, when we talk of cotyledons, we are talking about aflatoxin production. When we are talking of seed coat, we are talking about seed colonization. So there are reported resistance sources for these, and moderate level, you can say. We thought maybe they will carry the different genes. So what you call today magic population, we made the convergent crossing, 8 or 10 parents, hoping that we would be able to combine these three defense mechanisms in one genetic background. But again, we did not succeed. And we were using always very toxic strain. The biggest problem in this is sampling. In a susceptible, in a single plant, not all pods are infected. Only 1 or 2 pods would be infected. Remaining 98% of the pods would be healthy. So how do you do the sampling to capture it? Even if one pod is susceptible, that means the genotype is susceptible. So sampling creates problem, and problem is complex because they're interacting factors. One is plant itself interacting with pathogen, these two interacting with the climatic factors, and then these three together interacting with edaphic factor. Edaphic factor. So that there's so many interactions playing in this situation, and very difficult to capture what is happening. So that's why we tried, and then we gave up really. We thought now with new tools, some work was going on in Georgia also. Again, initial results were very encouraging, but I think in the end they also flopped. Using your genetic transformation and others, at ICRISAT also GMO approach was being used, and nobody was really convinced that the results are coming. What I would suggest, genetic registry is important because it is easy and very cheap source of resistance. Management issue is very important in aflatoxin. If your rainfall in Kharif season is well distributed, you will not have problem of aflatoxin at all. Irrigated crop in Rabi season, again, there is no problem of aflatoxin. The problem arises when you have irregular rains, and particularly if there is drought, when the pods are developing. The micro fissures on the pod wall and seed coat, they allow fungus to penetrate. So we need to give equal attention to management aspect if we want to really have a handle over this problem. Thank you.
Excellent. Well, I agree with Dr. Nigam's perspectives on aflatoxin. Of course, we're talking about it because it's a significant market access barrier, aflatoxin. I think that if we were in a perfect world, 100% of our peanuts would be irrigated and we wouldn't have an issue with aflatoxin, but we are not. So we have to use all of the tools at our disposal in order to come up with ways to tackle this barrier to market access. But also, I think sampling is a really critical component that a lot of people don't talk about, and the failures in devising sampling protocols that lead to a truly accurate outcome, because aflatoxin is one of those things. You could have a container of peanuts and you just happen to pull the one, probe that has more aflatoxin than the rest of the sample, and you're going to, those peanuts are going to be rejected at port. So we just have to keep working at it. We have to keep working together to solve this issue that we all face, all origins, and I'm confident that we'll get there.
I have 2-3 points. One was on markers. Definitely marker-assisted breeding is must. And what are germplasms we are having? Other day, while we were evaluating that how many germplasms are in use, we are finding it was only 4 to 5%. Remaining we were not using actually. So spectrum of utilization of germplasm also needs to be taken care. And before that, state-specific evaluation of germplasm needs to be done. Probably for aflatoxin, whatever we have evaluated, or what information is there now with us, still many are left out without being evaluated for that aflatoxin resistance and so on. So that needs to be done, sir. Utilization of spectrum of Gen Plus. While we are talking of, you asked me to talk on the global collaboration and this. It is really high time for collaborative programs. And as our institute also, we started these things, signing up the work plans with other institutes. And then what we don't have, others have. What others don't have, we have. And in that way, mutually, we can come out with the products on mutually agreed terms and conditions. MOUs are also almost standard MOUs are available, where credit can be shared with for the publication, product, IPR, all issues. So very clear-cut guidelines are there from ICR, and we are working on that and using it. But if you talk of the mixed farming or intercropping, then whatever present modules are there, 3:1 ratio or 3:2 ratio, like that, one crop to the other crop, it may not work while we are talking of mechanization. The drone technology we used in our safe flower crop. Now, safe flower require one particular sort of treatment one time. Other intercropping may not require that one. But if it is one is to turn like that, then it will become very difficult. So you have to revisit the production technology on the cropping system also, maybe strip cropping or whatever is there, so that that can be taken care. As far as aflatoxin again is concerned, harvesting is very important. Labors are nowadays constant in many places. And what are traits you identify that for mechanization also, maybe for harvesting, what are the traits you look for? Maybe strength of the peg. If it is strong enough, then it may not be there in field. But then management also aspect become very, very important here, that what are left out ports are there in field. Generally, farmer will collect that also and mix up with the good produce. And that one or two maybe source of infection, and then those sort of things are there. So if it is moderate resistance, Nigam sir, what I was thinking, that absolute resistance may not be available also. But if it is moderate resistance for the aflatoxin, plus aflatoxin management package, whatever is there, these two things can lead to good harvest of the produce. Thank you. Thank you.
I think in the interest of time, I can be fairly brief. If I think about the progression in the history of science, sometimes we think that we are almost at the end of the work. And if I think about all the great work that's happened in groundnut, but across crop science, we have made so much progress over the last century. But I would like to suggest that if we're really going to have global collaboration now, it's going to be about data. And I would suggest that we are going to be in the pioneering, in the earliest stage of how we will actually use data more efficiently. So many of you are data researchers working on groundnut, but how are we going to get all of your data, all of the data that is represented on this panel into one package so that we can actually explore those data to find the answers to some of these very difficult questions? I don't even need an agricultural scientist to do some of this work. I need the data and turn it loose with the machine learning people and the others, the reinforcement learning people, to find maybe those 50 genes that control some of the biggest issues that we have that even the best and most experienced breeder could never track down. So my suggestion around global collaboration is around an aligned interest in bringing data together so that we can interrogate for those most important of issues that we all know and that we've all discussed. And that is where all of us have a new challenge, because 10 years ago, this was not possible. But today we can be the pioneers. And I would suggest that AABG, just like in sequencing 10 or 15 years ago, could play a major role in how this data is annotated and brought together in a usable form. Thank you very much.
While our talk has started, I am in contact with a few crushers, oil exporters also, and I dropped the question what you asked. This is strategies in the for should be there. Surprisingly, aflatoxin no one has highlighted. The exporters and who are oil crushers across the place. Of course, the late train related damages. So to have some, because these are all the crushers and exporters. Ultimately, they are very useful in the chain because they are ultimately going to create the value for all of us. They the late trend and this what biotic, abiotic, and climate resilience, that sort of work they are asking because last year also there are 25 to 30% surveillance of groundnut damaging groundnut because of the late rain. Fields were ready and the rain has started and it has flooded everywhere, and it is repeating since last 3 years. That's what they are raising the point. So anyhow, because these are the feedback which is very important. Second, drought tolerance. All of a sudden, the heat fluctuation. Right now, the summer groundnut is in the very small stage, and the heat has suddenly crossed 40 to 44. That summer groundnut area is that is what biotic stress work. Rest pesticides, and that they are not worrying with. Yeah. That is what they think. The 3-grain seed. 3-grain seed is the word which they are using. They want maximum. That it is the requirement. Otherwise, 50% oil is sufficient for the country and export purpose also. That is what they are saying. High oilic acid is turning the table for groundnut. That's all. I put the Google form.
I think if you're going to conclude, then before that, much stress need to be given for the collaborations, partnership, consortia platform. I think these are very important because there are certain issues which remain unresolved. We could not address the fresh seed dormancy problem appropriately. We could not resolve the aflatoxin problem appropriately. Drought tolerance, we could not impart it in our varieties. There are certain issues that still remain unresolved. I think these can be resolved with the partnership. We cannot say that groundnut or peanut is a orphan crop. Be sure we are putting umbrella of CG Institute and many organizations, they are funding. Not only they are funding, they are doing good research. Here is the time to have a bilateral arrangements. Say, for example, if University of Georgia or University of Florida, if they are having the state of art of technology for managing one issue, can't we go one-to-one partnership to resolve some of the issues? I would like to make a share in Hindi, if you allow. सारी धरा भी साथ दे तो एक बात है. If you are having a global partnership, it should. But अगर तू साथ दे तो और बात. वैसे तो कुछ लोग हैं जो एक पैर से भी चल सकते हैं, अगर दूसरा पैर साथ दे तो और बात.
Yeah, thank you. No, he has summarized the importance of the collaboration actually. 1:1 is 11. That's what he actually meant for that one. I think I would request you all, I mean, it's difficult to summarize the thoughts of 8 people who are the apex of their careers, who have 30 years of experiences, thoughtfulness. But I will request you to join me in giving a big hand to all of them.
Quickly, I'm going to spot it out for the audience. Can I get a one word? What did you gain from this panel discussion? Quick from the corner, please. One word which you can summarize it. Good. Other one? Good. I was just checking whether our audience are awake. Thank you. You have done a wonderful job. From the savanna belts of Nigeria to Indian farms, a greater wisdom on peanut was shared here. Master your crop first and travel all along to target product profile or the systems approach. Tech advancement, AI, ML, and big data is a boon. Reap it well for boosting translational research with competitive funding and collaboration as well as thick partnership. Together we team up to turn the challenges to tangible impacts. That's the big message we are taking from this panel discussion on roadmap for increasing global groundnut productivity. Please put your hands together for thanking Dr. Kuldeep Singh, the moderator, and our wonderful panelists for bringing in greater wisdom for this discussion. Thank you, one and all. Volunteers, please help us out with clearing the stage. Next session will be session on gene bank genomics and pangenomes. I request Dr. Ran Hawao and Professor Kenta Shirashava for co-chair and chairing the session. Before they set up the stage, a quick heads up for the speakers. Speakers are given 20 minutes time here. Around 18 minutes, the first alarm will be set for you to wrap your talk. And within 20 minutes, everybody need to wrap your talk. Please, everybody stick on to your timing so that we can all join the early dinner and enjoy the networking dinner. Thank you, one and all.
Hello. Okay, good afternoon, everybody. My name is Dr. Ran Chovav. I'm a peanut geneticist and a breeder in Vulcani Center, Rishon LeZion, Israel. And together with me is Kenta Shirasawa, and he's from Japan. Maybe you want to say.
Yeah, yes. Good afternoon. My name is Kenta Shirasawa, coming from Japan, Kazusa DNA Research Institute. So I'm focusing on plant genome sequencing, and now we are collaborating with peanut breeding station in Japan.
Great. So without any other, we can go directly. So our first speaker, everybody already know him, and he will speak about harnessing gene bank diversity for further ready crops.
Okay. Though the conference is on peanuts, but what I was asked to really talk in general on the gene banks, I would say 50% of it will be on general, maybe 20-25% on peanuts specifically, but then other issues as well. My slides are a couple of things which are general. Most of you know it, but setting a stage, we need to really repeat those slides sometimes. Well, this is what gene banks are actually contributing directly to the 4 SDGs, but genetic resources are contributing to agriculture, other ways as well, and agriculture is contributing almost directly or indirectly to 12 of the 17 SDGs. something known, but these are some of the challenges we have because we have to see what are the major challenges. This slide I made in 2015, and it actually turned out to be true. 2015, it was expected that India will surpass China, our equalized China somewhere 2024, but in 2023 itself, we surpassed the population of China. So that means the demographic scientists, their calculations seem to be quite appropriate that world may reach sometime 9.8 billion, or India will reach 1.66 billion by 2050 or so. So and that means if we take 2015 as a base, we really need to improve the productivity by 70%. Those are general calculations. And those calculations, but we look at these things now, say, the climate change. It has happened, it's not happening. You have the 1.4 degree mark of temperature reaching last year. We have the incessant rains, 500 mm last year in Chennai in 48 hours. Drought spells, I will give you some of the examples, very severe ones. I say the drought spells are back, which were a part of our agricultural system in 70s, 18, but in between, there were not much of them there, but now they are back. And of course, we know the land degradation has all happened. And if we look on this one, these are a couple of examples of the drought. For example, what happened in Zimbabwe in 2023-24, complete maize crop was lost. The same thing happened in Karnataka state of India in 2023, very heavy losses in to corn. This is despite a crop that is being supported by public sector, private sector, international institutions, and everyone claiming that, but we have really achieved something important when it comes to drought. But when it was the real drought, we couldn't really reach. And here also you can see here this one. This was pearl millet, we generally consider as a crop which has in general very high level of tolerance to drought. These are 600 accessions of germplasm growing at the farm, at the Ecrisat farm in Bulawayo. Not even a single accession survived. And based on this one, I will tell at the end what we are doing now to respond to that one case. Temperature. Now, this is again an example of 2024 here. I mean, at Ecrisat campus, this is a perennial tree. You can just see the temperatures in April were above normal. And all the new shoots that came on this tree, they were all lost. And they never recovered. This happened in April and the photo was taken in October. So all the dead leaves were there. Imagine if it was a fruit crop, how much losses would be there. So these are things which are happening, which are really to us. And of course, nutrition, we also know that we do lack a lot of the things when it comes to nutrition. All these things are very well established. They're not the domain of mine expertise, but this is information based on that. My next talk, this talk is based on. Having said those are the challenges, we have the opportunities as well. And the genetic resources are the ones which are here. This gives you, if you see globally, almost 7.4 million samples collected are being conserved in 1,700 gene banks in the world. And the CGIAR gene banks conserving 11% of that one. But what is most important for the CGIAR gene banks, if we look on 2020 data of the total germplasm that was exchanged globally, 78% of that exchange happened only from the CGIAR gene banks. So that is the importance of the CG gene banks when it comes to not only holding, but the exchange of the germplasm as well. And of course, this whole information, anyone who's interested, you can have this views. There's an FAO site where authentic data on the availability of the genetic resources, though there's a duplicity in this one, that's there, but most authentic data anyone can find from here. Just, the CG gene banks which are involved, and coming to the Ecrisat gene bank, Ecrisat gene bank is currently hosting around 131,000 accessions, of which 15,000 are of the peanuts to the groundnuts. And 400 of these are the wild species accessions. It's not only conserving it, we are actually sharing the germplasm as well. Since inception, ICRISAT has distributed 206,000 samples across the world, both to the CGIAR breeders as well as to the other researchers as well. ICRISAT Gene Bank is probably one of the very well characterized gene bank. I don't have a hesitation in saying that. But if I look among all the crops, the most ill-referenced germplasm is the peanuts. It's not a very good where we have the complete information where from the germplasm these 15,000 accessions have come. We don't have a information of around 50% of those only. But I will tell what gene banks are doing to really see those. We cannot redo what has happened, but really bring them somewhere into the folk, more usable form is I will tell you what we are doing in this case. That was the International Gene Bank. And if we look at the National Gene Bank of India, which is hosting almost 463,000 accessions of around 1,800 crops or so, of which the groundnuts comprise again over 13,000 accessions. And there is a good overlap between the germplasm of the groundnut being hosted at the National Gene Bank and the germplasm being hosted at the ICRISAT Gene Bank, because ICRISAT Gene Bank got a lot of this germplasm primarily from the national system when it was established, as well as with the collaborative collections until 1993 when the CBD came into the formulation to the effect. By that time, we had good this one. But we know there's almost a good overlap between the two. This is what shows how ICRISAT has distributed this germplasm. And if we look into distribution and then its use, you can see the number of the varieties which have been released by the national partners, either from the breeding material or from the genetic resources directly supplied to them. Almost 250 varieties of groundnuts have been released globally in 39 countries from the material which either was a part of the breeding material or the genetic resources gene bank material directly in that one. And this I will show that when we talk about the gene banks, they have the essential activities. And we have to do these essential activities without any fail. That's the minimum requirement we have. We are doing it. How we are conserving it is not that important at the moment. But to share with you that all the genetic resources which are at the gene bank of ICRISAT, they are safely duplicated at 2 other places, one at Svalbard and one in other 4 gene banks, because no single gene bank could really accommodate all our resources. So this is how we are saying at ICRISAT, we have 2 copies, and 2 other places we have 2 copies of these ones as well. And this germplasm, for example, not all 131,000, but a part of that is conserved in the 2 regions in Africa, WCA as well as ESA, depending upon the crop they are actually looking for. For example, groundnuts is conserved in the eastern part of the Africa as well, which is easily availability for the people who are in the Africa region, this one. And of course, as I told you, that making these available, making knowledge sharing, field days, making this available to the participants is there. But then what sort of research we are doing for the future purposes to make them. So our research will be focused on 2 aspects. One is to enhance the operations or efficiencies of the gene bank and to enhance the use of the germplasm. This is how we are doing. And if we come to this one, the first one that is on enhancing gene bank operations efficiency, the gap analysis is something which is very, very important. And this I will tell you here. Based on, as I told you, that we have 13,000 accessions, but if we look at the gap analysis, this has been done in the South American part, which is the primary center of diversity and the center of origin for the groundnuts. of the 1,659 land races which are documented in this region, ICRISAT Gene Bank has only 1,000 of that one. And we see there are almost 2,000 or 3,000 region accessions are the spots which we have not accessed yet. Now, because of the CBD, because of the plant treaty, we may not access them directly. It has to be done either in collaboration or the people in South America have to take a note of that one. This is a concern actually important for us all. And as I told you that probably this is, we have only information, exact longitude, latitude information of just 50% of the samples. How can we really see the other 50% for which we don't have an exact information, whether their set will be genotyping all these 13,000 accessions. 8,000 of those we have planted now. They are taking the DNA samples. Remaining 7,000 we'll do next season. So by 2026, we will have the DNA of all the 13,000 of those. And at the same time, we are characterizing this one. See, have a look on this one. Yeah. This is the root structure. Since we are taking the DNA, we have analyzed these 6,000, this 600 land races which are planted, the variation in root structure from just 7 or 8 centimeters to as long as we have found only 51 centimeters. Dr. Nigam was saying that he was able to identify even up to 1.2 meters as well. So that's the sort of variability when we're talking of the drought tolerance. What are the mechanisms which we should really target for the drought? We are also looking at the nodulations of these one. We didn't ask any, there was no issue, time was less, otherwise discussing the nodulation, the nitrogen fixation would have been an important area. Do we have sufficient information available for the variability in the nodulation, variability in the actual nitrogen being fixed in per unit area? That is something we don't have much information. What we're doing here, the top 200 or so accessions, which have the more nodules, bigger nodules, we'll be really identifying those and started working on those to see if we can really have some improvement on the nodulation as well as the nitrogen fixing in those crops. In addition to that, this whole germplasm, we have the core sets, we have the mini core sets, we have the trait-based sets, and we are actually accumulating more and more trait-based sets so that the breeders and researchers can really start using those ones. I'm sure this will come in next. We have a lot of posters. You will have a lot of presentations from Manish as well. They will tell how they have been working on the genomic utilization for characterizing the germplasm. I will not go into details of those ones. They will tell it there. But what was more important here, some important traits, for example, I was asking Dr. Nigam that what's we say, 15 accessions have been identified as tolerant to aflatoxin, but still we have not been able to see it. And I think he was very candid in telling that probably would not know the variability, whether these 15 accessions have the same genetic mechanisms or different genetic mechanisms. And of course, our genomics group has to really look into developing a magic population of this one to see if they are same or different to take care. So for all leaf spots, we have these important informations available. Using the mini core, the group has been able to identify the sources which are resistant or tolerant to pre-harvest sprouting, or the dormancy you can call, and also identifying the DNA markers. Those could be used for the marker-assisted breeding of this one. This information is available here as well. And also, all the wild species we had, you screened that those last year with the late leaf spot, because we know the source of resistance for the late leaf spot is coming from a single line, single wild species, and that is breaking at many of the places. So we really need to look into the more and more susceptible resistant sources. These have been identified. This information is available with us. Another species called as Galabrata, which ICRISAT is maintaining for last 30 years as a vegetative part. These were screened for this disease, and almost all the accessions were showing immune reaction to late leaf spot. So that is a good source of resistance to this one. Okay, I will take 2 or 3 minutes that how this whole system, if we really have to see the gene banks, we really have to characterize the gene banks, how can we do? I'm taking one example, that's a collaboration between ICRISAT and NBPGR. This collaboration, and in fact, the national system, National Gene Bank at NBPGR, is doing this sort of work for 16 crop groups. And I will take example of one, that is chickpea. We had the cores of chickpea here at ICRISAT with a core of chickpea at NBPGR. This is almost 5,000 lines. These 5,000 lines have been genotyped through resequencing. These have been characterized for all the important agronomic traits. These have been characterized for almost all the important diseases at 16 different locations in the country. So this sort of information, so we have these 5,000 most diverse germplasm lines, genotyped, genotypic data is available, phenotypic data is available for all the agronomic important traits, and here is what the breeders can start using this resource. And we need this sort of actually collaboration for the peanuts as well. I don't know whether Bera has already left, but if he's around, then probably this could be a proposal to him that we need to really bring groundnut also into this portfolio and see how this has been done for different diseases. I'm taking Fusarium wilt as one of the examples. This is within India, we have out of 5,000 accessions, 131 accessions are resistant to Fusarium wilt. Now, and this is from how they have come from different states in India. This is how they have come from different countries of the world. These 131, we will have some information based on that. These 131 should be different from each other. I mean, there should be 5 or 10 different groups coming from this one, what we are really getting out of this one. Similarly for Ascochyta blight, similarly for a disease like the nematode, out of 5,084 accessions, just 2 accessions only having moderate resistance to the nematodes. So this is the sort of work probably gene banks need to do. Another example is from sesame. Sesame, this is again a collaborative project. And for sesame, the water logging is an issue. If the water remains stagnant for 24 hours, the whole crop is gone. Just screening 6,000 accessions, we could identify based on initial examples, a few dozen accessions which are showing good. But when we treated them in the field, in 96 hours of water stress, we got a couple of accession, and this one accession, the one you see on the left here, this one. This is the, I'm sorry, I cannot show it. There probably is not. This one, what's one accession that is showing tolerance to both water logging, and there was no further phytophthora development. Out of 6,000 lines, just one line coming out of this one. Same way we are repeating this one in pearl millet. As I told you that not even a single accession survived in Bulawayo. We planted a core of 2,400 accessions directly in the field, and we are withholding the water. After 30 days, you can see most of the lines, are 95% of the lines died by 30 days. You have one line there that's not showing much effect of water drought at the moment. So we have about a dozen such lines which are coming, and we'll be really looking into the mechanism of what breaks them drought tolerant for 30 days. And that sort of thing what the gene banks really have to do. I think my time is over with this one. My sincere thanks to all my colleagues because the presentation I made is based on the work of, I think, hundreds of people from the gene bank over the years. So, thanks to all of them and to different funding agencies who really made the funding to us. Thank you very much.
Thank you very much. I think we have time for one very quick question, please. If anybody have.
Yeah, it's a good presentation, very informative. And my question is, in your germplasm collection, each line the homozygous or you have seen any heterozygous?
Which crop are you referring? Pearl millet?
For the peanut.
Peanuts. No, peanuts primarily are self-pollinating, but there may be mixtures as well. We don't deny that one. Initially, when I said we are screening this one, we are taking a single plant and we will be maintaining the identity of single plant. And what we're doing, the single plant which we are taking is a representative of the whole germplasm. Very clear gene bank material. I mean, groundnut, it may not be heterozygous, probability of heterozygous is less, but yes, it could be admixtures. We are careful about that one. That's why we're taking single plants on.
Okay, I think we will finish now. And thank you very much. So our next speaker is a great colleague of mine, Dr. Charles Chen from Auburn University in Alabama, USA. And his talk is going to be about GWAS reveals genetic insight into disease resistance of leaf spot and seed germination and dormancy in peanuts.
Okay, good afternoon, everyone. So first, I would like to appreciate the organizer to invite me coming here to host this wonderful meeting. And today I would like to bring some of our PhD student research on the leaf spot and germination and dormancy. Before I really go to my research, I would like to acknowledge my collaborator, Dan. He is my PhD student, and other collaborator coming from the USDA ARS, Hansen Alpha, and also University of Georgia. So the name, I don't need to read the whole name. They are familiar with in the community. So that's why we are doing the leaf spot research here is the peanut Research Foundation. So we are being in the USA, we identified some very important tree, desire tree as a breeder to work with, like a leaf spot, river, aflatoxin, and then drought. Those are the four the top priority. As to talk about the impact of the leaf spot, so we know have two different leaf spot. One is the early leaf spot, another one is a late leaf spot. So early leaf spot is caused by fungus of Cercospora and Acacia decolor, and both the disease has the big impact about the yield. You'll lose like go to 70%, and also like staying, they will be mainly is defoliated the leaves, which will impact the photosynthesis. That's we are working on that. Another trait I'm talking about here is talk about the seed dormancy, also the seed germination. As you know, in American, we growing all the Arowana type, which is the hypergea. We don't have a lot of problem about the pre-harvest sporing, but you guys growing a lot of the Spanish. So there's a big problem when you harvest the season as a raining can, they were sprouting. So I think it's very important for the looking for some of the dormancy. Also in the American, we also very concerned about the germination. Germination is the biggest seed problem when the farmer to growing planting their land. So here is there are two publications coming from my lab. So one is published on the TAG 2020. It's talk about the GWAS on the leaf spot. So we use the mini core collection as our genetic diversity panel doing the GWAS. And we use genotyping. We know we use the F-Matrix 2.0 NEEP. And with that, publication, we really identify some of the same thing for the dormancy, also the germination. We do find some of the QTL over there. But after a couple of years, so the new technology is like pretty available. So we're doing the whole sequencing for the mini core. So because so then my student, she's interested in say, okay, let we redo the analysis, use the whole genome sequencing data. So this is our justification why we're doing this kind of research. We are in hope we can have, like I say, to allowing to create the high resolution genetic map, and hopefully we have the statistics power doing the GWAS. So therefore, we have our objective here is we want to reanalyze the US mini core collection to check the genetic structure. We also want to identify the SNP, and then also for the leaf spot, and then also for the dormancy. And we're looking for how we're doing this here. The slide showing you is how we're doing the phenotyping with knowing QTL identification. For the genotyping part, it's not any problem. Nowadays, it's very easy, very consistent. But the phenotyping is the bottleneck, is very challenging. So we're doing the leaf spot here, showing you in our university, the experiment station down to the south of the Alabama at the Headland Station. We like evaluation of 3-year and 3 replication. Here is this chart that tells you the, like I say, how the consistently. So I will tell you guys for leaf spot evaluation data, we got a very consistent. You can see the R square is 0.75 from year to year. So that's a good news for the breeder or doing the QTL. And here is showing you we're doing the seed germination and also the dormancy, especially dormancy. So after harvest, that lines is a fresh seed. So after drying, so we're doing some of the germination tests is in the closed chamber. So we assess the 3 different days. So like 7 days after planting, 14 days, also 21 days, based on the percentage of the seed germination, we can calculation the dormancy, also the germination. So here is what we're doing on the genotyping. We're doing genotyping, use the whole genome sequencing. So totally, we have a big data set, which is about 170 gigabytes. And then also each sample roughly about 2.5 GB. And then we also use that one trying to find SNP to see how many SNP we can discover from that big data set. Here is just showing you on the right side is the analysis workflow. So you can see basically it's like doing the genotyping, we have the population structure, and then we also combine those phenotypic data, and we doing the GWAS. After you identify the QTL, the next step is to mining the gene. We go to the gene bank and go to the the database, and we do the mining. Here is how we work on that. And this slide actually is showing. So what we find on your right side is the genetic linkage map. You can see totally what we found about the SNP number is 87,000. So you can see here there are distribution on the 20 chromosome, A genome and B genome. You can see the B9 has the longest genetic distance, also has the majority, has the most density. Also the like a label color is like a red color. You can see the 19 chromosome. But you also, of course, you also see some of the like AA, that's the shortest genetic linkage. So somehow, maybe for some reason, we're not able to cover that. But in general, we will see that B genome has a high SNP density compared to A. A genome, also the chromosome 19 will be a B9, has the highest density. So, and then next one, we are working on estimated LD decay. We're knowing if you do the GORs based on the theory of the dislinguage equivalent. So we were doing on the estimation of the LD decay. We found, you can see the figure on the red line. That's the turning point. That's the R squared equal to 0.1. Based on that, we do the calculation. The minimum required for the number of SNP for the GWAS is 44,000. But we have, in this study, we identified 87,000, which almost twice the number. So there will be a sufficient doing the GWAS. So here is the genetic structure we created. So on the right side, that is the for the phylogenetic tree, and you can see basically they classify by the three groups, group 1, 2, 3. So also group 1 mainly is from the hypergia, group 2 is fasciata, and group 3 is mixed. So on the left figure, that is principal component analysis. You can see those methods is pretty consistent. And here is some phenotyping. We do the ANOVA analysis, also do the honestly significant differences. On the right is the early leaf spot, other side is the later leaf spot. So those are three groups. So you can see that three groups based on the are significantly different. Normally, in general, group 1 has a higher disease, group 3 has lowest disease. And we also, these are doing the same thing for the seed dormancy. You from there, we have 7 day, 14 day, 21 day, also have a seed dormancy. You also can find they have you can find a significantly different among those three groups. So for the QTL, this table is showing you we got a SNP association with early leaf spot. Also, we find that 10 leaves association, 10 SNPs with the later leaf spot. And it's doesn't move here. The frozen. It's the frozen. What's going on here? Sorry. About that, yeah. So here is the slide showing you that's for the germination, also for the seed dormancy. We also found a bunch of the QTL. I not go detail, but here is the what we're doing mining the candidate gene. So you can see the spanning like 1 million base, and we go to the downstream, also the upstream. We're looking for the gene. For example, here we find on the gene on the B1, that is the gene. Then we against to the homologous gene in the apoptosis gene base. And we find that gene called the ATE1. So that is exhibit delay leaf resistant delay leaf synthesis and also respond to passaging. We're also doing the haplotype phenotype analysis. You can see the C, D, F, so and then C hepatides. That the two bar you can see is a significantly different. The C hepatipe showing the 13.3 decrease the resistance if you compare to the other one is ALT. And we do the same thing for the leaf spot here, late leaf spot. We find the one gene, significant gene we call the major gene, is the ERF34. They played a big role on the leaf synthesis. And we're also doing the same thing for the haploid phenotypic analysis here. You can see the three group here. So the CIF group has the smallest disease rating 4.0. If you compare those two, then that's another indication. So the gene is a very function. Then we can do the same thing for the seed dormancy, and we're also finding the gene on the B9, so that homologous with the gene called the ICE1. So that function is involved regulation the ABA signal, also the seed dormancy. So for that, I have a conclusion here. So basically, what we're doing is a pretty validation of our previous finding. So our result also supported with the published work. Also, like identify candidate gene for the leaf spot, also for the disease. Another most important for our community here is we identify the SNP or the 87,000 SNPs, which can be used make the chips for another genotyping. So for that, I will question my talk. Thank you for your attention. If you have any question, I will try to answer. Thank you.
Thank you very much. Right ahead of time. Any questions? Yes.
Marcus, you have identified for both these traits. That is the late leaf spot as well as the dormancy. Are those ready to be used by breeders now? And
yes, I think so. So actually, in my breeding program, we has been used that for the a leaf spot disease or early leaf spot for the screening. Yeah, so we already confirmed that.
Any other questions?
So for the more detail about that data, you can go to my publication on the TAG. Yeah.
Actually, I have one question, if I may.
Yes.
How did you measure the dormancy? Because there is a lot of variation in maturity between 90 days in this mini core collection, between 90 days to 170 days. So it's not have any effect on dormancy that you check?
Yes, that's a good question. Like I say, the mini core collection, they have roughly have 4 different botanical varieties, including the Fasciata and Hypogea. So we harvesting that based on the maturity. So that's a TTL job when we're doing that. So when we collect that data, we harvesting that PIs, and then we just go putting that in the refrigerator until we harvesting all the line, and we doing the dormancy evaluation. Also, we are check it based on the maturity. We check the stain maturity. So it's easy to just scratch the shell. So based on the color, you will know that's mature or unmature.
Okay, thank you. Thank you very much.
Mhm.
I think we don't have so much time. You know, dinner is on the way. Okay, so the next talk, as much as I understand, is a recorded one. It's from Dr. GOA Lee. He's from Shandong Academy of Agricultural Science. And the talk is going to be about genomic and functional genomic insight into key agronomic traits of peanut. After 20 minutes, we just shut down the talk.
Shandong provincial crop germplasm resource also maintained more than 8,000 germplasm resources. Genome cultivated peanut was issued just in 2019, and EG is exploring for important agronomic traits remain at the elemental level. Very few excellent germplasm resources that can be utilized, resulting in a narrow genetic foundation for breeding variety and fewer rates of variety. The Green Revolution has had limited impact on peanuts, highlighting an urgent need to explore genes related to plant problems, such as and the efficiency of nitrogen fertilizers. The Shandong Provincial Crop Germplasm Resource Bank preserves more than 8,000 germplasm resources, with 635 core germplasm resources from 63 countries, including 121 accessions collected from China, 97 accessions from India, 68 accessions from Brazil, 59 accessions from Argentina, 53 accessions from Nigeria, 49 accessions from the United States, 28 accessions from Sudan, and other peanut-producing countries, including all 6 varieties of cultivated peanuts, including 199 variety fastigiata, 197 variety hypogaea, 80 variety hirsuta, 47 variety vulgaris, 35 variety peruviana, and 2 variety equatoriana. In previous studies, about 7 terabases of high-quality reeds were produced of 203 accessions, with an average sequence depth of 14-fold for each accession. And totally, more than 6 million of high-quality SNPs were identified. About 50% of them locate in intergenic regions, about 10% locate in genic regions. And 25 wild accessions were resequenced by 10-fold depth were used as outgroup. Using these SNP data, we constructed a phylogenetic tree with the wild species as outgroup reference. The results showed that variety peruviana accessions initially split from the wild species at the root, and followed by variety hypogaea plus variety hirsuta, then the variety vulgaris and variety fastigiata. In admixture results, when K equals 4, the accessions were clustered into 4 groups. Most accessions of variety peruviana in group 1, hypogaea plus variety hirsuta is in group 2, variety vulgaris in group 3, and variety fastigiata in group 4. The PCA analysis is consistent with this result. Further, when K equals 5, group 2 and group 3 were clearly diverged from each other. Further, we analyzed the genetic diversity and population differentiation. The results showed that accessions from South America have the highest psi value, which is much higher than those from Africa and Asia. The diversity of the accessions from Africa is higher than those from Asia, which is consistent with the supposed migration route from South America to Africa and then to Asia. In groups, variety peruviana has the highest diversity, which is consistent that it was divergent earlier than the others. Variety vulgaris and variety hypogaea have lower genetic diversity, perhaps due to most of them are cultivars. For population differentiation, variety peruviana has higher FST value with the other 3 groups. And the genetic distance between variety vulgaris and hypogaea is lowest. The LD decay rates were the highest in group 2, followed by group 3 and group 1, and the lowest in group 4. Using the current populations, we investigated the demographic history of peanut. The effective population size declined at about 10,000 years ago and to the minimum at about 1,000 years ago. The effective population expanded around 300 to 500 years ago, coinciding with the European colonial period, when peanuts spread worldwide. Furthermore, variety peruviana was predicted to diverge 11,000 years ago, and variety vulgaris emerged about 9,000 years ago, and finally, variety fastigiata and Hypogea separated about 3,600 years ago. Like other crops, the cultivated peanut has also undergone continuous selections due to domestication and intensive breeding. Firstly, we estimated the genetic diversity of landraces and cultivars. As expected, the genetics diversity of the landraces is higher than that in the cultivars. And the LD decay is higher in the landraces. These results indicated that the cultivars have experienced more artificial selections than the landraces. Further, we identified the selection signals and candidate genes involved in cultivar breeding. Totally, more than 3,000 genes in 637 sweep regions were identified. Among these genes, response to auxin and nitrogen catabolism are among the top 3 GO terms. And a lot of QTLs and genes related to yield were identified. For instance, the cell number regulator, auxin response factor homologs, which have been proved to play a role in seed size regulation in plants. Phenotypic platforms have been proved as high efficiency technique to extract the crop phenotypes. We have established greenhouse and field crop drone phenotypic platforms. We used multi-view RGB images and 3D point cloud reconstruction methods to obtain three-dimensional morphological phenotype data of individual peanuts. Single plant phenotyping in greenhouse, plant height, branch number, lateral branch length, and flowering type can be recorded. Also, it can be used for seedling traits analyzed for early vigor and stress tolerance. We have investigated the phenotypic data such as plant height, branch number, lateral branch length, and inflorescence of the main stem for more than 600 core germplasms. Under field conditions, the phenotype acquisition mounted on drones, including 4 key devices. RGB camera captures image texture, measuring canopy width and canopy area. Multispectral camera provides spectral info, analyzing biomass, nitrogen accumulation, and leaf color. Hyperspectral camera offers detailed insights into canopy structure, chlorophyll content, and pest management. LiDAR generates 3D structure data, measuring canopy height and canopy cube. These technologies enable precise and rapid phenotype data collection, supporting crop monitoring, precision agriculture, and breeding research. For example, for the biomass data acquisition, we first constructed mathematics models. 4 machine learning models were tested, all showing good performance. Among them, random, forest, and K-nearest neighbors performed the best, achieving higher accuracy in biomass prediction. The scatter plots further illustrate the strong correlation between predicted and actual values, highlighting the potential of these models for efficient crop monitoring and breeding research. Based on multispectral data, biomass information for hundreds or even thousands of field-grown peanut samples can be efficiently obtained. Another example of advanced field phenotyping is plant height measurement using LiDAR. This technology enables time series acquisition of plant height, allowing us to monitor crop growth dynamically. In Section A, we can see a series of 3D LiDAR-based height maps taken at different growth stages, providing a detailed visualization of plant structural changes over time. Graph B shows a strong correlation, our square very close to 1 between UAV-based LiDAR plant height measurements and manually measured values, demonstrating the high accuracy of this method. In C, the plant height distribution across different days after sowing is presented, revealing growth patterns over time. Finally, D illustrates individual plant height trajectories, showing the continuous increase in plant height across the growing period. By leveraging LiDAR technology, we can achieve high throughput, precise, and non-destructive monitoring of plant height, which is crucial for breeding programs, agronomic research. and precision agriculture. This method significantly enhances efficiency compared to traditional field measurements, providing valuable insights into crop development. Based on the germplasms, genotype, and phenotype data, a preliminary bioinformatics database was built. With the database, we can do gene function queries, sequence alignment, and phenotype-genotype association analysis. Using a well-structured database, we can efficiently conduct genotype-phenotype association analysis, enabling a deeper understanding of genetic contributions to key agronomic traits. Based on our preliminary experiments, we have successfully identified a large number of QTLs and superior haplotypes associated with critical traits. These findings provide valuable genetic resources for breeding programs, facilitating the selection of elite lines with enhanced yield, quality, and stress tolerance. We employed GWAS to screen for candidate genes related to seed size and seed weight regulation. Higher association signals with seed length on chromosome 15 at 137 megabase, where there is a G to a SNP on the promoter of DPB2 gene. The seed length of accessions with haplotype G was significantly higher than that of accessions with haplotype A. We randomly selected 5 large-seeded and 5 small-seeded accessions. The RNA-seq data showed that at the seed 3 stage, DPB2 expression is higher in longer seed accessions. And the quantitative real-time PCR confirmed this data. The GWAS analysis showed strong associated signals with seed weight on chromosome 16, where there are 8 genes. This region is overlapped with the selective sweep signal, where includes several reported QTLs for seed weight. Among them, we found one SNP in the last exon of the AFAX1, which makes an amino acid and gene 3D structure change. The seed weight of the accession with G haplotype have a lower than that with haplotype A. The gene is highly expressed in leaf, and its expression increased with seed development. When expressed in Arabidopsis, seed weight of lines with G haplotype increased compared to that of COL0, and the seed weight of line with a haplotype is even higher. Interestingly, GWAS signals also show that AFAX1 plays a role in biomass regulation. Haplotype analysis revealed that HAP2 exhibiting a significantly higher shoot dry weight compared to HAPY. Additionally, transgenic experiments demonstrated that overexpression of AFAX1 positively influences biomass traits, including rosette dry weight, root dry weight, and total dry weight, under both normal and low nitrogen conditions. These findings confirm that AFAX1 is a key genetic factor in biomass regulation, providing valuable insights for peanut improvement and breeding strategies. Prospects. These advancements in genomics, phenotyping, and molecular breeding are shaping the future of agricultural science, driving innovation and efficiency in crop improvement. I think we should pay more attention to the following 3 points. Firstly, genomics as a foundation for functional genomic studies. High-quality genome sequencing and pangenome analysis provide a solid foundation for functional studies. These advancements enable a deeper understanding of gene functions and molecular mechanisms, facilitating targeted crop improvement. Secondly, precise phenotyping of key agronomic traits. Well-genotyped germplasm ensures research accuracy and reproducibility. Advanced phenotyping techniques, such as high-throughput platforms and remote sensing, improve efficiency and precision. High-efficiency transformation systems still lack, which can accelerate application of molecular breeding techniques. Modern molecular breeding technologies such as 5G, moss breeding integrate into traditional breeding method will enhance breeding efficiency. Green evolution lacked in peanut. To cultivate dwarf or half-dwarf varieties, high nitrogen efficiency lines will sustainable agricultural development in peanut. Finally, I want to extend my heartfelt thanks to all our colleagues from SAS, BGI Qingdao, and ICRISTAT who contributed to this work. A special appreciation goes to Professor Juan Shovo and Professor Varshne Rajiv for their invaluable support. I'd also like to acknowledge the generous funding from the national and provincial governments, as well as our own academy. Thank you all for your attention.
Any questions? Actually, I like the AI English speaker. I consider using it tomorrow morning in my talk over there. So the next speaker, I think it's online, right? This is Dr. Josh Clevenger, known from Hudson Alpha Institute in Alabama rules. His talk is going to be, is on? Okay. Accelerating breeding using custom pangenome graphs.
But here it's in the morning on Saturday, and going to be presenting to you from my home. Going to be presenting to you from my home. I wasn't able to be able to join you. I'm very sad that I wasn't able to be able to join you. Very much looking forward to your beautiful country. I was very much looking forward to visiting. Forming new collaborations with you and Alabama, where I do my research. And I was excited to visit GOA and to have the next generation of AAGB with you. But because of technology, I get to present to you, and I hope that you enjoy sort of learning about the work that we're doing here at Hudson Alpha to apply pangenomics for gene discovery and peanut improvement. And I'm really excited to share this work with you today. One of the things about peanut, which is great, is that it's very sort of straightforward to develop new genomes. So that was the thing a few years ago that I recognized, that because the peanut, cultivated peanut in general, is so similar to itself and to other varieties, that you could develop pangenome graphs very easily. When the PacBio Revio hi-fi sequencer came out, the throughput allowed to sequence a peanut genome on one flow cell, which made the cost around $2,000 US. And so now you can think about projects where you're developing pangenomes locally for important populations at a very affordable cost. And so we started doing this with many different projects, with some really great projects I'll talk about in collaboration with Baju Guo and his PhD student, Ethan Thompson, but also in collaboration with Peggy Ozias Akins and Justin Vaughn, with a very large magic population. And Samir Pokhrel, who is leading that project, is there with you. And so I encourage you to speak with him about it. And I think he's going to present as well. So another thing that's pretty unfortunate for us is that peanut high molecular weight DNA is straightforward to attain. And so you can easily and cost effectively get the DNA from peanut leaves and sequence it, and then construct a really great pangenome graph out of it. And to utilize and deploy those graphs, we developed a pipeline that we're publishing here in the next couple of weeks called Kufu Pan. Essentially, it is thinking about this process and how do we leverage it. So essentially, what I talked about is if you have a population, a magic population, or even a biparental population, you develop that pangenome graph of that sort of those founder parents with hi-fi sequencing, and then putting it together into a pangenome graph. And then the population that you're using to either make selections in, or to try and map or discover genes for editing targets or for marker development, you can easily sort of attain DNA from the seeds, run skim-seq on that population at a very low cost, and then utilize that low coverage whole genome sequencing on the founder pangenome to really unlock a lot of great insights. And, you know, we developed this whole low coverage WGS pipeline we call it KUFU. We call it KUFU because if you do QTL-seq and you get a really great hit like this is in watermelon, it actually looks like KUFU pyramid. And so Waleed at the top there is the one that sort of named the pipeline, and him and I worked on this pretty extensively 5 years ago. We continue to make improvements on it, and we're publishing in a few weeks as well a KUFU environment, which is 133 functions to do really high level genomics research with NGS data. Really excited about that. So this is really the main KUFU team. Bethany at the top is our sort of coordinator and organizational specialist, project manager. Zach Myers is Waleed's protege and does a lot of the analysis. And Catherine next to him. Hallie Wright on the end in that second row there is the one who's taking in the genomes and creating graphs and making sure the genomes are in good shape. And then on the bottom, Kelly Good, Patricia, and Abriel are specialists in the lab. We can, from tissue to library preparation, process about 15 plates of 96 wells, so almost around 1,400 samples every single week. And we're sequencing on full flow cells in NovaSeq X at this moment once a week, but our goal is to do it about every 2 weeks. So you can see that we have a situation where we have really grand access and quick access to high levels of genomic information. Yeah, just to go through that process and just to show you what we've like put into place. You know, we're getting DNA from seed chips, so we're able to genotype all of the seeds before we plant them. We prepare flex prep libraries from Twist. This is a new library prep that's cost-effective, fast. Has great complexity. It's a wonderful, wonderful prep. It auto-normalizes, and so after you extract DNA, you don't have to normalize the plates. You can go straight into library preparation. That has saved us in a very large amount of time and effort and labor costs. We do the low-pass sequencing. That's a picture of a NovaSeq 6K we've upgraded since then. And then we get to the core mapping and filtering. So this is where sort of utilizing the graph comes in. We can take that skim-seq of the population and map it onto the graph, and then do all sorts of things that we might want to do. We do a lot of QTL-seq, of course, because we're trying to help with marker development, but also, you know, genome-wide genotypes, and then those can be plugged into selection schemes, imputed very simply. And so this you have someone else help you with. There's nothing wrong with that. But for our sort of organization, we want to be completely vertically oriented. So if you collaborate with us and do some of these things, we can start from just accepting plates of seed chips and go to the very end of analysis, or stop anywhere in between. And a lot of that helps with the price, because we end up sort of buying large amounts of reagents, so we get large discounts. And also the fact that we've spent a very, very substantial amount of time optimizing each part of this process. It's not trivial to sort of get good sequence out of each individual when you're looking at thousands of them, especially with seed chips or small amounts of tissue. And so we're really excited about where we're at right now and the abilities to exploit genomic data, which was our goal. So I wanted to show you some graphs of what the Flex Prep library prep looks like. It's pretty phenomenal. This graph shows sort of each sort of vertical line is a plate, and each grouping is a pool of 12. So the library prep happens in 8 pools of 12 in a plate. You can see this is sequencing yield. You can see across that pretty much all the samples have been sequenced almost like within a few million reads of each other, which is incredible. None of these plates were normalized at all. And this represents a lot of different stuff. It represents peanuts, pumpkins, cannabis, and as well as like microbiome for Jake Fountain. That's just soil extracted DNA to look at microbial enrichment. So a lot of different things that you do. And then this graph actually shows the concentration of the different pools of libraries on the bottom. So actually, this is indicating on the left, we have the green samples, which were dandelion, extremely low input for those dandelion samples. But still, we are able to get good library preparation and sequencing. So really, really powerful prep that if you're going to do this type of work, I highly recommend sort of the Flex Prep for your workflow. So, you know, I want to highlight some of Ethan Thompson's work because it's really easy to highlight. This is a paper that he has in review right now. It's got really good revisions. She collaborated with Rajiv Varshney and his group on this as well. So he had a population where he was essentially using the high oleic acid trait as a control, and he mapped it onto a linear genome, and it worked out as expected. But then when he mapped it onto his 8-parent genome graph, he actually saw part of the peak. And when he went in and looked at it closer, he identified a third fatty acid desaturase gene, and then went on to show that mutations in that gene are also needed to have the pure high oleic acid trait, which solves a major industry problem that wouldn't have been possible to probably identify without using pangenome graphs. Really exceptional work, Ethan. And then also we have problems with tomato spotted wilt virus here in the United States, which I don't think is prevalent in other parts of the world, but it's really important here. And Baoju GOA has been working very hard to map sort of this resistance for a long time. But when we applied pangenomics, it very clearly pulled out a 63 kb insertion, which had a duplicated gene cassette, and that really is the driver of resistance. And so we quickly mapped and identified the causative functional variation for this important trait, of which we couldn't do before with a single reference genome. It's really powerful and impactful insights that are already being sort of used in breeding programs. It's just really great work. And because I'm sort of insatiable about, you know, what's next and what more can I do, we looked at the throughput of the Revio, and this is really, you know, a lot of the work Kendall Lee, the postdoc in my lab, has been doing. And we said, well, can we just do low-pass long-read sequencing as a genotyping tool? And so we looked into that, made a really nice study about what that looks like. And I'm going to show you some outputs from that. Presented that at PAG this year and got a lot of really good press from Pacific Biosciences. So really sort of a lot of people are excited about this new approach. It's really simple. You know, the biggest thing is, can we extract high molecular weight DNA in 96 well format? We have special buffers that we're able to do that. Do some cleanups with a Thermo Fisher Kingfisher, and then use the high-5-plex library construction, which allows multiplexing up to 96. And then we load those on the Revio, and we sequence 1 to 2x coverage, which makes the per sample cost in the range of how much I've seen people pay for exome sequencing. So long read low pass covers more gene space than short reads with the same yield. So of course we sequence everything with short reads. So we had a lot of samples. We resampled them and did the long read low pass. That's in the pink there, and the gray is the short read. And you're graphing the depth of coverage. So how much coverage did we get off the sequencer on the X with the percent of the genome covered on the Y. In this case, it's gene space. So really, when you get up to about 2x coverage, you're getting above 75%. And when you get closer to 4x coverage, you're almost covering 100% of the gene space. It's a really wonderful tool to unlock insights. The other thing that's really interesting is, you can use your pangenome graph to sort of genotype your populations with the long read low pass. Here, what we're showing is on 3 categories of variation. The pink is showing, the pink box plots are showing long read low pass samples, and how many different variants are we physically calling from the data. So we're not imputing, it's not missing data, but it's a physical, there was a read here, we made a call based on our filtering. So it's not just really that you are physically calling an exponentially more number of SNPs, but also if we identify indels as below 1,000 base pairs, you are calling exponentially more number of indels. And then your SVs, which are your 1,000 to however large they are, like the TSWV structural variant I talked about earlier, you're physically calling a lot more of those as well. And if you look at the sort of missing data in terms of the per sample missing data in the data set, it's not significantly different between long reads and short reads. So you're essentially by a factor of 100 physically calling more variation that have the ability to impact your population, your outcome, without impacting your resolution in terms of the missing data and how much you need to impute, with the same amount of base pairs sequenced. Just like an incredible increase in power and information in your data set. So we're really excited about using this in so many different projects. We're so excited about it that we formed a company called Veil Genomics. If you're interested in using this in your program, and I hope you are, please visit us at veilgenomics.com. There's a QR code there and emails where you can reach out to us and start talking to us about projects. I think this is the future of genotyping, and it's just a really exciting time to be working in this space. So for me as a breeder, what I wanted to see was, can I identify my impactful QTL more effectively? The answer is yes. So on the top, this is like selecting for leaf spot integressions and that insertion for TSWV with lines where they should be there. And you can see that the pink boxes are indicating a higher accuracy of selection, and that's your long read. So more information, higher resolution, higher fidelity, you're identifying your important sites at a much better clip. So if I sort of summarize what I've just talked about today, pangenomics have the ability to really amplify in a big way sort of insights enabled by genomics, excited about their use in local populations and not just sort of big diversity pangenome graphs. Peanut is a great system to maximize pangenomics because cultivated peanut genomes are very similar to each other, so the graphs are not very complex. And then if we overlay this new technology of scaled long read sequencing, it's exponentially more efficient at gene discovery and selection. And so just really, really powerful stuff. I'll end there. And my email is jclevenger@hudsonalpha.org. If you have questions, please email me, reach out to me. If you would like to collaborate, I would love to collaborate on peanut projects. We collaborate with many folks around the world. It's easy to send seed chips to us. It's not difficult. And so I would welcome everybody that is interested to reach out to me. These are funding agencies that fund a lot of the work that we do in the state of Alabama, United States, and private entities, as well as our fee-for-service operation. So thank you very much. And I hope the AAGB conference is incredible and that you all become more excited about peanut research than you did before. So thank you very much.
Okay. So actually, this one was also pre-recorded, and not as I thought. So with not further ado, we can go to the next one, which is also pre-recorded. Yes. Okay, and this is from Dr. Lu Hong from CRI GAAS China, and it's about advancing peanut evolution and breeding potential with complete genomes.
Hello, everyone. Greetings from Guangzhou. It's a great honor to be speaking at AAGB today. Unfortunately, due to visa issues, my colleagues and I are unable to come in person, so I hope I can make myself clear to this video. Allow me to introduce myself. My name is Lu Huang from the Crop Research Institute at the Guangdong Academy of Agricultural Sciences in Guangzhou. Today, I will be presenting some of our recent work, focusing on the utilizing genomics and population genomic approaches to unravel the evolution, population history, and the genetic basis of agronomic traits in peanut. First, let me present a map of global peanut production. According to the FAO statistics from 2018 to 2022, the total cultivation area spent about 29 million hectares, yielding an annual production of 53 million tons per year. And currently, peanuts are mainly cultivated in more than 100 countries, with China and India stand out as the major players in the global peanut production, contributing to half or more than half of the world's peanut output. And the peanut industry currently faces several pressing challenges that demand attention, such as extreme climate change, pest infestations, and diseases. In recent years, more and more breeders are applying molecular tools, such as genomics, transcriptomics, and proteomics, together with conventional field studies to address this issue. And recent advancements in peanut genomics have demonstrated that genomics can serve as an effective strategy for accelerating the genetic enhancement of peanuts. By using high-throughput sequencing, we can conduct de novo sequencing of individual genes and resequencing of populations and do GWAS and QTL analysis. By integrating data from multiple omic platforms, such as transcriptomics, proteomics, and metabolomics, we can collectively identify candidate genes associated with target traits and validate them through reverse genetics. Subsequently, breeding methods such as MAS, GS, and genetic modification can be applied for the breeding of new varieties. So it is evident that as the start point of this framework, a well-constructed reference genome is of key importance for further advancement. And with so many years technical development and the first launching of human genomic projects since 1990s, the genomes of a growing number of species have been decoded, including crops. First rice, then wheat, corn, and cotton, and finally comes to our turn. We can see during the last 2 decades, the application of new sequencing technology have enabled researchers to get more genomes and even pan-genomes at a higher throughput and higher accuracy, especially in recent years. And in the first part, I would like to reveal our research on decoding the peanut reference genomes and works based on it. And actually, a public accessible reference genome of peanut is not available until 2019. Well, one is due to its large genome size. It's 2.8 gigabyte, way larger than Rhodopsis and rice. And it's more than twice the size when compared to soybean. And moreover, the previously widely used short-read-based sequencing technologies have certain shortcomings on assembling genomes, especially to those with high content of repeats. And since 2016, with the help of updated sequencing technologies, we initiated the sequencing project of peanut genome. First, the AA genome progenitor, then the B genome progenitor, and finally, we reported the cultivated peanut genome of a variety of, with the name of Fuhuasheng, which is widely used in in breeding practices in China. The genome of cultivated peanut generally shows that some characteristics of its A genome and B genome progenitors with some modifications. And we identified a total of 83,000 genes in the cultivated peanut genome. After that, we tried to take a look at the genetic mechanism of several key traits in peanuts, including geocarpy, peanut allergen, nitrogen fixation, and oil metabolism. For example, in the genome sequence, we identified genes that play key roles in geocarpy that are related to gravity sensing, signal transduction, and organ response. And we identified 69 allergens or hormones, and 12 of which are newly identified. Similarly, the genes that's related to nitrogen fixation and oil metabolism is also being studied based on sequenced genome. And based on this progress, we hope to move a step further, that is to obtain the complete genome of peanut and to mark all the genes based on high-quality consensus genome sequence. So we carried out on the development of Fuhuasheng version 2, one of the most likely. So, which is also the one we use for the sequencing of the first cultivation genome. After the process of massive sequencing, error correction, primary assembly, IC-based assembly, and polishing, we are able to obtain Fuhuasheng version 2. And the T2T Fuhuasheng genome was generated utilizing high-fi reads, ONT optional reads, and high-C short reads. And finally, the genome size reached 2.5857 gigabyte of sequence with 0 gaps. And the centromere sequence of all 20 chromosomes are also resolved. In the updated genome, we added around 20 megabytes of new sequences in the new version, and it's of much higher quality and completeness, and all the previous gaps have been closed. And through the comparison with the genome of wild progenitors, we can see that there are higher concordance between B subgenomes than between A subgenomes, which indicates that a symmetric evolution of peanut subgenome. After these sequencing efforts, we need to think about what's next after an updated rough genome. We should admit that the power of a single genome is limited, and only when we have a variation map or the genome information of enough sample, we are able to uncover the structure and history of populations, as well as to uncover the mechanism and phenotypes. So as the second part, we will report some resequencing-based peanut population analysis in China. And in this part, a total of 394 germplasms from 15 countries were collected and sequenced. Among them, 261 accessions are from China. We can see from the bar plot that the populations from China, the red bars, shows that most of them are from two latitudes, which is from North China, NC, and South China, SC. We constructed a high-density genetic variation map of 8.8 million SNPs. We can see that the SC, NC, and other samples out of China, they form clear population structure. And then we use this data to decode the population history of peanut in China. According to the literature and molecular data, we came up with 8 candidate migration routes. After analyzed by using approximate Bayesian computation, the scenario 5 came out as the most likely one to present the population history in China, which indicates that the NC represents the populations that come from North China, and the SC population, they are more likely to derive from South and Southeast Asia. These two major propagation routes have led to 2 major peanut cultivation centers in China, which explained why peanuts from North and South China have so many differences. This part of work was majorly finished by Dr. Lu in 2024. It was published in Nature Genetics. And another part of our work, we studied the section of pod maturity traits based on the population genomics. And in China, the early maturity breeding is one of the great importance. By using early maturity varieties, we can increase multi-cropping index of farmlands and optimize land utilization in regions with less frost-free period. The maturity of peanut pod is difficult to evaluate. We all know that, because you can only get that information after you pull it out of the ground. And in order to get a precise measurement, you need to do more than that. So in our study, we borrowed the method from literature and made some changes. After the exocarp removed by pressure washing, we checked the color of mesocarp. The darker it was, the higher level of maturity it get. And we evaluated the maturity index of all our 319 sessions. And the left, the middle, and the right plot represent the maturity index, the ratio of mature pods, and the average grade value of the pods of the 2 seasons. And then through the GWAS analysis, we identified the signals from several chromosomes. From chromosome 1, we identified a candidate gene, EBF1, which gets 2 significant variations. One is in its promoter, the other is in its CDS. And the population was separated into 3 haplotypes with distinct phenotypes, especially grouping by the promoter variation. And from transcriptome data, we can see that the expression of EBF1 is mainly in pod and flower. And 2 typical varieties show that its expression is significantly higher in B5. which is the late maturity variety. So this shows that the EBF1 generally exhibits the negative regulation of early maturity traits. And further research on transgenic plant and molecular mechanisms is currently underway. Okay, here's the part of a summary. And first, we have successfully sequenced the complete genome assembly of cultivated peanut, the food fashion version 2. And the second part, we propose two major propagation routes of peanut to China, which leads to two major peanut cultivation centers in China. And the third is that we identified the EBF1 as the negative regulator of early maturation trait in peanuts. And I would like to express my gratitude to all my colleagues and their dedication and hard work, and all collaborators for their invaluable contributions to this work. And a special thanks to Professor Pandy for providing us such a valuable platform for academic exchange. And I would also thank all the experts and scholars present here today for your valuable time and attention. Thank you, and welcome to visit Guangzhou.
Okay, thank you. So thanks God, the last speaker is here, as even one of our hosts, Sunil Gungaude from ICRISAT, some affiliation with UGA, he will explain. He will speak also about pangenomes, but not of peanut pangenomes, but for Aspergillus flavus pangenome. Please.
Hello. Yeah. So, very good evening, all. And I think after a long stretch of online presentation, I think this would be a very refreshing moment for you all that in person I'll be presenting on this aflapan. So, aflapan is Aspergillus flavus pangenome, which is developed for the Aspergillus flavus diversity in United States. So this is the part of my postdoc work when I was doing postdoc in University of Georgia. So today in the morning, so Dr. Guo presented the history and significance of AAGB. So I did my postdoc with Dr. Guo in University of Georgia and USDA in Tipton campus. So as we all know that Aspergillus flavus contamination is a very alarming issue and affecting many crop commodities in agriculture. In US, if you will see this green corn belt, so this is highly susceptible to the Aspergillus flavus contamination, which is causing around 52.18 billion losses annually. So what kind of losses it is causing? It's direct crop losses. It's also reducing the market value of the crop, and then trade barrier, as there are very certain strict levels of the aflatoxin. For example, in US, only 20 ppb is allowed, and UK it's only 5 ppb. If a lot is having 6 ppb, even they will reject the lot and they will not consider it for the market. Infected, it will go through the food chain and it will go to the directly human body through the food chain. So what kind of crops generally this aflatoxin is infecting, or Aspergillus flavus is infecting, is chili, then sunflower, soybean, sorghum and millets, peanut, maize, cotton, rice and wheat. So these are the major crop commodities. Among them, corn and peanut are very susceptible one. I think it's because the oil content is the major carrier for this one, or supporting agent in this crop grains. Then I'll talk about the Aspergillus flavus, what this species is. So Aspergillus flavus belongs to the section Flavi, which splits into 8 clades, and currently it contains 33 species. So there are 2 species produces aflatoxin B1 and B2, and 14 species can produce B1, B2, G1, and G2 toxins. There are 2 sclerotia types, S sclerotia type and L sclerotia type. S means short sclerotia and L means large sclerotia. So S will probably produce, a large number of toxin than the L types. And 2 mating types are there, MAT1-1 and MAT1-2. Then some are toxigenic and some are non-toxigenic isolates. Even people, they are using the non-toxic to mitigate the aflatoxin contamination. A flavors and A oryzae are 99% similar, but the A flavors is highly toxigenic, and then A parasiticus, A oryzae is used in the fermentation industries in the food crops. So even there is a single 1% is difference in the isolate. So another where Aspergillus oryzae is just 1% different than Aspergillus flavors, but we are using it into the food industries. Then what are the key questions from my postdoc work we try to address is how to understand the different toxin producing gene clusters among various isolates. Can we identify a core set of toxin producing genes across the isolates as a target for genome editing? What are the structural variations between toxigenic and non-toxigenic isolates? Then whether geographical origin of the isolates playing any important role in boosting the toxigenicity. So these are the key questions we designed before starting this project. Then we thought that single reference genome we have for Aspergillus flavors is NRRL 3357. So a single reference genome is not really explains the entire diversity of the species. So because we know that none of the 2 genomes are similar, we have like 120 people sitting in the hall, everyone is different. Even 1% difference at the genome level makes phenotypically a lot of changes. Even genome has specific feature that makes it special. A single genome cannot explain complete variability of a species. So for capturing maximum variability of a species, we need to go beyond single reference genome. And pangenome and super pangenomes are the new techniques to represent the variability of a species or a genus respectively. NRRL 3357 is currently reference genome available in NCBI. Where both of these reference genome. NRRL 3357 produces moderate level of toxin, but AF13 produces large number of toxin. So this difference in the toxin production, we thought at genome level whether there is the difference. So we observed that there is 310 kb insertion in AF13 that makes it highly aggressive for the toxin production. So we thought that if we will sequence large number of isolates, maybe there are some more toxin producing genes present in different isolates, and maybe that are the causal agents in infection of this particular. From the human pangenome, recently we published in 2023 that they develop a pangenome from the diverse human species from different parts of the world, and they develop a single reference genome that they call as the human pangenome. And therefore, from the 12 states of United States, we collected the isolates from different fields. So around 346 isolates we collected from, for example, California, then Arizona is there, Texas, Oklahoma, Iowa, then Mississippi State, Louisiana, Georgia, then Florida, and North Carolina, and penicillins from corn itself, single, from different infected parts, for example, tassel, leaf, and then cob, and then stem. And we collected around 164 isolates from soils from different cropping systems. Then we sequenced all these 346 isolates at 15x to 20x depth. And we use AF13 as a reference genome because AF13 is a highly toxigenic isolate. So we consider it as a reference genome here. To confirm whether this is really Aspergillus flavors, we use some marker genes, not flavors. And in our study, we also identified 36 isolates as a finally 225 isolates from present study and 120 of the Aspergillus flavors. And these are the stats for the genome assemblies, we developed the complete genome assemblies for 346 isolates. So genome sizes were ranging from 37 to 43 MBs. Then we also checked for the gaps in the assemblies. Then average context size we checked. Then N50 and effect of gaps on the scaffold size we checked. So all these parameters were accessed to check the quality of the assemblies used in this pangenome analysis. So the population structure analysis we performed for 346 isolates using 1.03 million SNPs. We observed clear four genetic populations present in these 346 isolates from 12 states. And each population has a different linkage disequilibrium decay, and therefore there is a different levels of genetic variability present in each of the group of these isolates. Then we check the assembly stats. For example, the genome size, as you can see, the minimum size we observe is 36.51 MB to 43.2. The large genome sizes were observed for the soil isolates, as you can see in the bar graph. This is not. As you can see in the bar graphs, the gray color bar is for soil and the green color is for corn. The soil isolates were having the larger genomes, maybe because they are in the soil and they have to compete with other microbes and they acquire some genetic changes, and the genome size is increased for the soil isolates. Then this is the very complex structure where we try to understand the diversity of the isolates based on their origin, their source, Sclerotia types, then their mating types, and their toxigenicity and non-toxigenicity. So I'll not go into detail because this is just a diversity of the isolates, the 346 isolates from 12 states. Then we did a genome annotation for all the 346 isolates and a core set of genes, like total 7,855 unique genes were identified across the 346 isolates, and we classified them into core, soft core, shell, and cloud genome. For example, in core means all 17,855 genes are present in all 346 isolates. So that part we called it as a core genome. So like that, there are 7,315 genes were present in all the isolates. So that part we call it as a core genome. Then we had soft core, in which we had 3,887 genes, which is present in less than 100% isolates, but more than or equal to 95% of the isolates. Then there was another part of the genome, that is shell genome, where the shell genome means it is present in less than 95% of the isolates, but greater than 5% of the isolates. So there are 3,132 genes were part of shell genome. Then the cloud groups. Cloud means you see in the sky there will be clouds. Someone is here, and then some other one will be somewhere. So that means there are some genes which were very unique to particular isolates. So like that, we identified around 3,521 genes very uniquely present in few isolates. So in total, 5,994 orthogroups were identified as new genes which were not present in the current isolates. So there are two reference genome, NRRL 3357 and AF13, in which these 5,994 orthogroups were absent. So this 5,994 orthogroups we called as a novel genes when we compared with AF13 reference genome. Then this is the APLAPAN genome. For instance, there are total 7,855 genes, and around 59.1% of these 17,000 were we converted into or we classified them into accessory genome. So the accessory genome consists of singletons, cloud, shell, and soft core, and the core is 40.9%. So there is a large number of accessory genes we identified, which is having the more diverse accessions in this 346 isolates. Then we checked the number of gene clusters we identified in the aflapan with the existing genome assemblies. So the AF13 and NRL 3357 on an average having 13,000 genes, but the aflapan is having 17,855 genes. So the additional 5,900 genes we brought from the sequencing of these isolates, which is absent in the previous genome assemblies. Then we checked for the length of the protein in each core, soft core, shell, and cloud genome. So we observed that the highly conserved genes were present in the core genome, and therefore the protein length is high in the core, which is reduced to the soft core, shell, and cloud genomes. And we compared the pan genes and the core genes identified through these 346 isolates. And we observed that the maximum number of diversity we have captured, as you can see, the x-axis is having the number of isolates, and then y-axis is having the number of gene clusters. When you are adding the number of isolates, the core genes and pan genes, they are decreasing, and at the end there is no new addition of the pan genes or core genes after 50 isolates. So that means that we captured maximum diversity of this species through the pangenome of this Aspergillus flavus. So the number of novel gene clusters were decreased with the increased number of genomes after adding the 300 genome assemblies, and there is no increase in new gene clusters, indicate that the Aspergillus flavus is a closed pan genome after developing it. So what is closed pan genome and open pan genome? That is also another part of the study. The closed pan genome is the pan genome which is captured the entire diversity of the species, and the open pan genome means even after sequencing the large number of isolates, if still the new genes you are adding in the existing pan genome, then that means that pan genome is open pan genome, and there is a still scope to include the diversity in the pan genome. Then we also did pan-GWAS analysis to identify the novel aflatoxin producing gene clusters. So the 346 isolates were phenotyped for the pigment, then aflatoxin B1, B2, G1, and G2. And it's 17,000 genes, the present substance variations we used for the GWAS analysis with the help of the phenotypic data. And we observed that in the shell genome and the shell novel genome, we observed that the large number of gene clusters, those are associated with the toxin production in the Aspergillus flavus and in 346 isolates. So among the significantly associated genes, there were different toxin producing genes we identified. For example, AFLA-R, AFLA-S. AFLA-S is there, AFLA-T, AFLA-O, and AFLA-B. These are the reported toxin producing genes. And there are additional genes we have identified is cytochrome P450 monoxygenase, there is AFLA-V, and then satratoxins biosynthesis SC3 cluster protein, and sterol 3 beta glucosyl transferase. So these are the few key candidate genes we identified. Those are producing the toxin in this Aspergillus flavus. And this work was published last year in the BMC Plant Biology, which is called as the aflapan gene pan for the aflatoxin and secondary metabolite producing genes in Aspergillus flavus. So how these genes will be used for the mitigation of the aflatoxin contamination, that's another part of study. So now there's another technology now recently developed, that is spray-induced gene silencing, which is advanced version of RNAi technology, in which we have to develop the guide RNA, and that guide RNA will be converted into, will spray into the field, which will be silence the target gene. So now from our pangenome, there are four key candidate genes: A flavus alkaline protease, polygalacturanous P2C, then APLA-M and OMTA. These are the O-methyl transferase A. So these are the four targets we are now using for the post-induced gene silencing by using spray-induced gene silencing technology. So in summary, so we evaluated a total of 347 A flavus genomes. We observed that soil isolates have relatively longer genomes than the corn or the isolates collected from the plant parts. The long sclerotia type is conserved in soil isolates, while the short sclerotia types were very common in corn isolates. Among mating types, we observed that MAT1-1 is mostly observed in soil isolates, and the MAT1-2 is observed in corn isolates. We identified 4 different genetic groups by using the genomic variants. A total of 7,315 genes were identified as a core genome and 10,540 were identified as accessory genome, which are part of soft core shell and cloud genome. A total of 5,994 genes were identified as a novel orthologous genes clusters in accessory genome when compared with AF13 reference genome. So thank you very much.
Any questions? Okay, I'm asking, Howard is doing transformation in Aspergillus.
We are not doing any.
No, it's hard because you have many candidate genes. There is any way that you can see
So
there are
what?
real genes.
So
Because in
what?
peanut, it's very hard.
Yeah. So what we will do, for example, if there are some genes, we have to categorize. If there are really toxin producing gene, then we have to identify the conserved motif among all. For example, the group of 10 genes are there, and is there any conserved region? That conserved region will be used as a target in RNAi technology. So that's next part of this study. We are now using that conserved region into the silencing by using this spray-induced gene silencing. There we will use the nanoparticle to coat the double-stranded RNA and we will spray into the field.
Hello, can you hear?
Yes,
Yeah,
Dr.
you estimated the 3 LD decay. So my question is, what is the really mean for those 3 LD decay? Are we going to use that information?
So LDDK estimate, I just used to differentiate whether the LDDK estimate is different in each genetic group. So that LDDK estimate, I observed it is varying in each genetic group. So that LDDK I use just to differentiate the genetic populations, and there is no other significance of doing the LDDK in this study. It is just to differentiate yes, there are significantly different genetic groups are present in the population.
Okay, thank you. We have to finish now.
Oh.
Thank you very much. And thank you all for all the other speakers. And
Thank you all. Thank you, Ryan.
And thank you everybody for being patient and listening.
Ladies and gentlemen, we here wrap the sessions for the day. Thanking the chair and co-chair conducting the session and all the speakers for giving their presentation and the audience patiently waiting and completing all the talks here. Thank you once again. We'll close the day here and audience are requested to gather at the reception around 6:15 where the bus facility is provided to take us to the dinner venue. A quick heads up to the poster evaluation committee. The remaining posters which has not been evaluated, kindly wrap the poster evaluation within 15 to 20 minutes. And other posters which have been evaluated, requesting the presenters to remove the posters from the designated place. Looking forward for the dinner. Thank you one and all. With a round of applause, we close the day here and looking forward for you to join for the dinner. Thank you one and all. Please, everybody be on time around 6:15. If you miss the bus, then you miss the enjoyment and all the excitement we have planned for you.